Good morning. Welcome to the Biotechnology to Build a Brighter Future workshop. We have an exciting program for the day, organized in two main sessions, a morning session dedicated to biotech applications in food and health, and an afternoon session dedicated to biotech applications in environment. During the day, we will have keynote lectures, key project lectures, and flash presentations by outstanding international speakers coming from both academia and industry. Thank you all the speakers for accepting our invitation. Your contribution is key to our success. We will hear about diverse topics that range from probiotics, prebiotics, novel foods, synthetic biology, metagenomics, and circular economy. I also welcome our participants. We have about 350 participants coming from 28 different nationalities, 2/3 from academia and 1/3 from industry. I wish you all a fruitful workshop. Finally, some quick housekeeping rules and some guidelines. As you may have noticed, audios and videos are off. If you are a speaker, be sure to send us a private message on the chat so we can give you co-host permissions and you can share your screen. Questions will be placed on Slido, and we will put the link on the chat so the moderator will pick the most voted questions in the interest of time. Unanswered questions, they may be raised directly to the speakers on the chat. Also, please be aware that we will put a link also in the chat in each of the sessions, morning and afternoon sessions, that you should fill so we can send you later on the certificates. So any issue, please send the organizers a private message on the chat and we will try to help you. So we are identified by organizers and you can easily see us. Also, if for some reason there is a problem or a technical issue with some of the speakers, we will stop the session and we will wait for the time length of that presentation so we can keep the program as it is announced. Okay, so this being said, and without further delays, I will leave the floor to Joana Rodrigues. She will be moderating the morning session, and I wish you all a very nice day and enjoy. Thank you.
Hi everyone, good morning, welcome. My name is Joana and I will start now our session, Biotechnology Applications in Food and Health. Do not forget to ask questions to our speakers in Slido. Our first keynote speaker is Duarte Torres. Duarte, you can start to share your screen, please. Duarte Torres is an assistant professor at the Faculty of Nutrition and Food Sciences of the University of Porto. He works in the development of solutions to obtain healthier and sustainable foods. Duarte, you may start as soon as you are ready. Thank you.
Good morning, everyone. I hope you are seeing my screen.
Yes.
Thank you. So today I will talk about pulse-based foods in nutrition and health promotion. First of all, I would like to thank to the organization, to Ligia Rodrigues and to the team. Thank you very much for being here, for inviting me, and it's a pleasure to be here to talk about this topic that I've been researching in the last years. So without further delays, this topic, the pulse uses in food and nutrition and health promotion can be viewed in several perspectives because and I will try to cover them during my presentation because the health promotion of legumes and pulses as a food for humans and also for feed cannot be separated from the beneficial impact of the pulses in agriculture and in food production as a system. I will try to cover this. I will try to make a small definition of what is legumes and pulses. I will talk about the planetary boundaries that that we can use for measure our environmental impact and the environmental impact of food production. I will try to cover the most recent studies that study the effect of pulses in the intake of pulses in several health outcomes. The food in the Anthropocene, this is a framework that was developed in 2019, and this is like a guidance for a future health and sustainable diets, and how we are in this framework. So what is our adherence to this sustainable and healthy diets in Portugal and across the world? And then I will try to talk about how I see the role of pulses and pulses-based foods in these topics, and some concluding remarks. So pulses, as a definition, pulses are foods from the Fabaceae or Leguminosae family. So is a very wide family of plants, more than 2,000 species, and we talk. Sometimes we use without difference the terms legumes, pulses, or beans. They are quite different concepts. The legumes refers to any plant from the Fabaceae family, and it includes leaves, stems, and pods. When we talk about pulses, we are referring to the seeds, and typically beans, lentils, peas, and I will cover more the pulse as a food, but sometimes the term legume can be used without any difference. So these kind of plants have, unlike other plants, they have a very specific trait, that is, they master the art of symbiotic nitrogen fixation. So they have a symbiotic relation with the nitrogen fixing bacteria in soil, and they are an important source of nitrogen fixation. So this is like an ecosystem service that pulses provide to the environment. Other important topic is that they are a very particular source of protein, of vegetable protein. And this is also a very important aspect of the pulses, because when we talk about proteins, vegetable protein sources, the pulses are at the top of the table. They have also a low glycemic index starch, so the carbohydrates that are present in pulses have low glycemic index. They are an important source of fiber, minerals, vitamins, carotenoids, polyphenols, and other phytochemicals. When we talk about planetary boundaries, and this is a slide that I brought to you because this is a very important framework This was developed by the Stockholm Resilience Center in Sweden, and what we see here is different domains that are that can be that we can use to quantify the human impact related with climate change, ozone depletion, ocean acidification, biochemical flows, freshwater use, land change system, land system change, and biosphere integrity, namely the genetic biodiversity. I want to highlight this domain, the disruption of biochemical flows, namely the nitrogen. As you can see, we are behind the zone of uncertainty. So we are at high risk already. The use of nitrogen as a fertilizer has a very profound impact in the environment, and only about 30 to 50% of the nitrogen that is applied to the crops is actually used by the crops. So the remaining flows to the environment and causes most of the times eutrophication of several aquatic areas. So the nitrogen use is a very important topic when we try to solve a very important environmental problem. And the legumes, the pulses can provide this, can contribute to solve this issue, because when we harvest legumes, we can use lower amount of nitrogen fertilizers, and they can also provide nitrogen to the soil for them and for following crops. Another important issue is the use of the greenhouse gas emissions that are related with the protein production. And as we can see here, to produce 100 grams of protein, we use the greenhouse gases emissions are quite high when we talk about animal-based protein, mainly beef or lamb or even pork. But the use of, when we produce the same amount of protein using pulses or peas or nuts, the environmental emissions of greenhouse gases are very, very low compared to the animal sources. Also, the land use that we need to produce the same amount of proteins is very low comparing to the animal, mainly beef production. And when we see in the future, this is a projection between 2010 and 2050. When we see a projection regarding the several environmental domains, greenhouse gas emission, cropland use, freshwater use, nitrogen acidification, phosphorus acidification, when we see this, we see that the animal products, as if we continue to let's say, the business as usual. If we continue to using the same food production system, and and seeing an increasing in the demand of the food that is a result of the increase of the population, of the world population, the increase in the buying power of this population. We see that, and this is the 100% the planetary boundaries. So this is the planetary boundaries we have here, and we can see that we exceed very much the planetary boundaries that are established. So we will, this is non-sustainable according to these projections that we can see here. So in several domains, we are already exceeding this, as we saw in the previous slides regarding the nitrogen acidification as an example, but the projections that we will exceed. And we can see the legumes and nuts as an example. They contribute with very small amount to this environmental impact. So they can be seen as an important source of food, of nutrients, without with low impact in the environmental, in several domains of the environmental impact. Regarding the health, the human health, we see that one of the main causes of the burden of diseases are cardiovascular diseases in the Western world. We see that during the 20 years from 1990 to the current time, we see an increase, a continuous increase in the number of deaths, cardiovascular disease deaths. We have about 10 million at the time, and we see that the main risk factors for these diseases are metabolic risks, environmental and occupational risks, and also behavioral risks. And we can highlight here the dietary risks, the LDL cholesterol, the high body mass index, and these risks, and there are several studies that study the effect of the intake of pulses in these risks of cardiovascular diseases, and they have showed us that when we include pulses in our diet, the risks of heart diseases or cardiovascular diseases is lower. There is a decrease in LDL cholesterol when we include the pulses in our diet, and this has been showed by several cohort studies. This is a cohort study following more than 9,000 people during 19 years. And this study showed that people who ate 4 times or more pulses per week had a 20% lower risk of heart disease, 11% lower risk of cardiovascular disease than those who ate legumes less than once a week. Another meta-analysis showed that 14% reduced risks of coronary heart diseases when comparing people eating more than 4 times or more per week, comparing to the lower eaters pulses. Recently, a meta-analysis with including 36 human studies with more than 1,800 of participants show that when we substitute red meat by high-quality plant protein sources, we have a favorable decrease in total cholesterol or LDL cholesterol. So this is as I showed, a risk factor for cardiovascular disease. Regarding other health outcomes like diabetes, the evidence is not so strong. There are quite important aspects of the composition of pulses, like the low glycemic index, carbohydrates, the content of fiber, but the studies, the observational studies that are being made, or the clinical studies that are, the experimental studies that are being made, are not, the evidence is not so strong. So there is a limited number of randomized studies and controlled trials, and we don't have any consistent results showing the benefits in this health outcome. Regarding obesity, there is a strong evidence already that when we include more pulses in our diet, the risk, we have a lower risk of a high body mass index or a high waist size. So these are important contributions of the intake of pulses to decrease the risk factors for cardiovascular diseases. The food in the Anthropocene, these studies. So this was an important contribution to the joining health experts and environmental experts that produced for the first time a reference diet that includes both dimensions: the health impact of food, of the intake of foods, and the environmental impact of food production. And this was produced in 2019 and is a very important piece of information, a very important piece of work. And as we can see here, there are, regarding the protein sources, we have these reference intakes, the legumes, around 75 grams per day, the beef, around 14 grams per day. So these are the reference values that we have. And when we compare these values with the actual intake, let's say in Portugal, and we can see for legumes and for red meat, we see that more than 99% or almost all the population has an intake lower than the reference intake for legumes, according to the Food in Anthropocene, the Eat-Lancet Commission. So we have more than almost all the population eat less legumes than the reference diet, and almost 100% of the population eats more than the reference value for meat. So these two bars show that a need for a shift in the protein sources that we use. So neglecting legumes has compromised human health and sustainable food production. So my proposal here is to increase the food product development using pulses as a main ingredient. And we have several options. We have several options that can use pulses as a main ingredient. Of course, we have fresh pulses, dry pulses. We have cooked pulses. We can produce flours or flakes with pulses. We can sprout the pulses. We can make flours from the sprouts of pulses. These are important and can be options to increase the intake of pulses. We can even sprout even longer and have this kind of greens. We can use fractions of protein, fractions of pulses. We have pea protein. We can have fractions of fibers. We can have fractions of starch. So different kind of fractionated ingredients that can also be used as a raw material for food products, for food formulations. We can produce beverage from this kind of products, and we know soy beverages and other beverage produces from pulses. We can produce alternatives to cheese, to dairy products, to eggs, to different kind of alternatives that we can use, that we can produce using pulses or ingredients from pulses. This is an example. We can produce with this kind of fractions. We can produce different kind of snacks, crackers, cookies, biscuits, and pastas, or baking dough for several purposes. Meat alternatives are also an important, and when we see the main alternatives that are in the market related to meat, most of them use as a main component some kind of pulse, and this is also an important use of this kind of products. We can produce sauces like hummus or other kind of sauces. And of course, we can produce fermented foods, legume-based fermented foods. And we here have a very large palette of different kind of fermented foods, pulse-based fermented foods. They are mainly produced in East Asia, in Africa, in several, not so common in West world, with a few exceptions like soy sauce. We already have, or tempeh is also an ingredient, a food product that we have. But there are many more that we can use as an inspiration for development, for development new food products, or to study, as a scientific case study. And we have different kind of products fermented with several different kind of bacteria, of yeasts, of fungi, with several uses as a breakfast, as a snack food, as a condiment. So these are some examples that are here to illustrate the big diversity that we have and the big. So I have a student from Philippines that says to me that here in West, in Portugal, you mainly ferment bread, and in Philippines we ferment everything less bread. So they are quite they have a strong traditional culture of fermenting, of fermentation, and most of these products can be used an inspiration for new food product development. So this is other kind of legume-based fermented foods. The miso, the natto, the papadam. I will talk in a minute about the papadam. And tempeh from soybeans. This is a product that uses a fungi and can be used as a meat replacer, a cake, as a snack. So this is some examples, and at our university we developed in the recent years a papadam inspired product. So we use a papadam as a model for an inspiration for our development, and we produced a kind of a snack that is baked and use more than 60% of its composition is a pulse flour. We use beans or chickpeas or peas to produce this kind of snacks, and these snacks are a protein source, a source of vegetable protein. So with a low caloric value, with no gluten. So this is a product that targets the needs of a growing community of consumers. So this is an example of a development of a pulse-based foods or a legume-based foods that uses as an inspiration a traditional food like papadam. But this we are in the process that studying more fermented foods, pulse-based fermented foods, and try to use that as an inspiration for new food product developments. So, as a conclusion, to increase the intake of pulses as health and environmental advantages. So, we believe that the introduction of pulses, a higher introduction of pulses in our food production system, in our diet, is a very beneficial aspect. For that, there is a need to increase the range of pulse-based foods that we have in the market that are accessible to the consumer. So to promote the intake of pulses in non-usual places or non-usual times, occasions, this kind of product can be consumed in as a snack for children at school, for in our offices. So this is another time, different occasions to heat pulses that we usually don't heat pulses at snacks, but we can, with these kind of products, we can do it. The fermented foods in global age. So we need to learn. We need to learn more from traditional fermented foods from other countries, mainly the East countries, and to bring this knowledge to our diet. There is a strong scientific interest in legume-based fermented foods regarding the microorganisms that are used, that are fermenting, the biochemical changes, the increase in nutrition values, etc. There is also a need to improve the physical and sensorial properties of the food products. A modification, we can modify the substrate. We can produce tempeh from beans that usually are produced.
Sorry to interrupt, Duarte. Can you please finish? We are out of time.
Yes. And we need a strong communication with consumer. And that's it. Thank you.
Thank you, Duarte, for your interesting talk. We are a little bit out. Can you stop sharing? I'm going to share the questions on Slido. So we have time only for one question. The question is, legumes are a source of tryptophan, which is a precursor to serotonin. Is there any strong study linking the consumption of legumes and mental disorders?
No, as far as I know, we still don't have this kind of strong clinical studies. As let's say, it's biologically is plausible to the intake of tryptophan and the mental health, but until the moment we don't have any evidence of that.
Okay, thank you. Thank you for coming. It was very interesting.
Thank you very much.
Now our next keynote speaker is Ed Curryman. Ed Curry works at DCM Nutritional Sciences, and his work focuses on the identification of microbial communities that influence host health and disease, and then translation of this investigation into a clinical application. Ed Curry, you can start as soon as you are ready. Thank you.
Do you hear me now?
Yes, yes.
Wonderful. And how about my screen? Do you see already?
Yes, you can start. Thank you.
Very good. Thanks a lot for the invitation, and it's my pleasure to talk about this topic. This topic, that vitamin for the gut microbiome, a future microbiome modulator. And I promise you that in the next 25 minutes, I will convince you that why vitamins are so much important and why we have to really take care of it. And this is a breakthrough innovation, I must say. The presentation I'm presenting you, it's prepared by myself and with my colleague Robert Steiner, Tim Mark, Wilbert, and Farm. And I'm an EMEA principal scientist working for DSM at Kauseraut, Switzerland. So what I will present you in the next 25 minutes, it will be a snapshot, a reminder about the gut microbiome and host health, considering maybe we have a different kind of audience over here. And then to present you how vitamins modulates the gut microbiome, overview of mechanism of action that we propose, safety concerns, and conclusion, and the outlook, how we see it. If we just see the gut microbiome, we all know that in the colon there are millions or trillions of the microbes, including bacteria, fungi, viruses, so on and so forth. And they are interacting with the host. For the interactions for the host, the microbiome are using different molecules or the component, out of which short chain fatty acids remain the key metabolite or the main metabolite often discussed. They contribute the microbiome. They are contributing not only to the intestinal disease like IBD or IBS or the colon cancer, but it goes beyond extraintestinal diseases as well. For instance, you see anxiety, pain, autism, multiple sclerosis. To be honest, these days, if you follow the literature, you may find that there are, you can name any disease and you will find some correlation with the microbiome. If we see that, okay, in the gut, you have the microbiome and there are many factors which can modulate the microbiome, and some are very important and they are often discussed. For example, you will see that how a kid is born, is it a cesarean or it is a vaginal birth? They have the different microbiota. If they are breastfeed or they are formula feed, they have different microbiota. Drugs, of course, such immunosuppressive, beta blockers or proton pump inhibitors, even and antibiotic, of course, they have the high impact on the microbiota composition as well as of their function. Geography, if you are living in Japan or India or in China or US and Europe, they might be having different microbiota because you have a different diet and the lifestyle. And in terms of the diet, it remains the core that is one of the main determinant of the microbiota or shaping the microbiota. And there are a lot of correlations and the causation via the disease, yeah, especially the intestinal disease like the IBD, inflammations, diabetes, obesity. Based on the diet, so you might heard this name very often, prebiotic, probiotic, and a combination of both called symbiotic. And in last years, you see a lot of new terms are coming up: probioticals, symbiotics, oncobiotics, genobiotic, you name anything, and then there is a biotic attached to it. And they are all having a different type of benefit over there. Going back, I'm talking about the vitamins and how we see it. Very recently, this was in the ISAP, this is the International Society, Scientific Association for Probiotic and Prebiotic. They start to consider that, okay, apart from this carbohydrate and the bacteria that we are using as a probiotic for the health benefit, what are the other component or the constituent could be the modulator? And here comes the innovation of the breakthrough innovations that we started long back. This is a very recent one. So I think it's a 4 or 5 months ago it was published, where they have put a vitamin also in a category of a prebiotic, but considering is a non-carbohydrate substrate prebiotic, and it comes into the category of the microbial substrate. Earlier also there was in the ISAP some consensus, and we have already published or published it that riboflavin could be one of the prebiotic that can modulate the microbiota. Having said that, so I come from the microbiological background, and we all have learned in our past years a textbook knowledge that bacteria are able to produce vitamins. For instance, vitamin B12, the pseudo-vitamin cyanocobalamin, and vitamin K, they are coming from the bacteria. So we learn it very often. But it comes to the concept, if bacteria are able to produce vitamins, then is it make some sense that how it really goes and how it can interact with the microbiota? And this was the paper from one of our collaborator. What they have shown over here using the metagenomic sequencing from the Ellis-Thiene group, they have shown that if you go to the microbiota and you see intracellular concentration, that how microvitamins are produced, then you see B3, B6, folic acid, they are highly produced via the bacterial cells, or it is contained in the bacterial cells. And out of which, if you go to the percentage daily requirement intake from the human gut microbiome, B6, folic acid, and vitamin B12, they are contributing a lot. Not B7, B12, B1, and B5. So which gives you a good indication that the human gut microbiota is able to produce vitamins, especially from the B group, yeah? And if you go little more further, this is also from the Ellis-Thiene group, and they provide that there is a cooperation and the inverse correlation of the different microbial groups in terms of the production of the vitamin B. For example, here is the biotin example. And here is what you see over here, that there are different pathways or the intermediate steps which are needed for producing the vitamin biotin here. And this is produced depicted here with the X, biotin X. And different component or the different fraction of the bacteria are incorporating or playing a different role over here, which means the one biosynthetic pathway is not intact only in one bacterial group. It is scattered all over the groups, which means one product from the bacteria comes and that can work as a precursor to the different pathway, which means bacteria are coordinating or cooperating or having a symbiotic relationship within the biosynthetic pathways of vitamin B as well. Further evidences comes that how vitamin B synthesis in the colon or the microbiome is important. Here I'm showing you two different examples. One is the type 2 diabetes, another is the Crohn's disease. It was published not so long ago in 2019, Klassen and Das. And let's focus on the Crohn's disease. So in the Crohn's disease, you have a different set of inflammatory status before, during, and after. That's like inflammation, and then the remission stage. And what you see here, flavin biosynthesis, and that during the inflammation, their biosynthetic pathways goes down, and it comes from the metagenomic sequencing. And again, you see here in the thiamine, in between the during, it goes down. And again, for the folate biosynthesis, also going down. In the type 2 diabetes, it was shown in the biosynthetic pathway or the transporter pathway that in the healthy subjects and in the type 2 diabetes, there was some significant changes. For example, here in the minaquinone shown over here, or in the thiamine, further suggesting that the biosynthesis of vitamin B in the microbiota, in the health status and in the disease status is very distinct. And it provides you a rationale that vitamin B synthesis in colon is very important, and one can intervene via this way. This is another paper, is a piece of evidence, and it was coupled with the in silico analysis as well as the in vitro analysis. The data I'm showing you with the in vitro analysis, what they have done over here, they provide different vitamins and see that how the different bacteria they can grow. And this Faecalibacterium praunici and S variabile, these are two bacteria which are known to produce short chain fatty acid and butyrate, as I earlier said. These two metabolites are very crucial, and what you see over here that these butyrate producers, if you are lacking the niacin or pentathionate, pyridoxine B6 or the thiamine, then their production is going down, especially B3, B5, B6, which is further emphasizing that for the growth of bacteria, these vitamins are essential. If you deplete them, then they are not going to grow over there. With that, we come to a concept that, okay, vitamins are so crucial and important for the health and disease, and even they are a precursor, or they are the molecule which can trigger the communication between the different bacterial groups. Then how we can use this approach and to modulate the microbiota for the host health benefit. Here we propose is our hypothesis, and we are working on it, is proven hypothesis now, that rather than putting vitamins a systemic way where it gets used to absorb in the small intestine in most of the cases, how about if we directly deliver it to the colon? Because now we are producing, or we are giving a full opportunity of the microbiota to interact with these colon delivered vitamins, and then we can have a lot of health benefit. For example, we can enhance intestinal barrier integrity, inflammation, we can control pathogens, we can stimulate microbiota, we can increase microbial diversity, which is known to be beneficial. We go further and we worked on this concept. As I said, that is a now proven hypothesis. In this work, what our group, we have done it in 2016, that we administer riboflavin, that is the vitamin B2, 100 milligram in 11 volunteers. It was an open label trial. And after 14 days of administration, we took the microbiota. So we did take it before intervention started and after the finishing of the intervention. And what we see over here, what we observe, that Faecalibacterium praunice, out of 11 subjects, in 8 subjects, it goes down. And sometimes you see a very strong increase, such as in this case over here. And then there was a significant in the same number of subjects, it decreased in the E. coli. So you see this bar over here, and then there is a green bar that is the decrease in the E. coli. And this was very consistent. And you can imagine that riboflavin, so about 30 milligram, you can absorb it, and rest will spill over to the colon as per the literature. And we assume that about 70 milligram or 75 milligram riboflavin, it reached to the colon and it impacted the gut microbiota. Furthermore, it is from our collaborator from the Conares group, so from the Kiel University, from the Conares, and here they take a very elegant approach. Here they use this shellac technology to deliver the vitamins directly to the colon, and nicotinic acid and nicotinamide. So these are the two forms of vitamin B3 that were administered in the dose dependent, in the dose escalation manner, up to the 6 week. And what we observed in that intervention is a study that there was increase in the Bacteroidetes group after the 6 week. And it was not in the nicotinamide, but only in the nicotinic acid, because Bacteroidetes family, they don't have this nicotinamidase enzyme. And it was another evidence that delivering vitamins directly to the colon, making available to the bacteria, is a way to modulate the microbiota, and that can be used for the host benefit. In addition, it was also coupled that the insulin sensitivity and metabolic inflammation was also improved in those subjects. So this was the first line of evidence that we can modulate the microbiota by vitamins, and it can even provide you the host health benefit. Furthermore, to support it that, okay, how really it works, and then we plan that, okay, why not to give vitamins directly to the colon microbiota without having any intervention from the host side, because you have to prove it again and again. And here we did a experiment where the fecal microbiome that was inoculated along with the different vitamin. Here I'm showing the example of vitamin B6 in a ProDigest model, SHIME model from the Belgium company, ProDigest we call them. And after 48 hours of fermentation at the body temperature, that is 37 degrees centigrade, then we analyzed short chain fatty acid as the metabolite and the microbiota composition. What you see on the right-hand side, that in a dose-dependent manner, we tested 3 different doses. And you see there is an increase in the Faecalibacterium, there is an increase in the Akkermansia, there is an increase in the Bifidobacterium. So the people who are in the microbiome field, they will appreciate it that all these 3 bacteria are well known to have the positive impact on the host health. And here we should not overread it that it has to be dose dependent, but we tested 3 different doses, and we see that in all tested doses, there is an increase in the beneficial microbiome. In the same setup is another in vitro study. We did it in one arm. It was the folic acid, another important vitamin, vitamin B9, and again you see here that short chain fatty acid, especially the butyrate, there was a change, or there was an increase in the butyrate, important constituent of the short chain fatty acid. And again, we see here is a change in the Bifidobacteria positively. There was a change in the Faecalibacterium and Akkermansia. So again, it was proven that the vitamins B groups, they are interacting with the host microbiota, and then they can increase beneficial microbial communities or the microbial groups. Furthermore, we keep going, and then we went to a human study. And in this human intervention study, again, it was a pilot study. We take n is equal to 12 subjects in each arm. And then we tested vitamin A, vitamin B, B2, vitamin C, B2 plus C in combination, vitamin D, vitamin E, and then we compare it to the placebo before and after. In these 96 subjects, it was 20 to 50 years of age, and we take the fecal samples before initiation of intervention at the end of the intervention. And we took the microbiota composition and the short-chain fatty acid. We took the fasting blood samples to understand the immune markers and in vitro testing for the intestinal barrier integrity. And we also took questionnaires to understand that how it is having impact on the gastrointestinal symptoms and the quality of life. From this study, I will just present you two results, and just to give you an overview. These are the results for the vitamin C. And what you see over here is in the vitamin C, if you administer it in the vitamin C group, there was an increase, a significant increase in the butyrate, one of the main short-chain fatty acid having the positive impact on the human health. And again, there was an increase in the propionate, another short-chain fatty acid. And we also see an increase in the evenness, that is a marker of alpha diversity. And alpha diversity, increase in the diversity, the better the diversity and the better the health. This is the dogma, or this is the statement that we always see again and again in the literature. And yeah, so another result I'm showing you here is the vitamin B2, riboflavin. It was administered 75 milligram for a 4 period of time, for the 4-week period of time. And again, here you see there is an increase in the Akkermansia group, although it was not significant, but there was an increase. And you can imagine that Akkermansia is one of the key microbial species, which we call them, or we often say is a next generation probiotic. And how to increase it, and this could be the one way that if you directly put riboflavin into the colon where the Akkermansia sits, you can increase the Akkermansia and you can have all health benefits, especially for the metabolic diseases, obesity and the diabetes, well-connected Akkermansia. Again, here also we see the increase in the diversity. Yeah, observed number of species, better the diversity, better the health. Having said that, how it is happening, that will be a question that you and me, we will ask from ourselves. And now I'm showing you a few concepts and the proposal. I'm going back to the biotin synthetic pathway. So how it is working, it could be that, and this is our proposal, that when you are providing a vitamin inside, then you are giving a trigger to a bacteria to grow, and this bacteria growth will give a trigger to another bacteria to grow or biosynthesis, instigate the biosynthesis of other vitamins. Yeah, so these B vitamins can act like a cofactor or the precursor for the other gut microbiome. This is the first hypothesis or the proposal. Another, we know it that vitamins are well known to interact with the host. For example, niacin, well known to produce NADP, NADH plus, and is a ATP generation, energy metabolism. And this all will instigate or provide a niche to the bacteria, to the host, to provide a better environment to the bacteria. And these environment may further enhance the growth of the positive bacteria. Another could be, for example, riboflavin. So well known that riboflavin is one of the antioxidant. It is interacting with the glutathione reductase and is a coenzyme over there. And this glutathione reductase is well known to protect ROS, reactive oxygen species, which means less oxygen that will provide a better niche to the anaerobic bacteria, for example, Faecalibacterium. So it can induce a situation or the habitat for the host to work on. And in the same line, and this is a very elegant work done by the group of the Minho Raut. So Minho is the first author over there. What they have presented over here is that, for example, vitamin B2 and C, they are known to be antioxidant. And just imagine that you are putting an antioxidant into the colon, which means you can produce an environment that is less oxygen. So you deplete the oxygen over here. And here they have shown that the fecal redox potential, that is the measurement of the oxidation or the oxygen content into the lumen. And they have shown here that the fecal redox potential and the bacterial component, which is coming anaerobic and the anaerobic bacteria, they are going together, they are correlating. For example, you take the French adult control, their fecal redox potential is lower. So these are the healthy subjects. And same with the aero-tolerant bacteria, which are having a ratio of the aerobic and anaerobic. This is also going down, which means you're having more anaerobic bacteria. Another example you can take is the single-use children with the marasmus. So which means marasmus is a malnutrition conditions where you are supposed to have a more oxygen because there is a barrier integrity loss over there. And again, it correlates with the fecal redox potential with the aero-tolerant MAPI index, they call it. So you can imagine that you are putting an antioxidant enzyme, antioxidant vitamins into the colon, and you are changing the environment or habitat, which is more beneficial or more inclined to give a support to the bacteria which are anaerobes. Yeah, and this comes further in the same line that antibiotics. So we all know it that if you give the antibiotic, then there is surge of the Enterobacteriaceae group, which are known to be aerobic from the E. coli. And in this experiment from the research hall, what they have shown that once you give the antibiotic, so this is the red line, that the redox potential, it goes up. Yeah, so which means you are depleting the bacteria which are anaerobics, and you are creating a situation where there are more aerobes are growing and there is a more oxygen or there is a redox potential changes over there. And this completely correlates with the Enterobacteriaceae group, which is known to be aerobic groups. And you see here in the red, so once you give the antibiotic, there is a surge of Enterobacteriaceae or the oxygen levels, and then you stop it, then slowly it starts to decrease. Yeah. So this was another evidence that antibiotic is changing your redox potential, which means it is not doing good for your microbiota because it is giving more opportunities to grow the Enterobacteriaceae or oxygen levels, which are known to be pathobiont. Here is the example that we, in the same line that we have created some data. And here from the same experiment that we have shown you from the clinical trial, we measure the redox potential directly in the feces, and what we see that in the vitamin C group, where before and after, there is a decline in 10 out of 10 subjects, there is a decline in 8 subjects in the redox potential, which means the hypothesis that we propose is coming true, that via putting vitamin C directly into the colon, via colon delivered method, you can reduce the redox potential. And it was very encouraging to us, and we look forward to perform more experiment and to further validate it. Now coming to the safety aspect, whether if you put these vitamins directly into the colon, is it really safe or is it not safe? And these are the some results I'm showing you. They come from the mice. And here the vitamin B2 has been shown to enhance resistance to the different pathogens, including Pseudomonas, Klebsiella, Staphylococcus, Actinobacillus. In another experiment, B6 was tested, and they have shown that it clears the pathogens. Yeah, so this is a good sign. And this is another for vitamin B3 has been shown to kill the Staphylococcus aureus, yeah, up to the 1,000-fold. That's a lot. I'm coming to the conclusion and the take-home message. I have shown you in this presentation now that vitamin B are crucial and is produced by the microbiome, and you can intervene it and you can modulate the microbiota. They work like an essential growth factor for the gut microbiota. They may work like a cofactor. They can induce or changes your redox potentials. Is important for the pre-diabetic, diabetes, IBD, malnutrition-like subject. And supplementation and the overdosing part, you can modulate the gut microbiota, as I have shown you with the 2 different studies. And now we are looking forward more to establish that how these changes of the microbiota by the vitamins can impact host health. With this word, I will stop myself. And if you want to further learn about our technology and the methodology and this breakthrough innovation, please contact Wilbert Saibesma or myself, and here are their emails. And thank you very much for your time.
Thank you, Atkur. Very nice talk. Now we are going to check Slido for questions. So the first question is, do you believe that microbiome modulation can be used in the future for personalized medicine?
Yes. We are working on it, and it's a good question, and we look in that direction, and we are very much convinced that it could be used. Just imagine that if you know a priori that which microbes are depleted and you are lacking those vitamins, then you have the direct relation or the reason to provide the vitamins which has been depleted by your microbiome. And if we know that how your redox potential and how you look like today, then there's a personalized intervention so that can be done. And we are already working on that direction.
Okay, great. Other question, how do dramatic microbiome changes triggers the celiac disease?
Oh, this I must admit, celiac disease for the microbiome, this question I cannot really answer at the moment.
Okay, so maybe
Yes.
the next question, what
Yes.
can we include in diet to promote vitamins production by our microbiome?
From our perspective, we will say we are all taking healthy diet. If you are lacking something, take the redirect, the proposal that what we are doing it, that if you are lacking your microbiome producing bacteria, take the vitamin which can produce it, or you directly take that vitamins.
Yeah, sure. Yeah. Thank you very much. We don't have more time for questions, but thank you for coming. It was very interesting.
Pleasure. Thank you.
So now we have our last keynote speaker from the morning session, that is Lena Lange. Lena, you can start sharing your screen, please. Lena is a professor at DTU and the founder and owner of the startup company Bioeconomy Research and Advisory. She has been distributing her time between industry and academia, and the research focuses in the discovery of new enzymes for improved processing and product development. So without further delay, please, Lena, the floor is yours.
Thank you so much. I hope you can see my screen. Everything okay?
Yes, everything okay.
Very good. It's a great pleasure to have this opportunity to share with you some of the, what really has my prime interest right now is the microbiome, as also here so elegantly addressed by the former speaker. I would like to continue here and really emphasize that biotechnology can really contribute to building a brighter future, but we have so many solutions and so much of that is still not used. So I think we will have to focus on how to build momentum for having this impact. And I think we all know that it takes both technology, here, smart biotechnology, but it also takes behavioral changes to implement all these very good progress we have been reporting in the last 5-10 years. So I propose that we should be more focused on than when we are having new biotech solution, that we should design and remember that that the alternatives, the green alternatives we are working for should also be an attractive choice. And here I would like to emphasize that sequencing, as you all know, sequencing gut microbiome was a game-changing innovation. It's a focus for man, as we have heard, but it's of no less importance for animals and animal welfare, but also people welfare. How that comes in, I am addressing here. The game-changing innovation is highly relevant for improving industrial production of pigs, chicken, fish, etc. Gut health, as we know, is important for animal welfare. We can see it when, for instance, piglets are having a lot of diarrhea short after they have been born, and this is due to inflammation. But if we could improve gut health, we could have stronger animals and lower the early lethal events of diarrhea. But there's one more point which have not been much in focus. Try to put yourself in the position of a worker, a person working in agriculture in the area of industrial pig production. The daily start of the day is that you have to discard dead piglets. It's not much fun. You came into agriculture and doing livestock production because you love animals. And then you have to discard all these dead people, not people, but dead animals. Another thing is that MRSA is much of the people working in industrial livestock production are having MRSA infection. They are latent, they have no symptoms or any problem, but it makes it difficult. For instance, you have to go in isolation if you are in hospital, and it can be transmitted to others, which will not have it without having, or will have to suffer from it. So reducing use of antibiotics is a very strong measure for improving this kind of points I've just emphasized, but also very important for if you are treating, if you have less need of antibiotics, you can also contribute to lowering the threat of an antibiotic resistance pandemic to take place. So the really good news, as we have heard in the former paper also, that animal gut microbiome, just like man, human gut microbiome can be modified by what the food and feed intake. So how can we find the best kind of food or feed? You can find by analyzing the gut microbiome organismal composition, and that means improving would mean that you have a lower number of unhealthy and a stronger occurrence of the beneficial microbes. But you should also focus on analyzing the digestive function and the metabolite profile. So the microbiome is much more than just the organism there. It's also how they function, not just who they are, but how they function. So animal feed components, where we have found that we can really have an impact, positive impact, is for animal feed with probiotics, beneficial microbes, with prebiotics produced by enzyme, for instance, processing plant cell wall material, anti-inflammatory components, very important in feed biomass or released by a fermentation process. So all these points are mechanism where we can impact the animal feed and the animal gut microbiome. So what I'm here recommending is that we are contributing to reduce threat of an antibiotic resistance pandemic by making gut health improving animal feed additives as part of the dietary requirements for industrial animal production. Just so important for the reducing risk of this pandemic, which is a real threat. WHO tried to look up, it's amazing we are not talking more about it, but there are also add-on benefits, social benefits of the workers' health in reduced piglet mortality and workers' health condition, MRSAs. So let me just illustrate here, unlock the full potential of side stream. You can produce these prebiotics and you can grow probiotics on such side streams. And a good side stream for making beneficial gut health animal feed is to take the rapeseed meal, the side stream after having rapeseed oil made, and then you co-ferment it with seaweed, here the kelp, brown algae, and then you have a fantastic animal feed which delivers prebiotics, the fermented lactic acid bacteria, probiotics, prebiotics, anti-inflammatory, and your fermentation products. So that is a way forward. Then now I want to go to the food with the food focus. Biological solution can do so much more than what we do by now. And I think we all agree we have so many new results and which could be done. But I think we are marketing our results in a non-efficient way. So much is don't do this, don't eat meat, don't travel, all this we should not do. I think we should do much more in saying the new products we have and the new solution, the new biological products is actually that we can do things which are even more delicious or even more good in practices and open the world. And so we are choosing the good and green solution. So I'm impatient, international travel. Youngsters should also have a chance to see the world. Curiosity to see the world is a virtue. Let us get this new renewable jet fuel. You can make it on power to X and negative emission technology and build it on biogenic methane from biogas. We can make beautiful circularity by biotech, develop enzyme technologies for reusing cotton fibers, make circularity a delight. I am working on making circular used cotton to be basis for new types of very colorful pieces of art. And all this is possible. So circularity doesn't have to be refrained from, do not do. It can be colorful, beautiful, and very tasty. The last part of very tasty, I would like to say here. Industrial side stream doesn't sound like it's a fantastic menu, but it is. It's a huge opportunity. And by microbial or enzymatic conversion of such kind of biomass, improve resource efficiency if we're stopping food waste by breaking the recalcitrants and getting even more lovely food taste out of it. So what are the possibilities? Food and feed ingredients, alternative local protein from grass, rapeseed, substituting for soy, but also gut health, sugar oligomers from all the cell wall materials, hemicellulose. So we can get food and food ingredients from side streams. Very, very good for climate, for resource efficiency, but also feed and feed additives, the same thing, improved gut health, reduced need for antibiotics, health promoting products for gut health, nutrition, skin health. So, and also the new soil improvement products. We heard in the first very good, excellent keynote that the fertilizer is a source of emission as well. We have an opportunity from industrial side stream to get both food and feed and health promoting products and new types of fertilizers. So let's do that. And let us make it a favorite choice to produce more local and sustainable fungal protein. And how come? There are many details here, but I'd like to make sure that I get the major point across. A lot of the plant-based material for food is not very attractive to people who are used to eating more meat. They are lacking the kind of umami. And there is my proposal. Let us upgrade and use food, fungal-based products and other types to really make umami flavored food ingredients. And then we get more, we can use, it is alternative fungal, you can grow fungi on waste, just like insect, and you can make insect protein and fungal protein. You can get healthy beta-glucans from fungal cell wall, and the vegans can get the vitamin B12, which is very difficult to get from other sources. So much comes along with the fungi, and it is a fantastic supplement to the plant-based. And here, let me summarize this part. Umami enhancing food ingredient can make more people change to plant-based diets. So all what we heard, for instance, about the pulses, I totally consent, it's fantastic, but much of that would be even more commonly used. More people will choose it if you are adding a fungal-based food ingredient, or if you grow fungal mycelium, which has a bit of the chewiness like meat. You can take fungal enzyme, where you can get the umami potential out of selected types of plant biomass. For instance, cereal products have a lot of potential, which is not presented in the normal types of food we are eating. And don't forget the seaweeds, an excellent source of umami flavor. So I will now have 4 slides where I go very fast to, excuse me, where I will be presenting other types of things we don't use to its full extent. And there's so much we can do here. Blue biomass, products from fish waste, oil, protein, collagen for health, product from seaweeds. I have here already highlighted. Amazing what we throw 50% away when we have a fish right now. The blue bioeconomy has a tremendous potential in the whole area. We have here the blue bioeconomy. It's still only in Danish, but it will come in other languages. And I tell you, we can make almost everything we need from what is in the blue biomass. Portugal is a fantastic sea nation and a blue bioeconomy stronghold, and I think we could do much more there. So, but there are also in slaughterhouse and poultry waste many types of upgrading. You can make pharmaceuticals from upgrading blood to iron deficiency, upgrading leftover protein and fat, make it delicious high protein chocolate for elderly people, too low appetite, upgrading chicken feather, something I've been working on myself, microbial, you can make it much better use instead of pollution, this keratin, which is a protein source. And the whole strive where I've been talking about side stream upgrade, remember this kind of biomass pyramid. But right now, much of the biomass where the side streams or waste, organic waste are either polluting or are just going into energy and heat, lowest value at the bottom of the pyramid. But we can also make fuels, etc. But it's much better to go all the way up and make food and feed and even pharmaceutical, as an example with the blood or the collagen, etc. So remember always to use the full potential, unlock the full potential of the biomass. And that's where food and feed ingredients benefiting the health, but also the general and welfare and well-being. So also plant-based side stream, high potential. And you know, cereal milling, milk, potatoes, sugar beets, olive oil seeds, juice, dairy, brewing, etc. So much is there which can be used much better. So here I would like to finish in this slide and one an extra slide, but here finishing by saying, let us position that delicious and healthy by bioprocessing as a new driver for the necessary and urgent green turnaround. The delicious and healthy by bioprocessing, I mean, it is not just what you ought to do, but it's something you would like to do to change your diet. And we should, in doing that, we should promote that bioprocessing, probiotics, prebiotics, microbial fermentation, biological solutions, biological production, they are all made by biotech. But the change message, sorry for the organizers here in this session, I mean, this whole workshop, the change message is not biotechnology. The change message is the quality of the products, the solution it's provided, the effects which comes with choosing it. Let us remember that, because biotech in itself, a lot of people in Europe say that's just GMO and we don't want it. And it's not the hammer which make a house. It's a house we want to move into, which is what we choose. It's not the hammer we choose. So it's not the technology which sell the tickets. It is the products which communicate to the ones who have to take the decision of choosing, but it's also speaking to politicians, to decision makers, etc. So biology takes over now from fossils and chemistry, and not only that, it adds to health. Biology is a significant, really significant building block to a greener and brighter future. But most of the communication in climate change, in the whole need for a green turnaround, is not capturing that. We are not on the agenda yet. Time is right to place it high up on both climate and health agenda. And I really, really hope, and this beautiful initiative you are taking here with this workshop will spread the message, because it's urgent and we have so much to deliver for such a serious situation we are in, and we have so much where we can provide part of the solution. Thank you so much for your attention.
Thank you, Lena. I have enjoyed very much your presentation. Thank you. We have a lot of questions on Slido, so I'm going to share them. So the first one, the most voted, is what are the main barriers to get these fungal proteins in the market?
I think the main barrier is that not much of the products are out there. The second part is that most people have not seen fungi as more than the mushroom you buy in the supermarket. They have not seen that there are also fungal enzymes, there are also fungal filamentous, and they have not seen that all these beautiful, in the first speaker here, introduced about what you do in Asia and in Southeast Asia is actually fungal. It's aspergillus, it's rhizopus, it's temper, it's soy. So it's lack of knowledge. And then it's also, I think so few people have really seen that this umami can be a game changer for more people changing to the plant-based. So, and I would like to propose that the plant-based people join forces with people like me in the fungal part, that together we can make the change. Plant-based don't move fast enough alone, but together with the fungi, we can make it.
Okay, thank you. Another one, can we say that we are missing measures to improve health by strengthening the immune system to naturally fight pathogens?
That's a complicated question. Missing measures to improve health by strengthening immune system to natural fight pathogen. Yes, I think you're right. And here, my focus is the microbiome here. And the microbiome is a vehicle to actually strengthen the entire host, being the animal or people, to be stronger to fight pathogens. So, and it's a very clever question, because that is actually one of the reasons why it is so important to take this change of the microbiome as one of our key messages, that you can improve your strengths to fight infectious diseases by actually having a stronger gut flora. And that is, I call it a very democratic and a hopeful message, because, for instance, when you have been smoking for too long time, then you can never really repair. You can try, but you can change your food. You can change your food habits. You can start and getting stronger. You can keep out of hospitals longer. And the same for animal in the livestock. We can actually, with very small adjustments, affordable, already described, we can make the animals, the pigs and the chickens, stronger to fight the diarrhea invading pathogens, which is actually then benefiting also us. They have better lives, the animals, but we are benefited from not contributing to have a higher risk of the antibiotic resistance pandemic. So it's a very, very key point you put up forward here.
Exactly. The next one actually is connected to this one. This COVID exposed the importance of having a healthy lifestyle. Do you think the current pandemic situation will shape the way we approach health and food systems?
I think we can learn much, and again, a fantastic question, because we can learn much from this crisis. And I think it has a good message, and it is captured in the question that it is important to stay healthy. But I would like to expose even more. Remember that COVID-19 start invading and destroying the lung part of the lung. You have a microbiome in the lung. That is where it all started. It was called biofilm at that point in time. In the start of microbiome analysis, it was a biofilm we focused on. And could we study that more? Would be even more interesting. Maybe we have counteractive, so it wouldn't be so strong if we also stimulated the lung microbiome. Can you see? And so this whole part, also in the wastewater system, we have a microbiome as well. There we can trace COVID-19. And that is already done in many European countries, and I think all over the world start doing it, because it's the strongest early warning we can have. But then I'm advocating for that we should use the microbiome studies of wastewater treatment to actually also be a very early warning alertness for capturing development and growth of antibiotic resistant genes occurring in higher percentages or new types of antibiotic resistance. So that is what we learned from COVID-19, but now we should implement it to be stronger and more prepared for new types of possible pandemics, because we need hospitals to take care of the really sick ones. The rest of us who can eat healthy, take good care of our gut health, would then also could be able to stay out, which would mean there would be more room for the people who really need it, the vulnerable part.
Exactly. So one more. How long until we have these great solutions implemented in large scale?
Yeah, I'm very happy for this impatient. I can sense the impatience in the questions. I love it, because we need to impatient to do this. And I have 2 points, 2 focus points right now. One was they have this opportunity to the message. First time I express it so strongly is for this session, this workshop. Tomorrow, I'm going to give a presentation, which is organized by the European Parliament. And I will make a strong message there that they have to take away the obstacles for regulatory approval of many probiotics and prebiotics. They are waiting in line. Some of the strongest new measures will not be able to be commercialized because they have a warning signals. It will take 4 years before they will even start analyzing if they can be approved. I mean, this is a serious obstacle in this impatience, and it needs to be addressed because they are new and it's complex, but then we need to educate the people who have the regulatory to understand microbiome, prebiotics, and probiotics better. And then my next point is that in September, there is a UN Food Systems Summit, and there I am hopeful that it will be part of the Danish government's recommendation that we should make this, and I'm not going to repeat all the 4 different recommendations I have, but both for the animal, for the feed, the urgency in that because of antibiotic resistance threat, but also the other one, for the public health, that to really make it part of dietary advice to public health, that we should take in, use fermented product, use prebiotics, use probiotics, and more plant-based and very good fungal base, so it tastes well for doing this. So all of you who are listening, I would love you to go out and spread the message, because we need pressure also to get all these new products approved by the regulatory system.
Exactly. One more, since we still have some time. Which is the best approach to increase the umami flavor in food products, biochemical modification, or using different food sources?
Yeah, my favorite choice is always complex. That's biology. Take the type of material, food material, which have the best potential. And the best potential is, for instance, you find it among fungi, you find it among seaweed, but you also have certain, like bran is a fantastic source of umami from cereal processing. So there are different areas where that's one part, the best as a starting point. Then it is you can have enzymatic, it's a protease where you can actually unlock the potential of the umami flavor by specific proteases. And there are new proteases, both from Novozymes. I'm not making an advertisement, but I worked there for 20 years, but that's also many years ago. But and there are other companies out there who also have a specific protease, which will be very good for doing this. So it is microbial conversion, microbial enzyme conversion, and then end up having something which tastes good and has an attractive story to tell, because we're also telling a story of unlocking the potential of what was before wasted, meaning what was waste, we are going to use it for better health, better nutrition, better animal welfare, but also for freeing land by biodiversity, freeing land, for having more room for food production in plant-based. And that can be done by using the side streams upgraded for animal feed. Right now, it takes 75% of all arable land in the world to grow animal feed. We can start using the 30-40% we are losing, the waste from the food processing and food residues. All that can be used to convert into animal feed, and then we have ample of land for biodiversity and for growing more plant-based food. So please, please, please get it going, a momentum that we should upgrade side streams, we should upgrade food waste to its highest potential so that we can. And why is that so important when I'm saying this? The general is important, but why is it such a good idea? It's because the plant cell wall is a very big fraction of a big part of the waste. And the plant cell wall has the strongest potential for making the prebiotics. So what seems to be waste is now actually better because it is actually a very good starting point for making prebiotic.
Thank you, Lena. Very interesting. Thank you for coming, for joining us.
My pleasure. Thank you so much.
Thank you. Bye. So now we'll have a short break for 20 minutes, and we'll be back at 11 for the key project presentation. So see you soon. Thank you. Hi, everyone. Welcome back. Now we'll have 4 key projects presentations focused on food and health applications. Please leave your questions in Slido. We'll share the link in the chat. The first key project will be presented by Mariana Doria. Mariana, you can start sharing your screen, please. Mariana has a solid experience on innovation management and industrial sustainability. Currently, she works at Algae for Future, and today she will present the project Multistream. Mariana, you can start, please.
Hello, everyone. I hope everyone can see my screen properly.
Yes, yes.
Well, first of all, thank you very much for the invitation. We believe that the Multistream project is a very important project for the microalgae sector, and we hope to be delivering very important breakthroughs by the end of the project. So it's 1 year, the project has started in May last year, so it's 1 year now that we have started in the middle of this pandemic, but we are being able to adapt and go through it. So Multistream is a 9 million euros budget project and intends to create a sustainable multi-strain, multi-method, multi-product biorefinery based on microalgae biomass, integrating industrial side streams and creating high-value products for food, feed, and fragrances. So the key drivers for this project is regulation, market, and industry. Regulation, because as everyone knows, we have each year we have evolved to more strict regulation regarding sustainability. And in the case of Multistream, one regulation that has impacted one of the products that will be derived from Multistream is the intention of restricting microplastics of consumer goods. So this is something that we are looking, everyone is looking for degradable alternatives for these microplastics used applications. Market, given the societal awareness of environmental issues and conscious of consumers and businesses that we need to look for more sustainable raw materials, more sustainable processes and products. And industry, given that today we have a growing demand for alternative sources of raw ingredients, and all these new potential sources of raw materials for the old industries are a very big business opportunity for everyone and for the microalgae sector as well. So given this critical need to shift to a sustainable means of producing food, feed, and fragrances, that the consortium that is developing Multistream has started this project. So why microalgae? Specifically because it represents a promising solution to address current products regarding several. It can be a very good substitute to agricultural products and other fossil fuel derived products, and has still a vast biosynthetic potential that is still not being fully used, and it's a rich source of lipids, proteins, and other high-value compounds. But however, despite these advantages, it is still microalgae not so much in use, and mainly because of barriers of scale. So how to make a large production of biorefinery based on microalgae in a technical and economically viable way to substitute conventional products such as palm oil or soybean. So how to reduce these costs and boost sustainability? Multistream has put in some challenges, that is constantly to improve the strains, even though we don't use any genetically modified methods, but we can improve the strains. Also design engineering improvements that are fundamental for both cultivation and harvesting technologies and the biorefinery to reduce CapEx and OpEx. Circular economy, it is highly exploited in this project, looking for side streams, industrial side streams that can be used inside the cycle of the production, both during cultivation and production of the different products. The integration of different technologies, looking for this multi-strain, multi-method, and multi-product biorefinery. And finally, valorizing all fractions of biomass in a zero waste approach. So this intention of this biorefinery is to have a final, all fractions will be valorized and applied in different sectors. So the consortium is made of 8 players, from end users, that is upfield, an important food sector player in the market. For farmers, that is an important feed player in the market, and IFF, that is an important fragrance player in the market that will support that is supporting the project, putting these products in the market. Research institutions like Algatech in Czech Republic, LNEG and IBET in Portugal, and microalgae producers like A4F and FICOM. So regarding the impacts that we expect with this project is to demonstrate the validity and feasibility of this multi-biorefinery approach regarding microalgae valorization, to create a new cross-sectional interconnections. So we are going to be producing different products for different sectors and establish also new sustainable and economically viable value chains. Regarding environmental benefits, we can say that our targets is to increase overall resource efficiency by at least 20% over what we have with the conventional cultivation processing approach, and reduce energy consumption and GHG emissions by at least 20% over the state of the art that we had when we started the project. And these 7 consumer products that will be a conclusion of the project will comprise products for the fragrance industry, considering proteins and small organic compounds, lipids for spreads, and protein, carbohydrates, lipids, and feed ingredients. It's important to say that for the food industry, microalgae is already used, but we intend to enhance the applications of microalgae in the food industry by giving more specific, more complex and high-value products for this industry. We will be developing protein pigment as natural food coloring in these applications, enhance the applications of omega-3 and omega-6 enriched oils in different food applications, and lipids and phospholipids to replace also palm oil that has been facing different controversies and sustainability problems regarding where it is being produced. And we can have here in Europe very sustainable sources of lipids and phospholipids. So that's what I had to present to you right now. I'm open to any questions that you might have. Thank you very much, and you have my contact if you want to have any further contact regarding it. Thank you very much.
Thank you, Mariana. I have enjoyed very much your presentation. We have now time for 1 or 2 questions. I'm going to share them. So the first one, you mentioned improved strains. Do these strains have resulting lower growth rates, which results in auto-competition by contaminating strains in large scale?
Well, I'm not sure if I understood completely the question, but yes, we have a team that is working specifically in Algatech, but also other consortium members in improving the strains, and improving in the sense of looking for non-GM techniques to improve these strains, but to increase growth rates. So the intention during the project is to have higher growth rates to the strains that we are being used. We have been testing several, more than 6-7 different strains, and using also autotrophic and heterotrophic methods of production.
Okay, and next one, why not using genetic modifying methods to improve algae performance?
Well, in this case of this project, given the end uses applications that we have, like food, feed, and fragrances in Europe, we have some restrictions to genetically modified methods that can derive more difficulty to insert these products in the market. So we decided in this case of this project to not use genetically modified methods, but we don't have anything against, and we believe that it must be explored further, all sorts of methods to have better production rates.
Thank you. And what is the main challenge of the process integration?
Well, we will be using different biomasses from different strains of algae that will be entering this biorefinery. And from this biorefinery with different, it is an engineering challenge. Not only we have a demo scale biorefinery. I don't know if it is, I can't say if it is just the scale, for instance, of this high scale with different biomasses entering, and we need to have different fractions with a specific also purity and quality for the different sectors. So we have several engineering and technological challenges that we have to face. We are still in the beginning of the project, so I don't know if I can say what is the main key issue that will make us mad by during these years.
And all microalgae have the same potential?
Well, each strain has different proportions of protein, lipids. Some have different kinds of pigments, and we don't have, let's say, they're different, and we need to work with them differently to enhance each quality. Also, the ones that produce the proteins produced by each microalgae, they have different proportions of the amino acids. So each microalgae is being worked individually to have their derived products enhanced, and they have great potential, but differently.
Okay, thank you very much. Thank you very much for coming.
You're welcome.
So now we'll move to our second speaker, that is Adelaide Braga. You can start sharing your screen, please, Adelaide. Adelaide is a researcher at Center of Biological Engineering at University of Minho. She is currently working in development of production processes of prebiotics and will present the project for Symbio. Adelaide, you can start, please.
Sorry, I need to start again. Okay, you are now listening and see my screen? Okay, sorry.
No, the screen, no.
No?
Not yet. We were seeing the before we were seeing your presentation. Yeah, now you can see, you can start.
Okay, sorry. So good morning to everyone. Today I will talk about the full Symbio project that aims to use synthetic biology approaches to design and construct microbial cell factories for the production of fruit oligosaccharides. The increasing demand for natural labeled products and adoption of an healthy lifestyle drive the consumer's preference toward new foodstuffs that enhance the quality of life and reduce the risk of suffering from some disease. The current health-based consumer trends explain the increasing demand for functional foods, foods that have a potential positive effect on health behind the basic nutrition. Prebiotics are indeed attractive compounds due to their multidimensional beneficial effects, and its definition was recently updated to a substrate that is selectively utilized by the host microorganisms, conferring an health benefit. The prebiotic market is rising, and it is expected to reach $7.37 billion in 2023. Of all prebiotics known so far, fruit oligosaccharides, FOS, have attracted special attention. These non-digestible compounds are produced using sucrose as substrate, applying enzymes such as fructosyl furanidase or fructosyl transferase. However, during the reaction, large amounts of glucose are produced. And since glucose is a competitive inhibitor of these enzymes, some sucrose remains unreacted, leading to low sucrose yields and also low phos purity. Despite efforts that have been done to overcome these limitations, the presence of high sucrose glucose concentrations, together with some sucrose and fructose that compromise the prebiotic effect of the FOS mixture, and the presence of some sucrose and fructose is still a bottleneck. So novel approaches to further improve the process outcome are still required. In this sense, the Bio project aims to propose a simple and sustainable process using two different hosts, the bacteria Xymomonas mobilis and the yeast Saccharomyces cerevisiae, that will be genetically modified in order to increase the phos purity and also remove the small sugars, producing a final product with a high prebiotic effect and commercial value. The project is organized in 4 main activities. The first one consists in the design and construction of Xymobilis and Saccharomyces cerevisiae strains able to produce FOS. The idea is to construct mutant strains in order to prevent substrate consumption, and also to have overexpressed phase genes or phase genes from recognized FOS producers. These strains will be further genetically optimized in order to increase the FOS yield and purity. And after the process will be optimized, the idea is to use agro-industrial byproducts as alternative substrates and also scale up the process to bioreactor scale. Finally, in the last task, the produced FOS will be chemical and functional characterized and prebiotic potential assessed using in vitro gut model. Now I will show the work that we have been done with Xymomonas mobilis. First, we decide to explore the potential of Xymobilis to produce relevant amount of FOS in a simple one-step fermentation process. We performed an optimization with the wild-type strain using a box bank and design approach when we studied the effect of 3 factors: sucrose, yeast extract, and sodium chloride concentrations in the production of FOS. In the Pareto chart, we can see that the concentration of sucrose had a significant effect in the production of FOS. And when we look to the model, we can find a region that maximizes the production of FOS, and under the optimal conditions that are 300 grams per liter of sucrose, 20 grams per liter of yeast extract, and 2.5 grams per liter of sodium chloride. The model predicts a production of FOS of 45.3 grams per liter, and this value was further confirmed experimentally. Since sucrose is the most significant variable, we decide to increase its concentration to 350 grams per liter and see the effect in the production of FOS. Under these conditions, we increase the production to 51.6 grams per liter. After this, we scale up the process to bioreactor scale, and using the same medium composition, we are able to produce around 53 grams per liter of FOS, being the main produced FOS 1 custose and 6 custose, without accumulation of glucose and sucrose. Furthermore, we also have around 40 grams per liter of sorbitol and 8 grams per liter of flavin. At the same time, we are also trying to genetically modify the Xymobilis in order to improve the process yield, in order to avoid the sucrose competition and increase its availability for FOS production. The genes coding for the enzymes responsible for sucrolytic activity, in this case the SACC gene, will be deleted. In order to enable this, we will integrate a cassette carrying the tetracycline resistant gene and also an FRT sequence in SACC gene of Xymobilis genome using homologous recombination. This cassette will be further removed using the FlipPas expression system in order to maintain the GRAS status of our host. In addition, we are also trying to express phytase from several sources in this host. We select some fungi such as Aspergillus and also some yeast like Saccharomyces. The genes will be amplified, cloned into a plasmid, and further electrotransformed into Xymobilis. Now I will present some of the work that we have been done with Saccharomyces cerevisiae. Simultaneously with work that we have developed with Zemobilis, we are also trying to develop an in vivo bioprocess approach for one-step production of foes from sucrose using a recombinant yeast. In this case, we will use a commercial Saccharomyces cerevisiae that already lacks gene responsible for sucrose hydrolysis. And two different versions of the fructosyl transferase gene from Schiomyces occidentalis were transformed into this yeast. The wild type version that you called S160 and the mutant version with improved transferase activity, the L160. These two genes were cloned into the plasmid PS under control of a GAL promoter, inducible promoter, and we observe that you have a higher production of foes with L160 version of this gene. So this gene was selected for the next experiments. And the idea here is to try to reduce the production costs of the process. And instead of using an inducible promoter, we use a constitutive one, the GPD promoter. So the gene was cloned into this plasmid carrying the GPD promoter. And when we compare the production of foes for the two promoters, we see that we don't have statistically significant difference between both constructions. So we decide to select this one for the next optimization steps. When we try to improve the process productivity using different sucrose concentrations. And we see that we have a higher productivity when we use 300 grams per liter of sucrose. However, under these conditions, sucrose was not completely consumed at the end of the fermentation process. And in order to avoid this, we try a step-fed batch approach with two pulses. We start with 150 grams per liter of sucrose, and after 36 hours, we add another pulse. And we can achieve an increase in the productivity, like more than 1.3-fold increase when we compare with batch fermentation. The results obtained until now demonstrate the potential of this approach, and we are now still trying to improve this. To sum up, with the FOSIMBio project, we expect to develop a new prebiotic mix with potential prebiotic effect that can be further incorporated in food metrics or produced and commercialized as a prebiotic mix. And the output is thus expected to contribute to the development of a sustainable process to valorize agro-industrial wastes and residues, contributing to the circular economy and the European Green Deal. I would like to acknowledge all the team members that participate in this project and the financial support. This is what I want to share with you today. Thank you all. Any questions that you may have are now welcome.
Thank you, Adelaide, for the great talk on FOS production. We have several questions on slide. We are going to one or two. So the first one is, between Saccharomyces cerevisiae and Xenomonas mobilis, which one do you think has more potential to produce FOS?
Okay. To produce FOS, I can consider that both strains have potential. So looking to a more global perspective, based on the results that you have until now, we have a mixture with Zemobilis that seems that can seem more interesting, since behind FOS we also have other compounds that are also prebiotic, like leaven and sorbitol. So I think that Zemobilis can be more interesting looking to a final product, since we have more than FOS, also other prebiotics.
Okay. And does in vivo production of FOS can compete with enzymatic production? Yes,
because we can also have higher yields in vivo process. And looking to a perspective of a process for commercial production, the in vivo process will be easy, since in one step we can have the production of foods, and in the enzymatic production we need to produce the enzyme first, after purify and produce the foods. So yes, I can see that the in vivo production can be used.
Okay, so only one more. Do you foresee any problems in the use of byproducts or agro-food residues in the process?
Yeah, everyone, or most of everyone present here knows that byproducts can be an issue when you use this kind of substrate, but we can have some pre-treatments in order to have a more homogeneous composition, and we can use this type of products. Yes, we need to have a pre-step of preparation of the residues before using, but I can consider that you can use this type of products.
Thank you, Adelaide. Thank you for coming. It was very interesting. So now the next project will be presented by Claudia Amorim. Claudia, you can start to share your screen, please. Claudia is a researcher at Center of Biological Engineering at University of Minho. Her research focuses on exploring agro-residues to create value-added compounds and incorporate them in food products. Claudia, you can start the presentation, please.
Can you see my screen?
Yes, yes.
Okay. So good morning, everyone. I'm enthusiastic to present our work developed under the New Food Project. Overall, the main goal of the project was the development of innovative food products with potential to be launched in the market. So our team developed this idea, innovative traditional pastry, baking healthier choices. And we envisaged to create a healthier and functional option for the traditional Portuguese pastry. We ended up selecting and reformulating the Portuguese rice cake, as you can see in this picture, for the Portuguese ones. Okay, so specifically, we wanted to start by reducing the sugar content of the rice cakes and also to incorporate prebiotics. Besides the health benefits linked to the consumption of prebiotics, they have a sweetening effect and low caloric power as well. We evaluated the viability of our idea by analyzing market indicators, and we found positive trends, namely the increase of diet-related diseases. But on the other hand, consumers are more conscious about the importance of food as preventive medicine, including functional foods, whose market has been increasing, especially in the current pandemic situation. And finally, the pastry sector in Portugal has been expanding, but there is still a gap associated with the healthier alternative products that we want to explore. The carbo-based prebiotics are the most studied type of prebiotics, particularly inulin and galacto-oligosaccharides, or GOS, are well-established prebiotics, widely commercialized, and with consolidated evidences of their health benefits. Inulin is a polysaccharide that can be extracted from agave, for instance, while GOS are oligosaccharides that can be found in milk, for example. Both compounds have a sweetening effect, and then can be used to increase viscosity, which make them interesting food ingredients. So we first select the product we developed, the Portuguese rice cake, because it's a widely consumed cake in Portugal, especially by children, and a high caloric one. Then we tested different recipes, and we selected the one that better resembled the traditional cake based on the evaluation made by a tasting panel. In parallel, we compared the prebiotic effect of commercial agave inulin and commercial GOS using an in vitro model with human fecal inocula. The compound presenting the highest prebiotic potential was selected. Then we developed cake formulations by a panel of, the cake formulation having less sugar and containing prebiotics, and they were selected by a panel of tasters and the selected one was characterized in terms of physical and textural parameters. The fermentation of prebiotics by gut bacteria results in the production of lactate and short-chain fatty acids that play an important role on human health. And here we have a representation of acetate, lactate, propionates, and butyrate during 48 hours of fermentation. And here we have inulin and GOS and the respective control representing the absence of prebiotics. As expected, the addition of prebiotic increased the overall production of short-chain fatty acids, especially with GOS. And the accumulation of short-chain fatty acids also decreases the medium pH, promoting the reduction of pathogenic bacteria and causing changes in the metabolic pathways that will interfere, for example, in the production of ammonia. So a similar relative decrease in the pH value was observed for inulin and GOS, but GOS resulted in a higher decrease in the ammonia concentration. Ammonia contributes to a bad fecal odor, and it's a toxic compound that can lead to colon carcinogenesis. The fermentation of prebiotics also affects the production of gases, such as carbon dioxide, hydrogen, and methane. Well, methane is only produced in a small portion of the population who harbor methanogenic bacteria in their gut. But for both compounds, higher concentrations of CO2 are obtained when comparing to the control, and the production of hydrogen showed no relevant differences. Regarding the production of methane, no significant differences were observed between control and inulin, but GOS decreased fivefold the production of methane. High production of methane has been associated with different disorders, including obesity, irritable bowel syndrome, and chronic constipation. This gas reduced intestinal transit time. So this was a very interesting result for GOS. Regarding the modulation of the microbiota, the supplementation with inulin or GOS resulted in a distinct modulation of the gut microbiota. So here you can see the blank, now it's with inulin, and here with GOS. So putting all three together, you can see that both prebiotics increase the relative abundance of lactobacillus and bifidobacterium, which are linked to several health benefits. But inulin presented a higher selectivity towards lactobacillus and GOS for bifidobacterium. So these bacteria are able to metabolize complex carbohydrates, producing mainly acetate and lactate, and releasing CO2, which corroborates the results previously presented. Both prebiotics lead to a significant reduction of the relative abundance of proteobacteria, including Escherichia coli. So attending all the previous results, those seem to present the highest prebiotic potential, and so they were selected to be incorporated in the rice cakes. Concerning the characterization of the cakes, the selected cake was the one with 75% of sugar and 1% of GOS supplementation. But we also characterized the cake with the original recipe containing 100% of sugar because we wanted to see possible effects caused by our modifications. We also characterized the commercial cake from the vending machine, as you can see here on the left. And you can observe an interesting difference between the pore size from the 100 cake and the 75 GOS cake. So in fact, during the baking process, the sugar promotes the formation of micropores due to their interaction with the surface tension of the water, avoiding what we see here, which is the coalescence effect. And because the water stays entrapped inside the cake, it doesn't grow as well as in here. This is the physical characterization after 1 day of baking, and you can see differences between the height. In terms of volumic mass, the 75 GOS is more similar to the commercial than the 100 cake. And the addition of GOS and sugar reduction did not change the color of our cakes, although they differ slightly from the commercial one. So now we'll skip the rest. We measure the textural performance analysis by 2 compression cycle, and this is the TPA after 1 day of baking the cakes. So you can see bigger differences between hardness, gumminess, and chewiness related with the required energy to chew the cake. So reducing the sugar promotes the interactions between the proteins and the starch because of the free water that is not interacting with sugar because it's in less proportion. And this possibly explain why these parameters increase. These results are in agreement with the literature reports for muffins. So hardness increased with time as expected, and these results are interesting because they show that after 3 days, the cakes will have a different EPA, which gives us an indication of a possible shelf lifetime. This is our team involved in the work, some familiar faces from the organization of this workshop. So just to conclude, we believe that this product is a kickoff to target a market characterized by consumers who like traditional pastry products, but want to pursue a healthy lifestyle or even with medical conditions, such as diabetes, for example. So thank you for listening. If there are any questions, I'll be pleased to answer.
Thank you, Claudia, for your nice presentation. We have some questions here, such as, is this product ready to be launched in the market? Or if not, what additional assays do you foresee before it can be introduced in the market? Okay, that's a really nice question. So we are at an early stage of the product development, and this is just a prototype that we'll want to further improve. For instance, to overcome the impact of sugar reduction on the cake physical features that are presented, we can, for instance, incorporate xanthan gum to reduce the qual-sense effect, and so we'll have cakes that are more equivalent, and consumer will like them better. But we also want to replace the milk and eggs for vegan alternatives. And we also envisage to increase the shelf lifetime of the product. And after that, we may start by introducing the product in the market using local Portuguese pastry shops, for instance. But besides that, we have to establish how many cakes the consumer can eat maximum per day, for instance. That's also something that we need to do. Okay. And are you considering other food matrices for prebiotic incorporation? Yeah, actually, we started by choosing this cake because of the simplicity of the matrix. It was the first time that we were doing, and because it's a widely consumed cake in Portugal. So we started by this matrix, but we also foresee, for instance, different traditional pastries, not only Portuguese traditional pastries. Okay. And the last one, do you think that in the future we can have personalized cakes? That will be wonderful, but I think it's a future that is it's a little bit difficult to understand right now, because first we need to establish how we can modulate the microbiome in a personalized way. And after that challenge, we can for sure thinking about personalized diet for each different person, including to treat disease. Okay, thank you, Claudia. We don't have more questions. It was a very nice talk. So now we'll have with us Fatima Duarte that will present the last key project from the morning session. Fatima, you can start sharing your screen. Fatima is a researcher at CEBAL, that is the Agriculture and Agri-Food Biotechnology Center of Alentejo. Her research focuses on the use of different agro and forestry industry raw materials and residues as a source of valuable ingredients for new biotech applications. Fatima, the floor is yours.
Thank you. I hope that everyone is already seeing my screen, right?
Yes, yes.
So thanks for the nice presentation. And thanks also for the organizing committee for inviting me, for being here today presenting the work of my research team. So today I'm going to talk about the natural sources of cis-quaterpine lactones, and I'm going to give you as an example the Cinera carbunculus as case study. So we are pretty much all aware about the use of plants as natural sources for medicines, either for its use as pharmaceuticals or to be used or to transform into herbal remedies. In fact, there is a wide range of different chemical entities which might be extracted from plants, being the cis-quaterpine lactones one of the largest family with more than 5,000 compounds. This family of compounds have a wide range of biological activities. And to the purpose of today's meeting, I'm going to be focused on two of them. So I'm going to talk about anti-inflammatory properties regarding the biomedical applications, and I'm going briefly to speak about phytotoxic activity and how we can use it for the development of a bioherbicide. So coming to the point where I was speaking about cis-quaterpine lactones, which can be isolated mainly from members of compositae plant family. And here on the left side, you can see different examples of these plants. Some of them for sure are known for you, but today I'm going to focus and I will call your attention to this plant, the Cinera carbunculus, commonly named cardoon. So my research team started to be interested in this plant when we performed the chemical characterization of lipophilic extracts. So basically we divided the plant into stalks, capitula and leaves. And indeed, the results demonstrate us that leaves are a rich source of cis-quaterpine lactones. It's the major family present in cardoon leaves with a concentration close to 95 grams per kilogram of dry weight. Moreover, when we observed or when we characterize the different cis-quaterpine lactones present, we identified cineropicrin as the main cis-quaterpine lactone present in cardoon leaves, with a concentration over 87 grams per kilogram of dry weight. So at this point, we were completely convinced that Cinera carbunculus leaves were a natural source of several bioactive compounds, among them several cis-quaterpine lactones, in which cineropicrin seemed to be the prevalent one. So if we look to an overview of the potentiality of Cinera biomass, and in this graph here, in the calculations of 1,000 kilograms of whole biomass, we can achieve more or less 400 kilograms of leaves and head remains, and from which we can extract 1.10 kilograms of cineropicrin. This was already a great result, and it was indeed promising for what we wanted to do next. And we challenge ourselves to increase cineropicrin content on the leaves extract, following a concept of environmental friendly extraction. We were looking for a process from which we could have more cineropicrin and using environmentally friendly extraction process. We start by looking to different extraction methodologies, conventional and non-conventional ones with different solvents, and we end up with an optimized process using ultrasound assisted extraction, using solvents, using as solvent ethanol, and according to the results, we were able to increase in 30% the extraction yield of cineropicrin, and we were able to reduce in 97% the energy consumption. Follow this, we wanted to promote a fractionation in order to achieve cineropicrin enriched extracts, even more enriched extracts. And to do so, we developed a process, as already mentioned, based on DNA nanofiltration, followed by a nanofiltration process, in which we got two fractions. One of these fractions was rich in carbohydrates. We were able to remove 93% of glucose and 96% of fructose, and these just represent a maximum loss of 13.9 of syneropicrin. So at the end, we had two streams. This one rich in carbohydrates, and this one rich in syneropicrin, which was our main objective. This enriched fraction of syneropicrin presented a higher biological activity. So it was the process itself was working as we planned. So next we start developing different strategies. And today I'm going to speak or to share with you the work we performed regarding the wound dressing for anti-inflammatory drug delivery. So as you can see here, we performed the development of different films, chitosan films, supplemented with different percentage of syneropicrin-rich extracts. And we then evaluated the anti-inflammatory properties using the normal human skin fibroblast cell line. The cell lines were stimulated with LPS, leading to the production of EL6, a pro-inflammatory key element. And the results evidentiated that when we have the films supplemented with 5% of syneropicrin-rich extracts, we observe a significant reduction on EL6 production or expression. Moreover, when we compare the results we obtain with extract versus the results we are obtaining with syneropicrin, the pure compound, it was relevant to observe that the extract has a more biological potential than the syneropicrin by itself, most probably evidentiating the synergetic effect of the extracts. So with these results, we were able to demonstrate that this supplementation of chitosan films had here a potential for wound management, and indeed, in the present, we are still following this research line. The other topic I wanted to briefly show you is the development of a bioherbicide. And this has mainly to do with this intensive use of synthetic herbicides that leads to bioaccumulation of synthetic compounds that ultimately lead to an herbicide resistance development. And therefore, as we need to find different alternatives. One of these alternatives is the use of allelochemicals. And within the different allelochemicals, which have promising or which represent a promising tool for sustainable weed management, we have the cis-quaterpine lactones. So the strategy we performed was to look into the major or the cis-quaterpine lactones which are present in cardoon. We select those which are more expressed or which are prevalent and start the development of this bioherbicide. We use the membrane technology to obtain enriched fractions once again. And as you can see here, the next steps are going to be the evaluation of this phytotoxicity. This is a work in progress, which we are currently developing with the University of Cádiz in Spain. So to sum up, I hope to, in this briefly presentation, to show or to convince you that cardoon leaves are a natural source of cis-quaterpine lactones. This is one of the strategies we are exploring regarding cardoon circular bioeconomy, where we want to combine research, production, and industry for development of new knowledge, development of new skills, and also development of new solutions. So just to, just to finish and acknowledge those which participate in this work, my research team, the funding agencies, and thanks also for your great attention. And I will be happy to take any of your questions. Thank you.
Thank you, Fatima, for your interesting talk. We have several questions ready for you. So the first one, most voted, is how common is Cynara cardunculus in Portugal, Europe?
It is common more in the interior part of Portugal. So Cynara used to be quite well spread in national territory, but mainly due to the intensive agriculture systems, it has been demonstrated that the areas of this plant is being reduced. So in terms of Europe, there is a great, I will say, productivity in Spain and also in Italy.
And what are the main limitations of your process?
You mean the process in terms of production in the field, or maybe in terms of technology, right?
Probably technology.
Yeah, I guess so. So basically, one of the things that we are interested also, and obviously I didn't demonstrate here because lack of time, but is the economic viability of this process. And so far we evaluated how could we manage the scaling up, and I'm already taking the next question, how we could scaling up this ultrasound process. And yes, it is easy to scale it up. And also we were aware of the fact that membrane process leads to an investment, to a significant investment. So according to our results and the economic assessment which was done, in 3.5 years, you can have the payback of the system due to the quantity of cinnaropicrin you can get.
Okay, thank you. And the application in wound dressing for anti-inflammatory drug delivery is very nice. What are the main barriers to have it in practice?
Yeah, so thanks for this question, which is really pertinent because yes, it's true that results are very promising and we are still working on this because in terms of the material, as we look to the film itself, we still have to work on it. We hope simple things that have to be optimized, but we are on that point. Also to understand more how the drug delivery happens in terms of pharmacology. And basically, it's the point where we are at the present, yeah. So the next.
You also have here one related that if your extracts are not pure, can you have other compounds such as polyphenols that contribute to the anti-inflammatory activity?
Yeah, this is also a very good question. So obviously I did not have time to go into that detail, but we don't have that much phenols on this extract. So this extract was extensively chemically characterized, and the membrane process really helped to clean up too much these extracts, yeah.
Okay. And do you have any application for the 500 kilograms of stalks?
Yeah, this is great also because as I mentioned in the end, so our intention is, or we work under this umbrella of bio economy around cartoon. So the idea is to valorize all the fractions. And yes, it's true that stalks is a considerable rich source of lignocellulosic compounds. So our idea is to develop, integrated with other teams at national and international levels, different solutions for the stalks, which might pass through the development of materials, new materials for construction.
Okay. And the last one that probably you already focus on, do you aim to test these compounds on a more complex in vitro skin model?
So, yes, we want to do that,
yeah.
Obviously. Yeah. So far, we did not perform it, all of it, but is one of our aims, obviously, yeah.
Okay, thank you. Don't have more questions. Thank you for coming. It was very nice to have you here with us.
Thanks for having me.
So now we will proceed with the flash presentations. We'll have 6 consecutive flash presentations. Some of these presentations were previously recorded by the speakers and the organization will share them. In the end, if you have time, the speakers will answer the most voted questions before the lunch break. The first flash presentation will be from Ruth Limon from the University of Coahuila. Sarah, can you please share the presentation?
My name is Ruth Cubas, and today I would like to tell you a little bit about my research project entitled Evaluation of Bioactive Compounds and Antioxidant Activity of Aloe Vera Juice Fermented with Probiotics. This project is based on the development of a functional beverage based on the fermentation of aloe vera with probiotic bacteria and evaluate the biotransformation of bioactive compounds during the fermentation of aloe vera with Enterococcus faecium and Lactococcus lactis. So, why use aloe vera? Well, aloe vera has been used as a traditional medicine all over the world due to its chemical components and for its healing and therapeutic properties. In recent decades, aloe vera has had an impact on the development of functional foods. So, the experimental strategy that we followed consisted of the aloe vera gel was extracted and then extraction with water plus cellulase was performed to obtain the aloe vera juice. Aloe vera juice was inoculated with 2 bacteria, Enterococcus faecium and Lactococcus lactis, having 2 fermentations. During fermentation, we evaluate the biotransformation of compounds, namely total carbohydrates, organic acid, total phenolic compounds, and individual compounds, and antioxidant activity was determined using 3 methods: FRAP, DPPH, and ABTS. And we found that aloe vera sugars were consumed during fermentation with Enterococcus faecium and Lactococcus. Therefore, both bacteria were able to use aloe vera's carbohydrates as a substrate and a source of carbon. Enterococcus and Lactococcus mainly produce lactic acid during the aloe vera juice fermentation. Other organic acid identifiers were acetic, formic, propionic, and isobutyric acid. In general, the production of organic acid is related to dietary carbohydrates like asamannan and glucomannan from aloe vera has been reported to promote the increased production of metabolites such as short-chain fatty acids. No significant differences were found in the total phenolic content along with the 2 fermentation. Thus, Enterococcus and Lactococcus didn't degrade the phenolic compounds from the aloe vera juice. 14 phenolic compounds were identified in the aloe vera juice, like hydroxybenzoic acid, laquanilic, alagic, and HBA, dehydroxycinnamic acid, like picumaric, chlorogenic, and ferulic, flavonols like camphorol, flavanthriols like epicatecherin and catechin, stilbene like resveratrol, flavonol like spiridine and naringine, and flavone like quercetin and taxifolene, and anthracene like alwine. The result of staining for antioxidant activity with FRAP-SA shows that the fermentation with Enterococcus faecium had a slight increase was observed. However, the results for Lactococcus showed that the antioxidant activity was maintained during fermentation. For DPPH, the amount was maintained at 48 hours of Enterococcus faecium fermentation, and the DPPR results for Lactococcus fermentation indicate an increase which showed that fermentation doesn't negatively affect antioxidant activity and promotes the release of antioxidant compounds. The ABTS method shows an increase in antioxidant activity of aloe vera juice fermented with Enterococcus and Lactococcus. There was no significant differences in antioxidant activity between fermentation with Enterococcus and Lactococcus. The increase in antioxidant activity can be influenced by the metabolism of phenolic compounds, and some organic acids and sugars have antioxidant activity. This means that probably the increase observed in the antioxidant activity is related to the organic acids produced or synergistic effects between the phenolic compounds and sugars. In this sense, the consumption and presence in the organism of a beverage rich in bioactive compounds important since they aren't produced by humans. Fermented aloe vera juice can be an interesting source of bioactive compounds, mainly antioxidant, prebiotics, and probiotics, promoting consumer health. So, thank you very much for your attention.
Thank you, Ruth, for the nice video. Now we'll have Cynthia Ramos from Federal University of Jequitinhonha and Mucuri Valles. You can start sharing now, please. Cynthia, are you there?
Yes, I'm here. Sorry, how to do to share my screen?
In the bottom at green, you have share screen. Green arrow.
Yeah, yes. Sorry. Okay.
Yes.
Can you see?
Yes. You can start.
Thank you for organizer for inviting me. I'm going to show you a brief presentation about some results that we have done here in my university with my team. Thus, I'm going to present for you fermented food as a reservoir of probiotic strains. Probiotics are living microorganisms which, when administered in adequate amounts, confer health benefits on the host. Some strains of lactic acid bacteria, bacillus, propionibacteria, and some yeast have demonstrated probiotic properties. Spontaneous fermented foods have different characteristics. We have different spontaneous fermented foods with different characteristics, then showing different diversity microbiota, which makes it important to study strains from these different foods, these different sources. Some products that we have used in our work is here indigenous fermented foods called the Cauim, Iacuba, Calugi, Caxiri, and Xixa. These foods are produced by Brazilian Amerindians. They are spontaneous fermented foods. Cocoa fermentation is a key process to chocolate production. Is a complex fermentation with a diversity of lactic acid bacteria, yeasts, and the acetic acid bacteria. Kefir is a symbiotic beverage from milk, and Minas artisanal cheese that is produced in Minas Gerais states in Brazil from raw cow's milk and pingo, that is the endogenous ferment. A common characteristic of these products is the diversity of lactic acid bacteria and the yeasts in these foods. The microbiota of these products have been described. However, the potential probiotics is still poorly studied. Thus, the end of our studies, the main end of our studies is the probiotic characterization of lactic acid bacteria and yeasts isolated from different fermented foods, and evaluation of potential probiotic strains as a starter culture to produce non-dairy fermented foods. As methodology, we have used different in vitro evaluations, because a probiotic should survive through gastrointestinal tract and enhance intestinal functions or promote health benefits. Thus, different in vitro tests such as the tolerance of gastric The tolerance to acid and bile, adherence to epithelial surface, and the others have been done. The main finds of our work is the potential, we can say that the potential probiotic of lactic acid bacteria isolates from different Brazilian fermented foods. We can say that probiotic properties is strain specific. Some strains of lactic acid bacteria from cowin and from cocoa have shown interesting in vitro probiotic properties. A strain of Lactobacillus plantarum were able to reduce colonization and invasion of the IPEC and the Salmonella in CACO2 cells and the AT29 cells, and also exhibit an in vitro antimicrobial activity towards these pathogenic bacteria. Lactic acid bacteria isolated from indigenous fermented foods were potentially effective in reducing phytates in foods. One Lactococcus lactis showed it to be suitable to as a starter culture for fermenting yams. And this dough was used to produce ice queens, and these ice queens were successfully accepted by the consumers. Regarding yeasts from the Brazilian fermented foods, we can say that 12 Saccharomyces cerevisiae and 1 Pichia cluiveri isolated from caxiri, kefir, and the cocoa fermentation showed in vitro probiotic properties, and also the cells showed high antioxidant activity. These yeasts were able to produce phytase, that is an enzyme important for nutrient availability in plant-based foods. Among these yeasts, two strains of Saccharomyces cerevisiae from caxiri and the Pichia cluiveri from cocoa were used in single and in co-culture with the commercial probiotic bacteria, the Lactobacillus paracasei, to ferment a maize-based substrate. The co-cultivation of these strains of Saccharomyces cerevisiae and the bacteria produce a beverage with suitable probiotic population, a great number of volatile compounds, and the consumers neither disliked nor liked the beverages, which is a positive result since we have not adding no flavoring to this beverage. The semi-yeasts, Saccharomyces cerevisiae, these 3 strains of Saccharomyces cerevisiae in Pichia cluiveri were able to inhibit the addition of some pathogenic bacteria, as we can see here. The first here is the control. These yeasts were able to reduce up to 50% of the bacterial infection, as evaluated by laser confocal microscopy. And here we can see is the control, and here is the bacteria, the yeasts, for the exclusion experiment assay here, and for the competition assay in this side. And we can see that we have found the less fluorescence here. No antimicrobial compounds were produced by the yeasts, showing that the competition for nutrients and or receptors in the intestinal mucosa was the mechanism to bacterial inhibition. So the main conclusion of our work here is that potential probiotics were identified and characterized from different fermented foods. Probiotic characteristics are strain specific, and the characterized probiotic strains, lactic acid bacteria and yeasts, have demonstrated potential to be used as a starter culture in single and in co-culture to produce non-dairy fermented foods. Thank you, some references, and thank you for your attention.
Thank you, Cynthia, for the interesting presentation. Now we'll have the presentation from Gabriel Valtério from Federal University of Rio de Janeiro.
Hi, my name is Gabriele and I'm postdoctoral researcher at the Federal University of Rio de Janeiro. Today, I will tell you a little bit of my PhD research. Well, I did my PhD in Food Engineering Science at the Federal University of Rio Grande, Brazil. My thesis was entitled Production and Characterization of Xylanolytic Enzymes and Xyloligosaccharides obtained through agricultural substrates. Xyloligosaccharides, also called XOS, are non-digestive oligomers that show several interesting properties for human health, which include antioxidant and prebiotic activities. They can be produced by thermal, chemical, microbial, and enzymatic strategies, or by a combination thereof. XOS production by enzymatic reaction shows several advantages including enzymatic specificity, non-formation of toxic compounds, and mild reaction conditions. However, the high cost of enzyme can be a limiting factor for XOS production, so the xylanase production using low-cost substrate has been extensively explored. In my thesis project, the production of microbial xylanase using rice bran as a xylan source was evaluated. Xylanase was produced by a yeast-like strain using a byproduct of rice without any pretreatment. After the optimization of microbial cultivation, an enzymatic extract with a xylanase activity near 80 U per milliliter was obtained. Then I had a crude xylanase extract with high activity. So how can I apply it in xylan hydrolysis? Firstly, it was necessary to know some properties of the crude xylanase, including optimal temperature and pH, thermal stability, the presence of other xylanolytic enzymes, and if the extract needed or not a purification step. Next, the XOS production by enzymatic hydrolysis was fully investigated. Xylan hydrolysis using enzymes involves some important parameters that needed to be established to achieve agreed yield. Moreover, these parameters also influence the carbohydrate composition of the hydrolysate. In my thesis project, the effect of temperature, reaction time, type of agitation, xylan concentration, enzyme loading, and pH were evaluated in XOS production. At the end of enzymatic hydrolysis, through an optimized reaction, it could be achieved a hydrolysate with high content of total XOS and short XOX and low xylose content. In 2019, I did a 2-month exchange period at the Center of Biological Engineering at the University of Minho. During this period, the antioxidant and prebiotic activities of the hydrolysate were evaluated. The hydrolysate showed antioxidant activity through different methods. Also, the hydrolysate showed prebiotic activity, that is, high production of lactate and short-chain fatty acids, stimulation of megamonofuniforms, high production of gases, reduction of Methanobacteries family, agreeing with low methane production, and finally, the reduction of Clostridium bacteria, agreeing with low ammonia production. Thank you so much for your attention.
Thank you, Gabriel, for the great video. Now, Beatriz Cardoso from Center of Biological Engineering will be presenting. Beatriz, you can start sharing the slides, please.
Is it working?
Yes, yes.
Okay, so good morning, everyone. First of all, I would like to thank the organization for the opportunity to show my work in this workshop. And so today I will talk about prebiotic biosynthesis using cellulose to epimerase produced by Saccharomyces cerevisiae. So according to the most recent definition, a prebiotic is a substrate that is utilized by host microorganisms, conferring a health benefit. As prebiotic consumption has significantly increased in the last years, new strategies for its sustainable production are emerging in this research field. So prebiotics can include, for example, oligosaccharides, polyunsaturated fatty acids, phenolic compounds, and also dietary fibers. The class of the oligosaccharides are the main focus of our research, and they can include, for example, galacto-oligosaccharides, fruit-oligosaccharides, and lactulose that are well established already as prebiotic and used worldwide. But also we can have epilactose that is still considered as an underdeveloped prebiotic. So this being said, our strategy and our focus was to develop a new strategy for the production of lactulose and epilactose. In that sense, we decided to use the enzyme cellulose to epimerase that is able to produce both lactulose and epilactose using only lactose as a single substrate. So using the lactose, this enzyme is able to catalyze the epimerization of the mannose molecule from lactose into the glucose molecule of lactose into a mannose, thus generating the epilactose. And then in a second step, the mannose molecule of the epilactose is then isomerized into a fructose, producing the lactulose. This being said, our strategy was to clone the gene of this enzyme, cellulose to epimerase, from this bacteria into using as host the Saccharomyces cerevisiae, taking advantage of its GRAS status and also the fact that Saccharomyces cerevisiae is not able to metabolize lactose, allowing then a total conversion in prebiotics. After the assessment of the optimal conditions of the enzyme that were found to be 80 Celsius degrees and pH 7.5, we went to study the prebiotic production. After the recovery of the intracellular enzyme, we started by conducting reactions of 20 minutes with different concentrations of lactose. We found, as expecting, that higher the substrate concentration, higher the prebiotic production, as you can see here in the green-blue line. And also concerning the yield, we verify that it decreases with the lactose concentration, as shown here in the orange line. This fact was also actually a good result, because it means that we can use, for example, our enzyme in the dairy industry, because to improve the quality of the dairy products that are usually they have usually a low content of lactose. So in these 20-minute reactions, we only detect the production of epilactose, and so we decided to conduct the reactions for 2 hours. In this figure, you can see that lactulose was produced after 30 minutes, and its concentration increased until 1.3 grams per liter, representing a yield of 2.6%. And in the case of epilactose, it reached a maximum of 13.5 grams per liter and a yield of 27%. So summing up, this work showed for the first time that Saccharomyces cerevisiae is a suitable host to produce cellulose to epimerase towards further applications in the prebiotic production. The epilactose yield of 27% is the highest reported so far using a grass microorganism as enzyme producer, and that this enzyme is more suitable for the production of epilactose-enriched prebiotic mixtures with potential applications in food industry. So that's it for today. Thank you all for your attention, and I'll be glad to answer any questions you may have. Thank you.
Thank you, Beatriz. It was very interesting. Now we'll have João Rainha from Center of Biological Engineering. You can start sharing the slides, please.
You're seeing, right?
Yes, yes.
So good morning, my name is João Rainha. I'm a PhD student at the Center of Biological Engineering, and I'm going to, my presentation for today is exploring the Saccharomyces cerevisiae as a curcumin producer. Curcumin is a secondary metabolite produced in the rhizomes of a plant called Curcuma longa. Chemically, it belongs to the polyphenol family, and it's known as an antioxidant and as a yellow pigment. Curcumin is also an interesting molecule because several studies reported its therapeutic effects. The anti-cancer activity is the most studied effect. However, other biological activities are reported, such as in wound healing or in protection against neurodegenerative diseases, making curcumin to reach an estimated market size of more than $100 million by 2025. However, to perform clinical trials, high purity extracts of curcumin are required, and the curcumin production by cultivation is associated with geographic limitations, with a constant crop control, and takes long time periods. In addition, curcumin accumulates in low amounts in the plant, and it is difficult to isolate. The chemical synthesis could be another approach, but this process is very complex, making the production of curcumin very expensive and associated with pollutants. So, our idea is to develop a genetic engineered Saccharomyces cerevisiae that can be used as a cell factory to produce pure curcumin. For that, we need to understand how curcumin is synthesized in the plant. Curcumin biosynthesis begins with the conversion of aromatic amino acids into ferulic acid by several reactions, and the ferulic acid is then activated through a condensation with a CoA molecule, and next, in the two-step reaction, it is converted in curcumin with the involvement of malonyl CoA. So, to evaluate if yeast is able to produce curcumin, we decide to design a heterologous pathway that will allow us to convert ferulic acid in curcumin. The enzymes that we are first using were 4-CL from Arabidopsis thaliana and curcumin synthase from Oryza sativa that is able of synthesize the synthesis is able of synthesize curcumin in only one reaction. The column optimized versions of these genes were placed in were cloned in the yeast expression vectors under control of constitutive promoters, and we transformed this plasmid in our Saccharomyces cerevisiae wild type strains. In this case, the mutants were labeled as B4C. To produce curcumin, we inoculated B4C in minimal media, and different concentrations of ferulic acid at different time points of fermentation were added to the media. Interestingly, only 16 milligrams per liter added at 24 hours of fermentation resulted in the curcumin production, and this can be due to the toxic effect of ferulic acid to yeast cells. After fermentation, the media was removed and curcumin was extracted from cells using methanol. The HPLC analysis revealed that 111 micrograms per liter of curcumin were produced. So next, In order to test if the use of rich media would improve curcumin productivity, we replaced in the plasmid the oxotrophic marker by an antibiotic marker, and using this plasmid, we were able to produce 150 micrograms per liter of curcumin. Besides, curcumin was produced in yeast. The production yields remain very low, so we decided to test if the actual two-enzyme system from curcuma longa plant would result in better productivity. So we designed a plasmid as previous, and transformed this plasmid in our wild-type strain, and we labeled the mutants as B4DC. Actually, using the two-enzyme system, higher curcumin amounts were produced, as can be seen in these pictures. First, here in the flask fermentations, due to the yellow color formation in the culture media, also here in the pelleted cells and in the extraction. The HPLC analysis revealed that using this path, the 1.3 milligrams per liter of curcumin was produced, more than 8 times more curcumin produced than with the one enzyme system. Next, to prevent some ferulic acid consumption by yeast, we used CRISPR-Cas9 to knock out a gene that is able to degrade ferulic acid in Saccharomyces cerevisiae, and after the deletion of the gene, we transformed our pathway in this new strain, and the curcumin productivity with this strain reached 1.7 milligrams per liter of curcumin, the highest value reported. So, to finish, the biosynthesis of curcumin was accomplished in a genetic modified Saccharomyces cerevisiae. The genes from curcuma longa resulted in higher curcumin yields, and in the future, the genes from the phenylpropanoic pathway will be introduced to synthesize curcumin from simpler substrates. Thank you for watching.
Thank you, João, for the nice talk. Now, the last flash presentation is from Marcia Couto, also from Center of Biological Engineering. You can start sharing your presentation, Marcia, please. We are seeing your screen. Marcia, we can't hear you. Are you having problems with the sound? Sarah, maybe it's better to share the video from Marcia that she previously prepared, please.
Hello, can you hear me?
Yes.
Okay, can I make the presentation? I was not able to.
If you are fast.
Yeah, of course. Sorry about that. So, hi to everyone. Today, I will show you the work on alternative enzymes for glycosaminoglycans biosynthetic pathway design. Glycosaminoglycans are important molecules with nutraceutical, cosmetic, and pharmaceutical applications. My work is currently focused on chondroitin. Being a natural structural component of joints cartilage, its major application is as a dietary supplement for osteoarthritis. But it is also used in eye drop solutions, cosmetic products, and animal food. Usually, this compound is extracted from animal cartilage, which brings risks of pathogen contamination and requires environmentally hazardous extraction. Therefore, microbes have emerged as safer and more controllable alternative sources. The current biotechnological production of chondroitin is based in the well-known pathways that pathogenic bacteria use to naturally produce GAG-like polysaccharides. However, the yields are not sufficient to meet the increasing demand, and non-GRAS microorganisms have mostly been used. Also, eukaryotic genes and hosts have not been properly explored. So the goal of this work was to evaluate alternative enzymes and hosts for biotechnological glycosaminoglycan production. The enzyme UDP glucose dehydrogenase, UGD, was the focus of this work since it is reported as the major limiting step for GAG's production. 3 UGD genes were evaluated. The genes from Capra hircus, Ximomonas mobilis, and Lactobacillus johnsoni were expressed in 2 types of microorganisms, Saccharomyces cerevisiae and E. coli BL21. After culturing the recombinant microorganisms, the cell extracts were used for in vivo UDP sugars quantification and for in vitro reactions. Regarding the results obtained, the highest UDP glucuronic acid in vivo production was achieved using Capra hircus or Ximomonas mobilis genes. However, in the in vitro assays, Lactobacillus johnsoni's UGD showed the highest activity using either E. coli or Saccharomyces cerevisiae as the expression hosts. The enzymes were also characterized in terms of optimal pH and temperature. Generally, the enzyme activity was optimal at a pH between 7 and 8 and a temperature of 30 degrees Celsius. In conclusion, it was demonstrated that the 3 enzymes were actively producing UDP glucuronic acid in vivo and in vitro in both types of hosts. The gene from Capra hircus allowed the highest concentration in vivo reported so far. On the other hand, in the in vitro reactions performed, the recombinant enzyme from Lactobacillus johnsoni was the most active. Finally, the yeast seems to be a promising host for glycosaminoglycans production. Thank you for your attention. I hope I was fast enough. And I will now answer your questions.
Thank you, Marcia, for interesting talk. Now I'm going to share the questions on Slido, and we are going to answer the most voted ones. The first one is for Ruth. Ruth, can you answer this? Why did you choose these microorganisms for the fermentations?
Okay. Can you listen me?
Yes, yes.
Okay, because in the working group, previously Enterococcus faecium was isolated and it was shown that it's a good probiotic bacteria. And Lactococcus is used as a control, since it's a commercial probiotic bacteria, and they are probiotic bacteria that improve the flavor in the fermentations, in addition to production bioactive compounds.
Okay, thank you, Ruth. Now the next question is for João. Do you consider that your approach can be competitive with the process already available in the industry?
Can you hear me?
Yes.
Yes, because we are talking about that. We are talking about a compound that is produced in a plant. So the plant takes a long time to grow. So if you are comparing the time of production, we are decreasing it by a lot. That because yeast ferments in 3-4 days, and also when we extract curcumin from the actual plant, not pure curcumin is obtained, but a mixture of curcuminoids that can cause misunderstanding of the clinical trials. So I think that my process is better.
In the future, without optimizations, right?
Yeah.
So thank you, João. The next one is for Gabriel. What is the importance of the stimulation of Megamonas funiformis? Is there any report of the stimulation of this bacteria by other exose mixtures?
Hi everyone, can you hear me?
Yes, Gabriel.
Regarding health aspects, some studies have been reported that the reduction of this bacteria in the gut microbiome community of patients with multiple system atrophy and Crohn's disease, being this species present in healthy individuals and associated with benefic microflora. This stimulation is also important for production of acetate and propionate, since Megamonas species are recognized as producers of short-chain fatty acids. About the second question, the fermentation of the hydrolysate containing XOS is a new topic of study, but it agrees with some studies reporting the ability of Megamonas funiformes to utilize lactulose and galactoligosaccharides.
Okay, thank you, Gabriel. So next one is for Cynthia. Cynthia, do you expect to test in vivo the effect of those foods on human health?
Yes, can you hear me?
Yes.
Yes. Okay. Yes. Now, of course, it's the next step of our study. Now we are doing some evaluations. We select some strains, and we are doing some evaluations in animal models. And then if we have this safety to use, we are doing to do these assays in humans too. So this is the next steps of our work.
Okay, thank you. Thank you, Cynthia. Thank you. Next one for Beatriz. Beatriz, do you foresee scalability issues?
Yeah, of course, because it has all processes have some issues that need to be considered. But for example, now we are working with an intracellular enzyme, so we need to extract it from the cell. And I think in terms of scalability, this could be one of the main bottlenecks in this process. We are hoping to move to an in vivo process. Let's hope it works. I think that's it.
Okay, thank you. Marcia, similar question. What are the main limitations you foresee in this process?
Okay. The main limitations, I would say improving the yields and the scalability of the process. I don't know any more specific. The main challenges I have now is the analytical methods. They need to be very specific for these compounds.
Okay, thank you, Marcia. Thank you, everyone. We don't have more time for live questions. Thank you, Ruth, Cynthia, Gabriel, Beatriz, João, and Marcia for coming. It was great to have you here with us. I also want to thank to all the fantastic speakers that we had this morning. And I also want to thank everyone that was watching this session regarding biotechnology applications in food and health. We are going to do a break for lunch. Please join us in the afternoon at 2 p.m. for the session Biotechnology Applications in the Environment. See you soon.
Okay, good afternoon. Welcome back. For those that have been in the morning session, welcome all the ones that are joining us now for the afternoon session. We will be starting right away. I just want to leave you some housekeeping rules and some short guidelines. So as you know, audios and videos are off. If you are the speaker, please let us know that you have arrived and send us a private message so we can put you as a co-host and you can share your slides. Any questions should be placed on the Slido. We will share the link in the chat in a moment. And then the moderator will pick the most voted questions in the interest of time. All the unanswered questions may be raised directly to the speakers through the chat on a private message if you want. And also be aware that we will be sharing a link to register the afternoon session, so we can later on send you the certificates. So any issue that you may have, please send us a message. We are identified as organizers, okay? And yeah, so this being said, I wish you a very nice afternoon, a very nice session. Enjoy the day. And I will also would like to thank once again the speakers for sharing their experience and their knowledge with us. So I will leave the floor now to Sara. Sara Silveri will be moderating the afternoon session. Sara, please go ahead.
Hello. So hello, welcome to the afternoon sessions. We will start with the keynotes presentations, and our first speaker will be Susana Lopes. Susana is a senior technician at Lipor, a waste management service of Greater Porto, Portugal. He has a large experience in waste management and transformation. She is also a board member of the European Compost Network. Please, Susana, you can start sharing.
Hello, good afternoon to all. So I will try to share my screen. Let me know if you all see the presentation. It's okay?
Yes, it's okay.
Yes.
Okay, so first, on behalf of Lipor and the European Compost Network, it's a pleasure to participate in this interesting and also inspirational event. So a big thanks to all. In terms of my agenda, so I would like to address in these few minutes some topics such as why is bio-waste relevant? I think we already saw during the morning some introduction about bio-waste EU policies, how to turning challenge into opportunities regarding bio-waste. And then I would like to share with you our experience as a waste management system and in terms of bioeconomy practices. So as we know, the climate emergency and the continuous loss of biodiversity, among obviously other environmental issues, show the importance to act and to promote an effective transition to a circular economy model. In this meaning, bio-waste has a key role in this transition. So I hope this during this session and in this day, we can learn a little about the set of opportunities we have to deal with bio-waste. So why is bio-waste relevant? Bio-waste accounts for more than 34 to 40% of the municipal waste generated and is the largest single component in our bin in municipal waste. Besides, land degradation affects roughly 1/4 of the global land surface. What means about 75 million tons of fertile topsoil are lost each year. So this is a huge amount of organic matter that our soils need, and we have this organic matter I will just do this better, in our bin. So we have here a lot of organic matter and valuable nutrients, and our soils need this organic matter. Beside that, the new Waste Framework Directive imposes new and very ambitious targets, including obviously mandatory separate collection for bio-waste by the end of 2023, a binding landfill target to reduce landfill to a maximum of 10% of the total amount of waste by 2035, and we need to reach carbon neutrality agreement by 2050. So with all this in mind, we know now we understand why is bio-waste so relevant. Here, some bio-waste policies. The new Waste Framework Directive it's important to comply and to monitoring food waste from 2022 using a common methodology for all member states, mandatory food waste prevention programs, very important, mandatory separate collection for bio-waste or ensure recycling at source by 2023, and the very, very ambitious preparation for reuse and recycling targets. At the same time, we know that the farm to fork strategy reinforces food waste prevention. It's important to highlight that 1/3 of the edible food produced for human consumption is either lost or wasted globally. So food waste prevention is really, really important. Beside that is an aspirational target according to the Sustainable Development Goals to halve food waste by 2030, and the bioeconomy action plans. Again, as the main goals, deploy local bioeconomies, scale up bio-based products. And so this is a huge challenge for the EU member states and namely for Portugal. Here, the EU Circular Economy Plan, the main goals, again, reduction of food waste production in Europe, promoting high quality recycling, saving primary resources, and establish markets for secondary products. It's important to say that we will have in force the new EU fertilizing regulation by 2022. So here it's important to say that our bio-waste, mainly food waste from households, office, the HoReCa sector, retailers, and garden and park waste are a key waste stream with a high potential for contributing to a more circular economy, delivering obviously valuable soil improving materials or as a fertilizers, as well as biogas as a source renewable energy. But to achieve this and to close the cycle, we need to have in mind the separation of bio-waste at source is a basic condition for achieving high quality products and obviously this transition to a circular economy. Here, I would like to share with you some information about some data about municipal bio-waste generation per person and share of bio-waste in the municipal waste generated by country in 2017, just to explain and to highlight the amount of resource that we have in our municipal waste, that means in our bin. So in 2017, the EU 28 member states generated almost 250 million tons of municipal waste. That means 250 million tons of resources. In a circular economy, these are resources. And these, among these, we had 86 million tons of bio-waste. That means almost 90 million tons of bio-waste. Thus, approximately 100, here you can see, so this is the average, Portugal is in the average, that means 170 kilograms per person of food waste is wasted every year in the EU along the entire food value chain. So this is a huge amount of organic matter and other key nutrients that we are waste every year. Regarding Portugal, so the annual municipal waste production is about 5 million tons per year. And in terms of bio-waste, our municipal solid waste composition shows that 37%, that means 1.7 million tons is bio-waste. That means almost 180 kilograms per person here is again produce it, and most of this is wasted because we send to a landfill or we burn it. So there is a huge amount in our waste of valuable organic matter and very relevant nutrients. Here, just to show, because sometimes we think that food waste is in the production or in the processing. No, most of our food waste is in our households bins. So households are identified as the sector contributing the most to food waste. That means preventing food waste at home should be a responsibility for all. So we need to change our behavior as a consumer, and how we need to see and to change the way as we deal with our meals, with our healthy products, with everything. So we need to change here our behavior. Again, okay, we produce bio-waste, a lot amount. Are we manage this correctly? Not at all. So what we can see here, again, is how we are dealing in terms of bio-waste management in Europe. So we need to turn this challenge into opportunities. So here we can see, in average, for the EU member states, on average, in 2017, almost around 40% of municipal bio-waste was collected separately, while 60% of bio-waste ended up in mixed municipal waste and was lost for recycling. Again, because we send this almost 60% of bio-waste to a landfill or to an incinerator. Separate collection, it's important to say, is a key for using bio-waste as a resources in a circular economy. Now we go to Portugal. Well, we are not in the good position. In 2017, only 5% of the total of the bio-waste produced was separately collected in two areas, Greater Porto and Greater Lisbon, but in the most of the cases in pilot projects. So we have a huge effort to do in this area. That means this 5% represents only 8 kilograms per person year. But as I show you in the previous slide, we produce almost 180 kilograms of bio-waste per year and per person. So really, we need to think and to change our behavior. And obviously, the entities in charge of waste management need to improve the management system. Here is another study addressed by the European Compost Network, just to show the European potential from bio-waste. So here is the current situation and the amount, the jump that we can have if we disseminate separate collection and if we promote a high quality standard and treatment for bio-waste. So we produce around 12 million tons a year of compost. If we collect all the bio-waste that is produced among the EU member states, we can reach around 32 million tons of compost or digestate, obviously. That means a huge contribution to mitigate land erosion and to improve the health and the productivity of our soils. As we saw, they need urgently organic matter, and we need to preserve the soil. So to do that, we need to have all the time in mind the food waste hierarchy. That means more than to recycle, or before to recycle, we need to prevent, we need to reuse, then we recycle, then we can recover, like to take back the energy, and the least option is to dispose. So more than other waste types, preventing food waste should be perceived as an ethical responsibility for society. Here, just to highlight, and I think we all know the benefits of compost. So if we regularly applying compost to soil, we can reach several benefits, like increase, obviously, the soil organic matter, like improve soil structure. Obviously, it's a good, also regulates the water, increase the soil buffering capacity, increase the soil biodiversity, and we can also, it's important again to increase the resistance of crops to disease. It's also important not to forget that compost is a useful source of nutrients, including nitrogen, phosphorus, and potassium. So we have all these in our bin at home because we wasted a lot of food waste, and we just need to collect this waste, to transform these, and at the end we can have high quality products and to promote the sustainable use of this compost. So very important for this, and this event is showing this, the importance of the innovation, the importance of research, and here for the bio-waste there is several emerging opportunities for higher value products from bio-waste. Like, for instance, we know that innovation plays a key part in valorizing bio-waste. Anaerobic digestion with the biogas production is increasing. Biogas is a source of renewable energy. We can purify the biogas and produce like biomethane. And we know that research and innovation increasingly explore the opportunities for using bio-waste as a potential source of chemicals, products, and energy. For instance, we can take the volatile fatty acids, the biohydrogen, the biofuels, the biochar. We can transform bio-waste to animal feed and insect protein. We can transform bio-waste into bioethanol. So there is a huge amount of opportunities to create valuable products only using this huge amount of enormous amount of bio-waste. So just to stop this part, and then I will pass for our practical experience. So the bio-waste policies also in Portugal, we have this national strategy. You can find all the information. Here is the link. So we just go there and surf, and you will find all the goals, the guidelines, the enables, the task, and the targets that we need to reach. So first, we will have prevention targets, mandatory separate collection, and to do this, obviously, we need some enables. We need to have more informed decisions. We need to fight against food waste. We need to mobilize all the stakeholders, and to have good decisions, we need to know, we need to have reliable data, and like that we can act better. And obviously, and during this morning I heard, it's a key part is we need to more an effective regulation. So now I will pass to a practical experience. So how to promote a circular bioeconomy. So this is how a waste management system can implement, improve, and disseminate circular practices among its territory. So LIPOR, a glimpse about LIPOR. So LIPOR is a supramunicipal waste management system from Greater Porto, founded in 1982, 8 municipalities, almost 1 million inhabitants with a high amount of waste, almost 520,000 tons per year. So this is not waste. We need to change. We need to think in terms of resources. So we have an enormous amount of resources to transform and to create new products and with high value. So here is our vision, mission, and values, and our purpose. Just to highlight our mission, we want to transform waste into new resources. How? By implementing innovative and circular practices. And here is our circular economy approach very quickly. So since 2018, LIPOR has its voluntary commitment with the aim to implement and share circular practices at local and regional level. Our top priorities, our biodiversity strategy, our climate strategy that we call 4M, means less waste, less carbon, more less waste, less carbon, more climate, and more biodiversity. So it's for that we have here the fourth. Prevention projects, high quality recycling, but at the same time and always improve raising awareness, and we want also to improve sustainable public procurement practices. So in terms of bio-waste, our bio-waste circular strategy, the main challenge, obviously, prevent and reduce food waste, promoting the return of organic matter to the soil, to take part in this decarbonization path, and promoting biodiversity. How we do that? So with several projects in terms of food waste prevention, recycling at source by doing home and community composting, separate collection and centralized treatment in our composting site. And at the same time, we promote organic farming by using compost, sustainable gardening and biodiversity. And to do this, we need to have our citizens informed, well informed and committed. So we need awareness and communication all the time. So here you can see some important milestones. So it's not in one year we do everything. So we start long time ago, step by step, innovating and involving the society. So you can see here several projects, and now our main goal is to disseminate the door-to-door separate collection for bio-waste involving householders. So we start in 2005 with door-to-door separate collection from large producers, and since 2018 we start with this big project with door-to-door separate collection for food waste from householders. So this is our main task, and where we are putting all efforts to reach the national and obviously regional goals that we have. Here, very important. So first step is food waste prevention. So here you can see our several projects addressed to food waste prevention. This one is the right portion meal. This one embrulha, that means if you don't eat all, wrap it. So we give free of charge these small boxes to take leftovers home. And you can go to, you can surf in our website and you will find all this information, including how to participate in free workshops, online training, guides, food tips. So you can have all this information just in a click. Here, our bio-waste treatment at source, some projects and results. So how to do recycling at sources? Well, doing home or community composting. So what we can see here, the goal is promoting local treatment for bio-waste and creates a network of organic urban gardens, either promoting and preserving biodiversity and sustainable practices. At the same time, we promote healthy and balanced food. So now we have almost 2,000 plots free of charge available to citizens and families to produce their products. So about bio-waste separate collection and our composting site. Here you can see we have several options to participate in terms of garden waste. So we have door-to-door schemes, the bring banks, that means roadside containers like this, with a sticker where you can see information about regarding the rules. We have 19 civic amenity sites where we can drop off the garden waste or by request. Now we are giving, this is a very interesting project, so we deliver free of charge these reusable bags with a identification with a chip to identify the producer, and like that most now a certain number of citizens from the 8 municipalities can have this door-to-door collection scheme for garden waste. The amount that we can recover up to 28 kilograms of garden waste per person and year. Now this is the same, but for our food waste. So since 2005, we started with the food waste separate collection from HoReCa sector. And because we produce, and here you can see the amount of food that is wasted per year. So this is the amount per establishment that we collect and transform in compost. But this means more than 10 tons per establishment a year of food waste that we collect separately and we send to our composting site. But we need to reduce this amount by changing practices in terms of preparation of meals, how we do our when we purchase everything. So same for the separate collection from households. Last year we collect around 67 kilograms per person a year of food waste. Again, we have a huge margin, so a large margin to change these behaviors and to improve the bio waste management in terms of waste hierarchy. That means prevention, prevention, prevention. And then we need to think in recycling, but first prevention. Here, so some results. So here is composting at source. Here is our composting site, so centralized or industrial composting. So just to have an idea, per compost bin, we can recover up to 400 kilograms of food waste, of bio waste per year. We also measure the greenhouse gas emissions avoided. So more than 1,000 tons per year of avoided CO2 emissions. And here you can see the impressive figures that we have with our composting plant, and namely with these separate collection schemes, that from 15 years of activity at our composting plant, we recover more than 300,000 tons of food waste, same of green waste. We produced more than 130 tons of compost. Most of this compost is suitable for organic farmer. That means with a very low level of heavy metals, and we contribute to avoided up to 50,000 tons of CO2 emissions. So very impressive numbers. So just to conclude, here again, our compost called NutriMais, so an organic soil improver. It's an important carbon sink, and I would like to highlight the price that we can have on the market, above 50 euros per ton. So it's important also to have this price, because we can cover obviously the costs related with the collection and treatment. Challenge for the future: a new ID plant, a new anaerobic digestion plant with a capacity with 60,000 tons to treat mainly food waste. And as I said, we want to produce biogas, converted this in biomethane and injected in the grid. In terms of digestate, we'd like to, and we are now working with, and it's important to improve this relationship with academia, because we need to increase this digestate in terms of recovery and value for agriculture. At the same time, we are now with a pilot project. This is preparing growing media using our compost. We will expect to see the results very soon. So innovation always is a first step to reach this circular economy transition. And just to conclude, so what we need to do for the future: reduce the total and per capita amount of municipal solid waste, reduce food waste, strongly increase reuse and recycling, increase home and community composting, increase bio-waste separate collection, and obviously we need to engage and involve the citizens, and for that we need to have new incentives and the pay-as-you-through system also. So here you can go to, again, to our website, and you will find all this information is available to all. So just click in this link and you will see. And to finish, so lessons learned, we need to be committed. We need to think out of the box. We need to be simple. We need to be innovative, and very, very important, we need to improve these community beliefs and attitude to waste prevention and reuse. So it was our experience. I hope it was important for you at least to have this idea in terms of the importance of the bio-waste that is very, very close in our homes, but sometimes we forget. So if you have some questions, please, and thank you again. Thank you.
Thank you very much. Very interesting talk. Now I will share the questions that we have in Slido. So the first question: what are the main adaptations Portugal needs to perform to its infrastructures in order to implement economically sustainable circular economy? What do you think?
Yes, a very good question and update question. So one of the, is not a problem, it was a decision in the past. So our option was mainly to construct and to operate mechanical biological treatment facilities, the MBT plants. Lipor has had another option, and for instance, the Valorsul, the Lisbon system also, so only separate collection. And but for these MBT plants, the option was, okay, we collect mixed waste, we send to these MBT plants, and we try to produce compost. What we see, or what we've been seeing is, yes, the idea was good, but because we have not only bio-waste in our bin, we have plastics, we have papers, we have metals, we have glass. The problem is the amount of compost that we can produce from this mixed waste is really, really, really low. So what we need to do, the main adaptation, is to convert all these big facilities, these MBT plants that are now operated with mixed waste, we need to change to separate collection. So we need to adapt these infrastructures. At the same time, and our national strategic plan shows that very well, Portugal has a problem. So we have this, we are quite asymmetric. That means we have the coastal part with a huge pressure from citizens, and then we have the interior where citizens are older, small village, and we need to think in terms of how to act, not only in the coastal area, in the urban areas, but how to act in the interior. So we need to have this, we need to have more capacity with anaerobic digestion plant, with composting plants. We need to think in terms of green garden waste and try to decentralize the composting sites for garden waste in order to recover all this potential that we have in our bins. Because just to reinforce, we produce around, that means around 40% of our waste is bio waste. So there is a huge amount of resources that we need to recover, but for that we need to adapt our big infrastructures. Yes.
Thank you very much for your answer. Unfortunately, we don't have any time to, we have to proceed. Thank you very much for coming and for your nice talk.
Thank you.
So, our next speaker will be Héctor Ruiz. Héctor is a full professor at University of Coahuila, Mexico. He is also the founder of the Biorefinery Group and leader of the biomass pretreatment stage in the cluster of bioalcohols in the Mexican Center for Innovation in Bioenergy. Héctor has a large experience in lignocellulosic biomass conversion and valorization. Yes, Héctor, please, you can start.
Yeah, thank you. Thank you so much, Sarah, for the introduction. Okay. Well, can you see my presentation? It's okay?
Yes, it's
okay. Well, thank you so much again for the introduction, Sarah, and thank you for the invitation to Ligia, Sarah, Joanna, and all the teams for this great workshop. Well, I wanted to talk about of the processing of biomass in terms of the biorefinery, and in this case, in terms of the bioeconomy, how these concepts are integration for the production of high value compounds. Well, before to start my talk, I want to talk about my city. Saltillo is the capital of the state of Coahuila in the north of Mexico. And we have a good relationship with Portugal in the case of, wait a minute, the pointer. Yeah, okay. In terms of the founder people from Portugal here in Saltillo and the Ocaso Saltillo. So we have a good relationship with Portugal. Well, Saltillo is in a big city. It's an industrial city because we have a good and spectacular companies in terms of the production cars companies, mainly from USA. Well, in this case, our research group here in Saltillo, in the Autonomous University of Coahuila, is in a group in terms of valorization and processing of biomass in terms of the biorefinery concept. So in this case, our principal process is the hydrothermal pretreatment for the fractionation and for start to produce these high value compounds from different feedstocks. In this case, we are working with lignocellulosic materials and aquatic biomass. In this case, macro and microalgae for the high value compounds and different kinds of biofuels. In this case, bioethanol. So in this case, also our research group at this moment is part of the Mexican Innovation Center for Bioenergy in Mexico. This is a great project in terms of trying to introduce the biofuels in Mexico, mainly biogas, bioethanol, biodiesel, and all other solid biofuels. So it is important for us here in Mexico because 3 years ago, Mexico integrate the International Energy Agency. So this is a very situation to take in account because we have the opportunity in terms of development technology and developing these introductions biofuels here in Mexico. Yeah. Well, in terms of the bioeconomy or circular bioeconomy, we have in this part now, in these regions, in terms of the linear economy, circular economy, but we are here in circular bioeconomy with integration of the green economy. But this is important, see that we have the biorefinery concept because it's the mainly stage for trying to processing the biomass, and they integrate with the concept of the circular bioeconomy for high value compounds. So the relationship between this concept are very interesting. And now in this talk, I want to introduce this part of the biorefinery for the bioprocessing of biomass. Well, what is the context? In this case of Mexico, 3 years ago, was developing this bioethanol technology roadmap in terms of trying to see what are the most promising raw materials in the case for the introduction of biorefineries here in Mexico. We have the first generation, in this case, with sugarcane bagasse, the sorghum straw, there's a sorghum grain, for example, and we have the second generation that maybe in more 5 or 7 years, when it's possible to introduce here in Mexico with different agricultural residues, for example, sugarcane bagasse, other kinds of agricultural residues, for example, wheat straw, corn stover, or we have a promising raw material, in this case, agave bagasse. Well, agave bagasse is an agricultural residues here in Mexico. In this region, in Jalisco, is an state in Mexico, we produce tequila. Tequila is an alcohol beverage, very typical, you know, in all other parts of the world. And with this processing of this kind of cactus, agave, we can produce about more or less 40% of these agricultural residues. So in general terms, this can consider promising raw materials for using and introduce in the second generation of biorefineries. With this concept of the circular bioeconomy, we can integrate for the high value compounds and biofuels. Well, in terms of the integrate biorefineries using the biochemical platforms, we know very well this kind of process. For example, for produce bioethanol or for produce other high value compounds using these biochemical platforms from lignocellulosic materials. But in all this process, we can use the stage of the pretreatment. This is a very important stage in terms of the fractionations of different biomasses. So the question is, what is the bottleneck for developing of this process for second generation if we know very well the process for developing biofuels or different high value compounds from lignocellulosic materials? So in this case, I show to you our general process for the integrated biorefineries in terms, for example, in agave bagasse, or we can use other lignocellulosic biomasses. The principle for us is try to fractionate these biomasses using the hydrothermal pretreatment. I explain more and specific for this process, and we can obtain these high value compounds, for example, from hemicellulosic fraction. In other talks for this workshop, I checked that mention in terms of the production from oligomers from hemicellulose are very important in terms of high value compounds, and we can produce directly for this process. And we can obtain other kind of solid phases for try to produce biofuels, different kinds of biofuels. In our research group, we are more expertise in terms of the ethanol productions, and we can produce the lignin for applied for different areas. For example, in the food research areas or in biomaterials, are very important at this moment, the use of the lignin for the different properties and characteristics that has this biopolymer now. So in terms of the hydrothermal processes under subcritical conditions, we want to find the equilibrium between the operational conditions for try not degrade these biomasses. We can obtain a good yields in terms of the sugars for the solid phase. In this case, in solid phase, we can produce the oligomers from hemicellulose fraction, and we want to produce a good solid phase in terms of the lignin and in terms of the cellulose. This cellulose in subsequent process, we can convert in biofuels or other kind of compounds. So in this case, we are working here in this region in terms of the extraction of the compounds and in terms of the fractionations. But if we pass for other kind of conditions of the temperatures or the pressures, we can pass for other kind of thermochemical process, for example, pyrolysis or gasification. No? So in this diagram, I want to show the integral bioprocess for biorefinery concept using lignocellulosic materials applying the hydrothermal pretreatment. In this case, for example, when we apply these lignocellulosic materials, after of this process, we can obtain these phases, in this case, liquid phases, and we can produce these high value compounds, oligomers, of course, for applied in the hemicellulose fraction. And in terms of the solid phase, we can produce biofuels, we can produce other high value compounds applied in the biocomposites or biomaterials area, and we can delignify. In this case, the lignin, we can apply other kind of process, subsequential process, for example, organosolve, for try to recover this lignin and apply it, because the lignin at this moment, we can apply it in terms of the food research area, because the compounds, mainly compounds from these biopolymers are phenolic compounds. So we have different kind of characteristics for applied in this area now. Well, in terms of the processing, or what is the reason that we are working for more than 10 years for hydrothermal pretreatment is for this reason. No? We have a different advantage for applied this process. For example, it's a considered an environmental friendly process because we can use water in terms of liquid or in terms of the steam, whatever, for applied on lignocellulosic materials. We have a specific process for the polymerization and solubilization of hemicellulose fraction during this process. In this process, when we applied the heating, for example, microwave, steam, we have an equilibrium in the reactor. And with this equilibrium, we can increase the pressure and we can modify, we can change the structural composition and characterizations in terms of the biopolymers, and we can obtain different fractions. So we have a different advantage in these terms. The other important, for example, in terms of the biofuels production, and in specific for ethanol, we have the relocalization of the lignin on the surface after of the pretreatment. So with this relocalization of the lignin, we can improve the enzymatic hydrolysis for glucose production in this case, and subsequently biofuels productions. No? So what are the most important variables that we can consider or take in account for example, the scale of process in the lab or in the pilot plant? We can use this parameter for use in terms of the strategies for the scale up, the severity factor. This severity factor is not considered the operational conditions. In these terms, the temperature, in this case, the time during the process. So, and we can use different kind of conditions depending on what kind of products we have obtained during this process. We can use the non-isothermal heating process, for example, for production of the oligomers, or we can use the isothermal regimen in terms of produce the high concentration of cellulose after of the process for try to produce ethanol, for example. And the most important variables, temperatures, times, and pressure. But if we can apply high temperatures and high pressure, we can degrade the lignocellulosic materials. 10 years ago, with this degradation, we can produce, for example, furans. These furans are toxic compounds for the fermentations, but at this moment, in terms of the biorefinery concepts, are very high value compounds. So depending of what kind of compounds, we can apply the different operational conditions in hydrothermal pretreatment. In this case, in our research group, we are developing different kind of strategies for the scale up. We can develop different kind of technologies in terms of applied for the lab scale and the pilot plant. And in this case, we have in our lab different small reactors. It's very nice in terms of the optimization condition, because we can apply different experimental designs on this kind of biomasses. And when we have these optimal conditions, we are introduced, or we can find different strategies for the scale up now. This is our pilot plant in our university. It's a tubular hydrothermal reactor for processing in mulch bat operation. In this case, the heating of this kind of reactor is through this boiler for produce steam, and we can pressure the reactor inside, and we can increase the temperature, and of course we can fractionate the biomass and produce the different compounds. In this case, for example, we apply the sugarcane bagasse. In this case, we feed the raw material here in the top of the reactor, and we can obtain these oligomers from hemicellulose, and we can obtain this fraction, solid fraction, in terms of the cellulose and in terms of the lignin. So in this case, we have the automatic control for this reactor. For us, it's very important to measure the pressure and the temperature with different kind of sensors in the reactor, because if we have different changes during the process, we can modify, we can degrade the lignocellulosic material. So for this reason, it's very important to try to control the temperature. We can see in the control system, we can a very nice control in terms of the temperature during the process. Now, well, in terms of the applications, for example, for agave bagasse, in this case, when we want to produce the ethanol, we need to take in account different kind of stages for this process. What happened in terms of the cost? In terms of the cost, the most percentage contribution for this kind of process is the feedstock, in this case, the raw material. We need a high volumes for the conversions. We have the enzymes for the conversion in fermentable sugars, and the processing in terms of the pretreatment are the most contributions in terms of the cost for the lignocellulosic materials for ethanol production. So in this case, it's very important to try to use the high solid loading during this process for try to obtain high concentration of these fermentable sugars for convert in other high value compounds or biofuels. So in this case, we applied the agave bagasse in terms of the optimization during the hydrothermal process, apply these different kinds of temperatures and different kinds of pressures with different kind of resident time. And well, we can obtain the optimized condition in terms of the cellulose, because, for example, in this case, we have here the cellulose, and we can see how degrade or depolymerizate the cellulose during the hydrothermal pretreatment. But we can obtain this optimized condition only after 220 degrees Celsius in terms of the polymerization of cellulose. So in this case, we can find a different concentration of degradation compounds, in this case, furans, and we can see how the monomers and oligomers decrease. It's an atypical behavior for the hydrothermal pretreatment, in this case for agave bagasse. So in this case, we can see the different changes in terms of the structural modification on surface after of the hydrothermal pretreatments, apply these optimized conditions. And after of this solid phase, that is in this case, we apply the enzymatic hydrolysis. As I mentioned, it's very important to try to apply the high solid loading for produce high concentration of glucose. In this case, we designed different kind of reactors for high solid loading and produce more than 195 grams per liter of glucose. Is a good concentration in terms of produce, for example, ethanol, because we can produce more or less 100 grams per liter of ethanol if we have 100 yield of conversion. So it's important to try to develop in this kind of process, developing this kind of designs for enzymatic hydrolysis and different kind of bioreactors. So we apply different kind of modelings in terms of reduce the use of the enzymes, because as I mentioned, the cost of the enzymes are very expensive in this case, and we can apply different kind of model for reduce and try to optimize the enzymatic loading during the enzymatic hydrolysis. And we can see that we can check the inhibition of these enzymatic of these enzymes during the enzymatic hydrolysis on the surface for this process. And we can see that during the first 12 hours, more or less, we can obtain the high concentration of these sugars, in this case, glucose. No? We have other kinds of papers of works in terms of the biorefinery concept applied agave gas in terms of the Mexican context. It's very important for us. And for the conclusions, in terms of the agave gas, here in Mexico is a promising raw material for applied for introduced development, the biorefinery concept. But for example, in Portugal, there are many kinds of feedstocks. In the north of Portugal, we have eucalyptus. In the center, in Alentejo, have a wheat straw. So depending on the region, it's very important to try developing this kind of biorefineries. And in general terms, the hydrothermal processing is considered a good process in terms of the scale up, in terms of the application. At this moment, in many, many industries around of the world, we have applied this process for the biomass fractionations. So this is our research group. This is my email. If you want to contact, please, about of this process or whatever in terms of our research line. And I invite you to send your last work to the Bioenergy Research Journal. We have accepted different kind of topics in terms of the bioprocessings, in terms of the biomass for biofuels, or in terms of high value compounds for different chemicals. And the last, for this special issue in the Biotechnology, in terms of the hydrothermal pretreatment, I invite you to send your work. So thank you so much for your for your attention, and muito obrigado.
Thank you very much, Hector. Very nice talk. I will share now the Slido.
Okay.
Here. I think we have time for one question.
Okay.
So the most voted question was, do you think that the biorefinery process will eventually economically outcompete oil-based fuels in terms of absolute price, despite taxes and regulation, and how? A very complex question.
Yeah. Well, depending of the situation, in terms of what country needs to apply this kind of biofuels now. For example, in Europe, it's typical that use, for example, biodiesel. Here in this part of the world, here in Mexico, we don't apply it much in terms of the biofuels for the auto transport sector. So depending of the situation, depending of the process, but the most important is to try to develop it and apply this concept of the biorefinery and the bioeconomy, depending of the biomass that we have in the regions and what is the politics or policies in this case now.
Okay. Thank you very much, Hector. I think we have to finish here, unfortunately.
Bye-bye.
Now we will have our last keynote, and this keynote will be presented by Paula Pinto. Paula is a technological R&D coordinator at Institute Raiz, which is part of the Navigator company, Portugal. In the last years, her work has been focused on lignocellulosic materials, including extractives, lignin, and cellulosic compounds. In this case, we have a video to share with you, so we can start. I think we don't have the sound. Please, Joanna, can you share again? We are trying to share the video. Just a few seconds.
To the organizers for this. Hello, good afternoon to all. It's a great pleasure to present you the Impact Project, the research program of 4 years at the Navigator company. But first, I would like to thank to the organizers for this invitation and to congratulate you all for this high standard event. I will start with a brief presentation of the Navigator company. We are the European leader and one of the world's largest manufacturers of bleached eucalyptus kraft pulp. This pulp is sold to final applications such as decor paper, packaging and filter papers, demanding high quality pulp. Part of this pulp produced in our mills is as well integrated in paper mills to run office paper and also tissue. We are the European leaders in uncoated wood-free printing and writing paper and the 60th largest producer in the world. Here you can find some figures, but it's also important to highlight, because sometimes the community is not aware about this, that our company is the leading producer of green power from biomass and wood byproducts, accounting for more than 50% of total national power generation from renewable sources. In Setúbal Industrial Complex, we have also the largest photovoltaic plant installed in any industrial environment in Portugal. Navigator is the world best-selling premium paper with consumers in more than 130 countries. Other figures: the company manages woodlands, about 110,000 hectares, and we are more than 3,000 employees. Moreover, with its activity, the company provides indirect jobs and have more than 7,000 suppliers in Portugal. These figures demonstrates the importance of the company for the Portuguese economy. The Navigator company operates 4 industrial units, one at Aveiro, producing pulp to the market and tissue paper, at Figueira da Foz and Setúbal, producing pulp, most of them to be integrated directly in paper production, and one at Vila Velha do Rodão, producing tissue paper. So the Navigator company's activity covers all the value chain: forestry, energy, pulp, paper, distribution, and also R&D. R&D is developed in Forest and Paper Research Institute. Raiz is a private non-profit research institution recognized as an interface center committed to support competitiveness of forest-based bioeconomy. Through research, services, technological transfer and training, most of our activity is based on collaborative projects. Raiz is owned by the Navigator company with participation of University of Aveiro, University of Coimbra, University of Lisbon. Regarding activities areas, Raiz acts on forestry and technological R&D and consulting. Technological scouting as well, and management of intellectual property. in demonstration, scale up, and also supporting new businesses. We have also facilities for laboratory services for the company and the board. And finally, training. In technological R&D, we cover all pulp and paper industrial fields. Here you can see some of our capabilities and materials. This is bleached kraft pulp, and this brown material is lignin isolated from kraft black liquor. So these are the two main components of the wood. And I would like to highlight in next slides the biorefinery topic. Okay, biorefinery. A forest-based biorefinery is a multi-platform unit converting lignocellulosic biomass into several intermediates and final products. What is the relationship between biorefineries and pulp and paper industry? In fact, pulp industry has been recognized as the leading industrial sector in biorefinery. Since long time ago, due to the raw materials, due to the generated bio-based products, as well as due to the integrated production of pulp and energy, mostly provided by burning black liquor and biomass. In a more extended sense, pulp and paper industry is one example of circular economy implementation, considering, for example, the chemicals recovery cycle for pulping. So what is missing? What is missing is the diversification of bio-based products. This sector has the utilities, logistics, and operational conditions to be at the front head of the forest-based bioeconomy and take the chance to diversify the range of the products. These are some examples of companies, pulp companies, that are moving for new concepts and increasing the range of bioproducts. The roadmap for a low-carbon economy refers the ambitions to add 50% more value in 2050 compared to 2010 to European forest fiber and paper industry. High-value products and tailor-made solutions are the targets. Packaging, hygienic and specialty papers will generate about 5 billion euros. About 3.5 will come from the development and growth of new bio-based products, food additives, biocomposites, advanced fuels, for example. These are some of the emerging bio-based products. So intensive innovation and research for new products, processes, and proof of concepts are highly required. And this is one of the scopes of RAIZ activity. RAIZ Foundation dates from '96. The objective of the first project was to understand the species behavior toward kraft pulping, bleaching, and paper making, aiming to understand why the eucalyptus would perform better than other species. Interesting to note that this fundamental research has led at that time to novel findings and then open up the perspective for new products and then for new research in line with emerging biorefinery activities in Europe at the end of '90s. The following R&D work at RAIZ was then focused on new possibilities coming from eucalyptus and coming from forest biomass. The range of topics was extended in the 2010 and the work front has included new applications for fibers, for inorganic residues and evaluation of biomass as source of sugars and lignin testing also several final applications. More recently, since 2016, RAIZ has been expanding its activity in collaborative projects, including demo activities and also European projects. In the meanwhile, Navigator has been supporting PhD programs in several fields. And finally, Impact Project, starting in the 2018, which represents the umbrella research program at the Navigator company. I will come back to this project in some minutes since it is the main topic of this presentation. So, before Impactus, what topics have been developed at RAIZ in partnership with universities and other research institutes? I'm presenting here in the next slides some highlights about the past activities, which were also the baseline for the current research program. Okay, one example is the extraction, fractionation and purification of extractives from forest biomass, focused on tritropanic acids and polyphenols. In the tritropanic acids topic, an environmental friendly process was developed, and the purification has resulted in a joint patent between University Aveiro and RAIZ. These fractions of obtained by extraction of bark were pointed out as promising ingredients in cosmetic and nutraceutical products. RAIZ and the Navigator company, in collaboration with several institutions, we have been working in the evaluation of the construction processes to obtain polysaccharides and lignin from forest biomass. Polysaccharides fractions were tested in conversion to simple sugars, evaluating the process, for example, glucose yield and final concentration. Why? Because this could be the platform chemical for ethanol, lactic acid, which in turn are the chemicals, for example, for green solvents and polymers. Also, these sugars were tested as feedstock in microbial production of cellulose and also polyhydroxybutyrate. Also, lignins were isolated, characterized and tested in different applications. To produce phenolics and oligomers for polyurethane foams. Finally, bleached eucalyptus kraft pulps were evaluated as model for incorporation in polymeric systems, evaluating the mechanical and environmental performance. Also, in another project, pulp was evaluated in an integrated process for production of xylans and microfibrillated cellulose. All of this knowledge was possible thanks to our partner, some of them being with us since 1996. Impact Project is a co-promotion project started in 2018 and represents the R&D roadmap at the Navigator company for 4 years. It involves 11 institutions beside the Navigator company with about 180 human resources, 15 research grants, and also 2 invited chairs. In total, we have 45 sub-projects running. And it has been a big challenge to manage all the knowledge that has been generated. The goal of the project, in fact, is to reinforce our competitiveness in core business and simultaneously to get or to increase the knowledge on bio-based products and materials from forest biomass. So what was the motivation? The core business of the Navigator company is centered in market pulp, office paper and tissue. Considering the high market competitiveness of these products, we need the product differentiation, diversification, innovation, and also a contribution to the cost reduction and the environmental performance of our products. For this is required to develop new knowledge and also to consolidate the competencies. Biorefinery and new bio-based products are emergent areas, which could lead to new business opportunities. So from pulp to take cellulose and hemicelluloses and convert them to new products. From side streams, forestry biomass coming from plantations, branch, leaves, bark. From black liquor, the stream coming from a pulping process with high lignin content. This is what we intend to reach with Impactos project, representing the research roadmap, finding new value by creating adjacencies to the core business. So this project represents a platform, industry, university, taking as baseline the previous knowledge and competencies from the past, from Raiz, from the Navigator company, and from the partners. So what are the expected outcomes and new challenges in each of these fields to work in Impactos? We are preparing pulps with new properties. We are developing new technologies, for example, for bleaching. We have also activity in the upgrading residues in the perspective of circular economy, and we are also developing tools to support the process decision. In uncoated wood-free paper, we want to improve the traditional properties of the paper, such as mechanical and surface resistances, the printability as well, and we are also developing new functional papers. In the tissue paper, we want to improve the key properties such as smoothness and absorption of papers, and also to develop novel functional papers related with this market. In biorefinery, our focus are in products from lignocellulosic biomass, processes and technologies adjusted to our specific biomass. So in the project, we are covering all the value chain, starting with residual biomass, such as bark, branches, and foliage. In this activity, we will develop green technologies to maximize the extract's value, and we are also performing a deep evaluation of biological activity. We are considering a process for the whole valorization of this biomass. So this is another example of the importance of a multidisciplinary team in this project. In another sub-project within the Impactos, we are covering different delignification processes to maximize the yield of sugars produced. We are also evaluating the integration of extraction and the construction, because this pre-treatment could have a positive impact in the construction and subsequent processes. Then we use these sugars as feedstock to several products. Ethanol and bacterial cellulose are two of these products. Also included in the Impactos are the thermochemical processes, pyrolysis, gasification, and liquefaction for biofuels and for high added value products. Finally, to complete the cascade approach for an integral valorization of biomass and meal streams as well, we have 3 projects dealing with lignin, concerning isolation and concerning heat application to new materials. The last topic is related with new products from eucalyptus fiber. We have been working with bleached eucalyptus kraft pulp and also with pulps resulting from the lignification or the construction processes of biomass. We are processing it and modifying the cellulose to impart some specific properties, aiming to apply it in the composites, producing microfibrillated cellulose, producing xylane derivatives with high value in the market, and also new additives to be incorporated in paper production. interesting to stress is the interaction between the different projects from different areas and integrating and finding complementarities. One example is the project dealing with production of xylo-oligosaccharides. The pulp with a content in xylans of about 20% is processed by an enzymatic treatment, and then the liquid streams follows to separation and purification process to obtain the final product. The prebiotic activity is now under evaluation. The solid stream is a pulp with lower content in xylans, but not degraded thanks to the high selectivity of the process. So this pulp could be reintegrated in the pulp pipeline, imparting some interesting properties to the paper, or at least instantly to not affect the properties. Or it can be a good raw material for other cellulosic products that are now under evaluation. Our commitment with the funding agency are the creation of spin-offs, to submit patents, to get proof of concepts and prototypes, and also to produce publication and to participate, of course, in conferences. We are also training young researchers that could represent a good pool for the Portuguese modern bioeconomy, being this part of the REIS and the university's mission. So, Impacts project is generating the support for the new bio-based businesses, and are also promoting innovation and competitiveness in our country. But more than this, the project is a good example of industry-driven research, and we are making partnerships, of course, for the future. Thank you very much for your attention.
So now we have time for questions. Hi, Paula. I will share the Slido. Okay. So the most voted question was, considering the environmental issues caused by eucalyptus overplanting in Portugal, how do you weigh the pros and cons of developing eucalyptus-based products?
Hi again, and again, thank you very much for this invitation. And this is a very complex question, and it is very difficult to answer. But what can I say about that? I would like to explain. In fact, eucalyptus, we have several advantages coming from eucalyptus. From the point of view of process, when we compare eucalyptus with other woods, and this work was made in REIS in 96 until 2000. We finally realized that with eucalyptus we get less consumption of chemicals during pulp and even during bleaching. And we get a very high quality product at final. So our early research at REIS since 96 was precisely related with that. So eucalyptus is still, Eucalyptus globus, this species, is still our raw material for high quality pulp and with a very good environmental performance. So another aspect that I would like to give you is that our wood that is our raw material for the 3 mills that we have that produce pulp comes from certified forests, certified plantations. So we will continue, of course, to use eucalyptus coming from these plantations, and we have at forest the biomass, the branches, the leaves, the bark, and we have been in our research, we find out very good opportunities to valorize them, not only for energy that we already use. For example, bark is used for our biomass boilers at mills, but to recover some important compounds just before burning it. So in any way, we will have this forest biomass, and it's important, of course, to have a cascade valorization of this to get as much as value that we can for new products.
Okay, Paula, thank you. I think we have more 3 minutes, so we can go to another question. Concerning the full valorization of biomass, is the goal of Navigator approaching new markets of activity, namely the prebiotic markets?
Well, what can I say about that? Is the mission of Raiz to find out new opportunities for the company, for sure. So we are working in the finding new products that could lead to some diversification of the final products of the company, of course. But first we need to prove it, and we need to prove the sustainability from the economical point of view, of course. And we are working on, for example, some pilots to get the upscaling of some of the processes, that ones that are more viable. For example, this example, prebiotic, we are in the stage that is very important. We are just waiting for the analysis about the prebiotic activity of xylem, of a derivative from xylem. So we can decide after all if this is a promising market or not for a derivative from our pulp. So our pulp is produced, and we have about 20% of xylem that, and we have proven as well that we can get xylem oligosaccharides from this pulp. If we can scale up the process and make economic evaluation as well as pre-engineering studies, of course, this will be presented to our board, and this could be a new business. But the mission of Raiz is to go step by step and present these opportunities for the company.
Okay, thank you. Thank you very much, Paula, and all the speakers of the keynote sessions. Now we have a short break, and we will be back in 15 minutes. See you. Welcome back. Now we will move to the session dedicated to the key projects. And in this section, 4 interesting projects based on biotechnological applications in the environment will be presented, and we will start with Andrea Santos. Andrea is a researcher at Center of Biological Engineering, University of Minho, Portugal. In the last years, she has been focused on the biotechnological application of microorganisms in fermentative and agroindustrial processes, and also on the application of metagenomic approaches to explore novel biocatalysts. Please, Andrea. Andrea, you can start. Okay. Andrea?
Sim.
Okay, now we can hear you.
Só um minuto, estou tentar compartilhar o o vídeo. Não estou conseguindo compartilhar o vídeo.
Andrea, we are...
I cannot share the video. Just a minute, please. Okay. Well, good afternoon. Can you see my presentation?
Yes, yes.
So, good afternoon. Thank you for the presentation. And I would like to present the Enzymes project that is entitled Metagenomic Approach to Unravel the Potential of Lignocellulosic Residues Toward the Discovery of Novel Enzymes. Starting with a brief introduction about the lignocellulosic residues, it's well known that one of the greatest challenges in the world has been to replace the fossil resource with the renewable one. In this sense, the lignocellulosic residue has a great potential to be used as a low-cost and biorenewable resource, as they are largely available around the world. However, an efficient hydrolysis is necessary, and this requires intensive pre-treatment that can be carried out through physical, chemical, and biological treatments. In this scenario, the enzymes, including cellulases, hemicellulases, and ligninases, plays an important role because they will be responsible for the breakdown of the lignocellulosic material into monomers that can be converted into different high-value added products, as was well explained by Dr. Hector Ruiz. In addition, the lignocellulosic residues can be an important source of enzymes also. The main classes of enzymes involved in the degradation of lignocellulosic material are cellulases, hemicellulases, and ligninases, in addition to other accessory enzymes. The enzymes are biocatalysts that have a great application in different industrial sectors, and due to the high demand of novel enzymes with higher efficiency and stability, novel approaches have been tested, and in this sense, metagenomics has been widely used. Here we can see a general schema of a metagenomic approach that consists of several steps, including the genomic DNA extraction, the construction of the metagenomic library that requires the selection of a vector and a host, and the screening of the target substance that can be through a functional screening or a sequential screening. In this sense, the main goal of the Lignozymes project is to use a functional sequential metagenomic approach to find novel and promising enzymes responsible for the decomposition of lignocellulosic residues from industrial composting samples. The task 1 corresponded to the collection of the compost sample that were obtained from the companies Terra Fértil, composed of agroforest residues and municipal sludge, Lipor, that were composed of basically of food waste and agroforest residues, and Gintegral, composed by agroforest residues, municipal sludge, wood, and cork chips. After the collection of the samples, it was performed the physical and the chemical characterization of the samples, and it was possible to identify a great variation between them. The task 2 corresponds to the genomic DNA extraction, and it was the main bottleneck in this project because it was necessary to standardize an ideal protocol for extracting the metagenomic DNA containing high molecular weight from compost samples containing high humic acid content. We have tried many different protocols, including commercial kits and traditional protocols, as we can see in this table. Finally, we will obtain the very good results with the protocol 10, where sodium phosphate was added to maintain the integrity of the DNA. CTAB was added as the humic acid removal agent, and calcium chloride was added to prevent the oxidation of the humic substance that binds to the DNA. After all of this difficulty and effort, we have optimized this method and submitted a patent process for this protocol. It was a national patent. The task 3 consists in the metagenomic library construction. For the functional screening, it was using the copy construct phosmid kit according with the protocol, and the clones obtained were inoculated in culture medium with a specific substrate to evaluate the enzymatic activity that could be visualized through the development of halo around the positive clones or a color change of the medium. Until now, this functional screening it was performed only for the sample of terra fértil, and we have obtained 563 clones that were tested for the enzymatic activity of cellulase, silanase, lacase, lipase and esterase, and amylase according with the following protocols. Initially, the tests were performed in petri dishes where we inoculated 40 clones. However, we face a problem of diffusion of the enzymatic activity. So we have decided to isolate the clones and isolate the experiment using microplates with 96 wells. Here, it's possible to verify the results in the microplates and observe the positive enzymatic activity of some clones due to the color change of the medium. From a total of 563 clones, it was possible to identify 217 clones that exhibit positive activity for beta-glucosidase, 1 clone exhibit positive activity for xylanase, 10 clones were positive for lacase and peroxidase, 93 clones presented activity for lipase and esterase, and 9 clones were positive for the activity of amylase. For the sequential screening, it was used the NebNext Ultra DNA Library Prep Kit, and the sequence was performed using Illumina HiSeq platform. Following the sequencing of the DNA, several analyses using bioinformatics tools were performed for the identification of the genes present in the samples, according with the following steps that consist of the DNA assembly, the gene prediction, and the gene annotation that aims to identify the taxonomic origin as well as the function of the predicted genes. The results of the taxonomic annotation at the kingdom level indicated the dominance of a bacterial community, representing 88% of the predicted genes. Regarding the taxonomic annotation at the phylum level, the sample was dominated by 3 bacterial phyla, represented by Proteobacteria, Bacteroidetes, and Balneolaeota. Here we can observe the relative abundance regarding the kingdom, Archaea, Eukaryota, and viruses. Concerning the functional annotation, it was performed against 3 databases, KEGG, ENGNOD, and CASI, being CASI the most important for this study. Concerning KEGG database, this database is divided in 2 levels. In the level 1, the majority of the predicted genes were annotated with the metabolism functions. And in the level 2, it's possible to observe that the majority of the predicted genes were related to amino acid metabolism, followed by carbohydrate metabolism, energy metabolism, and metabolism of cofactors and vitamins. The ENGNOG database is divided into 24 functional categories, and the majority of the predicted genes was annotated to the category S, related to unknown functions, followed by amino acid transport and metabolism. CASI database is divided in some levels also. In the first level contains 6 functional classes correspond to glycoside hydrolase, glycosyl transferase, polysaccharide liases, carbohydrate esterase, auxiliary activities, and carbohydrate binding modules. It's possible to observe that the majority of the predicted genes was annotated with the glycoside hydrolases and glycosyl transferases, representing about 4,200 and 3,500 genes respectively. Here we can see the functional annotation in the level 2 that corresponds to the different families within each functional class. For the class glycoside hydrolase, the biggest number of genes were annotated with the families G813, G823, G83, and G843 respectively, and all of those families are involved in the degradation of lignocellulose material. Concerning glycosyl transferase, the majority of the genes were annotated with the family GT2, GT4, and GT51, and however, only the enzymes from the family GT2 are involved in the degradation of the lignocellulose material. For carbohydrate esterase, the biggest number of genes were annotated with the families CA4, CA14, and CA11. However, only the family CA4 is related with the lignocellulose degradation. Concerning the class of auxiliary activities, the majority of the predicted genes were annotated with the family AA1, AA3 being in the family AA1 and AA3 being in the family AA1, the only one that is related with the degradation of the lignocellulose material. As a conclusion, concerning the terra fértil sample, it was possible to identify several clones or genes that presented lignocellulolytic activity, either by the functional or the sequential screening, revealing like this that this composting obtained from the Terra Fértil company it's a promising source of enzymes that can be new enzymes or not. And so it's a potential source of lignocellulosic enzymes as we identified with our results. Concerning the LIPOR and concerning the samples obtained from LIPOR and G-Integral, we will still need to perform the functional screening and analyze the results of the sequential screening. And it's also necessary to perform the taxonomic and the functional comparison of the genes present in the 3 different composting samples, because our objective now also is to compare the different enzymatic activity present in the 3 different composting samples. And the last objective in this concerning these 2 samples is to find new and promising lignocellulosic enzymes. Those enzymes, after we identify and isolate those enzymes, we will perform the identification and the optimization of those enzymes with the objective to propose it for the different industrial sectors later. So here we have the team involved in the Lignozymes project, and I would like to thank you all for your attention, and I will be glad to answer the question that may you have.
Thank you very much, Andrea. Very nice talk. I will now share the slide. Okay. So maybe we have time only for one question, I think, but in your opinion, which is the most suitable metagenomic approach, the functional or sequential?
It's an important question, and the answer will be concerning the objective of the study, because with the functional metagenomic approach, we can directly identify enzymes that can be new enzymes, and we will identify those enzymes, and we need to sequence the DNA. And with the sequential, we will identify basically the genes. And now the biggest difficulty is to analyze the results, because we have all the results from that are already in the database of the enzymes, which gene corresponded to which enzyme. And now the biggest challenge that we have now is to identify the genes related to enzymes that were not identified, because those ones can correspond to new enzymes that can be a promising enzymes for the different industrial sectors.
Okay.
So in my opinion, we should always associate both metagenomic approach, because one will complement the other.
Okay, thank you very much for your answer. We have to move to our next speaker. That will be Diana Souza. And Diana is a professor at Wageningen University and Research, the Netherlands. Her research work has been mainly focused on the design and development of synthetic co-cultures for the conversion of syngas. Please, Diana, you can start.
Thank you very much, Sarah. Hello, everyone. My name is Diana Souza, and I work at the Laboratory of Microbiology from Wageningen University. And one of the subjects in my group is the conversion of syngas to chemicals. And with syngas, I mean here the gases that are composed of carbon monoxide and sometimes also CO2 and hydrogen. So today I will try telling a little bit of the research and the approach that we take in my group for the conversion of syngas. So here you have a representation. So we have here, well, a common waste stream where you can find lots of carbon monoxide is the one that is generated in heavy industry. So there's already available sources of carbon monoxide, but we are also interested, of course, in the more renewable ways to produce carbon monoxide. And with those ways, I mean the utilization of, for example, lignocellulosic materials and wastes that can be gasified. And during this gasification, we generate syngas. And syngas has a rather stable composition, so mainly it's composed of carbon monoxide, carbon dioxide, and hydrogen. And we've seen already several approaches to convert the lignocellulosic biomass via fermentation, where we have several routes. So what we do here is actually having a simplified route or an alternative route from this. And from syngas, we can use microbes to produce many different compounds. And this can be done using pure cultures of microorganisms, and a group of microbes that is quite well studied for the conversion of syngas are acetogens. And acetogens, they are able to convert CO and to mainly produce acetate and ethanol, as you can see here in this picture. And you also have an example of an organism that does this, an acetogen, that is Clostridium autoathenogenum. The problem here is that the range of products that can be produced is rather limited, and you can see there acetate and ethanol as main products. So there's groups working on genetic engineering of these organisms, but that's still quite challenging also because they are not so difficult, so easy to genetically engineer. And also their energy metabolism is quite limited. So what we do is to take also another different approach here, and we try to work with synthetic co-cultures. And with synthetic co-cultures, I mean putting organisms together in a way that you can create a food chain to have a substrate that can be difficult to convert by one organism alone to a product of interest. So as a proof of concept, we started with a very simple co-culture in which we use this organism that I had in my previous slide, one that can use carbon monoxide, Clostridium autoethanogenium, and we couple these in a same vessel to a chain elongator. That's Clostridium cloiveri that you also have here. And what we wanted to produce with these was syngases being converted to acetate and ethanol, and then acetate and ethanol are taken by the chain elongator and you produce medium chain carboxylic acids. So basically you produce a product that could not be produced by this acetogen alone. So this was the theory, and we tried to put these two organisms together, and what we got in the end was indeed when we set the two organisms together, we start having butyrate and caproate production, so these even chain, medium chain carboxylic acids. Later on, we also showed that having them together is actually beneficial for ethanol production. So the presence of Clostridium cloiveri creates a thermodynamic pool in Clostridium autoethanogenium that makes it produce more ethanol. And ethanol it's very important because it's the electron donor used by Clostridium cloiveri to produce butyrate and caproate. So this shows that having them together, instead of having, for example, two sequential processes with each of the organisms doing the independent conversion, is better. So this co-cultivation is working well for the organisms and to improve the production of the medium chain carboxylic acids. So we were enthusiastic with these results. So we thought, can we produce something else besides these even chain fatty acids? And a logical follow-up for this work was then to think of the production of odd chain medium chain carboxylic acids like valorate, that is a C5 carbon, 5 carbon molecule, or caprylate, that is a 7 carbon molecule. And in principle, if we have a Clostridium cloiveri that was already in the other co-culture, if we have it fed with propionate and ethanol, this organism is able to produce these odd chain medium chain carboxylic acids. The problem here is that so far there's no organism that can produce propionate and ethanol from syngas, or we don't know any. But at this point, we also had an enrichment in the lab. So this was an enrichment culture, so not a pure strain. It was a highly enriched culture, but it was being fed with carbon monoxide that you have here in blue. And after a while, we actually saw production of propionate. So we thought maybe this culture has an organism there that can produce propionate from carbon monoxide, and that would be ideal for our co-culture. So we looked into the microbial composition of this enrichment, and we saw that it was highly dominated by an organism that is closely related to Acetobacterium. And Acetobacterium are also acetogens, so they have a very similar metabolism to Clostridium autoethanogenium. The ones that are known, they use carbon monoxide and they produce acetate and sometimes also some ethanol. So we thought, okay, maybe this is a different one and maybe it produces propionates. We managed to isolate this organism. And here you can see, we called it strain JM. Here you can see that indeed this organism can convert several additions of carbon monoxide in blue, but the main product is acetate, so no propionate. It's a very good CO converter. For example, if you compare with the type strain of the same genus, it converts CO very fast, but it cannot produce propionate. Also, when we looked into the genome of this organism, we could not see any genes for propionate production. We did see, of course, the genes for ethanol production. And we also saw then in cultivation that when carbon monoxide is fed at high concentrations enough, then there's some ethanol production. So we went back to the enrichment culture and we looked a little bit better into the composition. So Acetobacterium is not producing propionate, but we also have their Pelobacter species. And Pelobacter species are known for producing propionate from ethanol. So what we then thought was that maybe this strain JM was working together with Pelobacter propionicus to form propionate. And how this works, it would be in the way that carbon monoxide is converted to ethanol and acetate, and then the ethanol is used by Pelobacter to produce propionate. So this was our hypothesis.
So we set up this co-culture, and we set up also a very similar one, also with strange AM, but with another organism that has a similar metabolism to Pelobacter, and that is called Enerotignum neopropionicum. And we set this second one up because we wanted to increase the chances of having a co-culture that was able to produce propionate. We call this phase 1 because we are testing just this part of the system, so not yet including Clostridium cloiveri. And what we saw then was indeed with this second culture that we tried, we could have very good results. So again, you have a graph with carbon monoxide now in green and several feeds of carbon monoxide. And then you start seeing at a certain point, there's the purple that is ethanol, and almost simultaneously you have this orange line that is propionate. So indeed, we could show that strange AM together with an Enerotignum neopropionicum was able to take syngas and to produce propionate. So then we thought, okay, let's now try phase 2, and we still want to do this in parts. So we are not including the 3 organisms yet. Now in phase 2, we are testing just the chain elongation part. And why we want to do this is because ethanol is actually a substrate for both. So they could actually be competing for this substrate. What did happen? Well, when we did the trials in batch, we could have carbon monoxide conversion and production of mainly acetate and propionate that was remaining in the medium, and a little bit of C5, so of valorate. We could see that both organisms are in the culture. You have cloiveri that is a little bit thicker, and you have the Enerotignum that are these thinner rods. So we did this in continuous to try to improve the performance of the system. And indeed, after a startup phase, we have a very stable co-culture in which we produce both odd chain and even chain fatty acids, with odd chain fatty acids representing about 40% of the fatty acids that are generated. So we are now with phase 1 and phase 2 proved. We need to do phase 3, something we are working on at the moment, and that I hope to show you in the next talk. So to conclude, we showed that these synthetic cultures can be used to expand the range of products from syngas fermentation, so as an alternative to genetic engineering. And that was, the case was the production of chain elongated acids. And we also could see that our co-cultures are very stable. We could run them in bioreactors for months without crashing. For the future, we want to optimize these co-cultures, for example, adding more organisms so that we can have more robust and also co-cultures that can handle better changes in operational parameters. And we want also to include a module for CO2 fixation that is generated during carbon monoxide oxidation. And with this, I want to finalize, thanking your attention and thanking, of course, the people that were doing the work in the lab, Martijn and Yvette in my lab, and also in collaboration with the University of Minho, Ana Luisa and João. They were both in Wageningen also working on these topics. So thank you very much.
Thank you very much, Diana. Very nice presentation. I will share now the question. So, do you see the symbiosis between microorganisms as a requirement for the co-culture approach?
So here they are, these 2 organisms, the first culture that we work with. They don't need to live together. Well, at least not the first one. The second one is depending on the first one because we are not feeding any ethanol or acetate. So the second one, it needs the activity of the first one. So they don't need to be obligatory syntrophs, they don't need to be obligatory together, but I think they benefit from being together. And a way they benefit is by thermodynamics. So having a second organism that uses the product of the first stimulates the first to produce those products. And we also saw, and I did not show here, but we saw many, when we did transcriptomics, we saw many other interesting clues on how they might be interacting, and that involves also, for example, the generation of flagella and pili. So they do have a reaction on the fact that they are together.
Okay, so only one more. do you think that syngas production by fermentation will replace the conventional production methods?
Well, this is a nice question. Of course, I don't know, but I don't expect that. I think we need in the future a range of different processes to, and each process will be better in a certain situation. So I guess that syngas fermentation will be useful for treating more recalcitrant waste, so that we can have this combination of valorizing also recalcitrant wastes that are very difficult to convert directly by other fermentations. But of course, for very easily fermenting materials, I think just biological processes might be more economically more feasible. But gasification technology is improving, and it's getting also cheaper. So I do think there's a room for this in the future.
In the future. Thank you very much, Diana.
Thank you, Sarah.
We will move to the next speaker. The next project will be presented by Marcelo Tenio. Marcelo is a researcher at Embrapa, dairy cattle in Brazil. He coordinates several research projects dedicated to water quality, management of biological waste, bioprospection of dairy cattle effluents, and also the production of electric energy using biogas from bovine manure. So please, Marcelo, can you start sharing your screen?
Hello, Sarah.
Hello.
Thank you for invitation. Hello, guys. Let's start.
Yes, you can start.
Okay, my presentation?
Yes, we can see your screen.
Okay, let's go. I speak of the co-digestion of agro-industrial residues in production of bioenergy and prospecting for potential biomass. We participate with a group, and we work with a new approach to agro-industrial residues. Here, waste not as a problem of production, but as an input for agriculture and for the production of clean energy. Here, this photo, beef cattle in Paraná, Brazil. And this photo, clean floor in Embrapa dairy cattle, in my farm, experimental farm, here in real scale, Minas Gerais, Brazil. A bovine eliminated 9% of its weight per day in excreta. A dairy cattle produce in excreta and feces and plus urine 15 kilograms by day. These residues can be an opportunity for the producer if properly managed. We work with anaerobic digestion. It is a natural and complex process that occurs spontaneous in environment and in the digestive system of ruminants. The ABE is a metabolic process with multiple syntrophic relationships that occur between microorganisms to degrade and transform the biomass into carbon dioxide and methane. I want a lot of methane, okay. The degradation of organic matter is divided in 4 phases. Hydrolysis, acidogenesis, acetogenesis, methanogenesis, and in each there is the activity of
specific microorganisms. This is our Embrapa biodigester. It destacate it's necessary a separator to procedure a separator and the fibers of the feces, bovine feces, and sand separator because the bed of cows, and finally a plug-in flow digester produce biogas, and finally a biofertilizer. We apply this biofertilizer in an experiment and normally in farm. The extensive use of bovine manure, BM, in biodigesters in Brazil has a direct relationship with the diffusion of cattle farms in this country, in our country. It is estimated that the number of cattle in Brazil is close to 213 million head. Anaerobic co-digestion is a simultaneous and homogeneous biodigestion of two or more complementary substrates to produce biogas rich in methane, while reduce the supernaturals in the environment. This practice is recommended especially for recalcitrant substrate as industrial waste. Clean floor in Embrapa Dairy Cattle Farm. And we collect feces, fresh feces, raw feces, and process it at Embrapa in Juiz de Fora laboratory. Separate solids, fiber, and an appropriate choice of co-substrates, their missing proportions, and the analysis of operational parameters of the process are important points to consider. While designing a co-digestion system, these factors will determine the dynamic of microorganisms and efficiency of process. BM and wastewater are weekly collected in Embrapa Farm, and other residues, food industry residues, ricotta cheese whey, RTW, and residual sludge of brewery, RSB, are examples of byproducts with right pollution potential, and we worked with these two residues and prepare here. We RBS plus BM and RTW correct PG and plus bovine manure. In this concentration of mixture, 20, 14, 18 to 2 residues. And the control, pure bovine manure, BM. This is a model of laboratory biogas system of anaerobic biodigester, 16 liters, and your gasometer. The real photo in operation last year. And the experimental design had three phases. First phase to acclimatization and change anaerobic system to anaerobic system. Phase 2, where you put on the mixture by 13 days, and with these days, we finished the change of influence. And phase 3, anaerobic co-digestion fully, okay? And we analyze temperature, gas, volume, and collected a sample of gas to chromatography and microbiology and physical chemical analysis. And our results here, methane production by 120 days. It's interesting to observe. We RSB 18%. It's not possible to good evolution, and with 100 days stop to produce methane. Others mixture, others proportional mixture have a like control, not more better, but maintain a possible to management this reason. Page valence, it's not problem in biodigester RSB 18%. It's not PD. No acid or this is a big problem. We have a big production of ammoniacal nitrogen in this biodigester. And the better results are 80 dBU, 20%, 14%, 18%. And here, hydrolytic bacteria, E. coli and Pseudomonas aeruginosa. And here, show hydrolytic fall, E. coli and Pseudomonas aeruginosa to berry. Okay, and here control. And now here stability of the count bacteria. But we during archaea identification during anaerobic digestion, right evidence of Methanosarcina genus was observed in DM samples. And Methanosarcina and Methanoseta are responsible for producing a large part of methane generated during anaerobic digestion. This is control, both in manure pure. Here, with brewery results, is clear beginning of the congestion dominated by Methanosarcina and Methanocorpusculum with and prevalence of Methanobacter. I think so. A problem of nitrogen. And here, the same or the like bovine manure, we have results of RTW biodigesters. And we have, it's possible to use the right concentration of ricotta cheese whey in small anaerobic digestion system. The concentration of brewery slurry in the system interferes in the archaea dynamics. Biomass from agriculture and industrial results from the production of biogas diversifies the energy metrics with energy from renewable sources, supporting sustainable development. Okay, my contact, grateful.
Thank you very much, Marcelo. We will now see the questions on Slido. Just a few minutes, seconds. I will share now. I think we have time only for one question. So, how does the Brazilian government stand in terms of policies to promote this type of waste valorization?
Okay, thank you for the ask. Brazilian, there is programs, specific programs to financial waste valorization to agricultural producers. We have determinate of ABC program of agriculture, lower carbon to promote this kind of policies.
Okay.
But research of this is not very good. I think so.
Okay.
Financial not common. It's very difficult.
Better
now.
In the future, maybe.
Yeah.
Thank you very much, Marcelo. We have to move to our last presentation, last key project that will be presented by Gonçalo Lourinho. Gonçalo is a researcher at Bioref, a collaborative laboratory for biorefineries. He develops his research at Alentejo Hub in Porto Alegre, Portugal, and he has been focused on solid and liquid biomass conversion into energy and value-added products. Please, Gonçalo, you can start. You have to turn on your micro. Okay.
Okay, sorry.
Yeah.
Thanks, Sarah. Hopefully you can see my slides. Can you confirm?
Yes, yes.
Okay, great. So good afternoon, everyone. First of all, thanks for having me, and thanks for the introduction. It's my pleasure to kind of round out this panel by providing some insights of what we're doing at Bioref. My talk is really going to look at how we can maybe direct our efforts in terms of advancing biorefineries in agro-industries, but let me start with a few words about our collab. Okay, so Bioref is then one of several collabs that started in 2020. We are composed of 20 associates, 9 academic entities, and 11 companies, all very interested in investing and entering these novel markets of advanced technologies. And we intend to be one of the key players of this change, at least in Portugal, and help to improve the competitiveness of the biorefinery sector, and at the same time contribute to a more circular and a low carbon economy. In line with our vision, we have defined some strategic targets that are set for the next years. And in general, we aim to promote a market-oriented research and innovation agenda, which is based on the development of biorefinery-related technologies to achieve our goal of decarbonization. Bioref aims to be recognized as a technology provider, mostly as a technology provider in this area of biorefineries, but we know that to really innovate and have a market impact, the interaction between our collab, private companies, and universities is of great importance. And our role here is really to
to try to develop and strengthen this relationship. Our business model states that the basic knowledge needed in terms of fundamental research is developed in our academic partners, mostly in activities with TRLs from 1 to 3. And we want to play a role on advancing these technologies to TRLs between, I would say, 5 and 6. And this is what creates, in our opinion, the right environment for cooperation and for knowledge transfer to the industry. Right now, we have around 20 highly skilled human resources. Our staff is currently divided into 5 research teams on 4 locations. We are based in Matosinhos, but we have other hubs, one in Porto Alegre, other in Aveiro, and another one in Lisboa. Our R&D activities are also divided into areas or domains, one more linked to bioenergy, the other to renewable gases, and another one more dedicated to sustainable bioeconomy. The research teams work in these strategic areas according to their expertise, but of course we collaborate a lot when necessary, and we really work in a network. Our bioenergy domain covers a set of technologies generally used to produce energy from biomass and waste. I include here, for example, the thermochemical conversion of biomass for syngas, or to produce advanced liquid biofuels. The renewable gases domain includes a set of technologies to produce biogas, biomethane, green hydrogen, through, for example, anaerobic digestion, also gasification, and also water electrolysis for power to gas. And finally, the sustainable economy domain considers technologies for the production, recovery, extraction, and purification of bio-based products. Okay, this was just to give you an idea of what BioRef is and what we do. Moving on to our topic of today, I think everyone knows that the concept of circular bioeconomy is really the key regarding the future transition to sustainable development. And I would say that everyone agrees that agro-industries should be at the center of this transition because they pose serious challenges to ecological systems, but are at the same time important for social and economic development. But I would like to argue that to achieve this change, we need to start seeing agro-industries in a different manner. This is nothing completely new, but my first point today would be to suggest that we, in fact, need to start conceptualizing these industries as biorefineries. We know that, in a sense, biorefineries are nothing more than factories where biomass can be converted to chemicals, electricity, or heat. However, for these novel agro-industries, this understanding seems a bit narrow. And I think it's better to try to rethink agro-industries in a broad way, for example, as larger production systems that incorporate different small farms or different small factories, which may be geographically dispersed and operate throughout the entire value chain, let's say from raw materials to raw materials production to consumer goods production. But let me explain a little bit better what I mean. I am suggesting that agro-industrial systems should not only be viewed as isolated targets for sustainability measures or for specific policies, but we should understand these facilities on this larger scale as, for example, interconnected nodes in a network. And this is, of course, related with the concept of biorefinery, and is a critical step if we want to advance the bioeconomy and scale our current use of biomass feedstocks to produce chemicals, energy, and fuels. Then I would also like to argue that these future agro-industries should have certain features. So first, future agricultural production should be an integral part of the circular bioeconomy, which is to say that residues are received from one or other production systems and then returned to the biomass cycle. This means that future agro-industries should be viewed mainly as waste biorefineries, in which waste recycling is completely integrated with traditional production. Second, I would like to argue that the process of anaerobic digestion should be at the center of this transformation, because this technology really is like a cornerstone for residue management, and in my opinion, a valuable way to provide clean energy source and a strong economic basis for operation. Finally, the future agro industry should also be knowledge-based and information-driven. Now I would like to give you an example of a bio-based biorefinery, just to illustrate the opportunities that current agro industries will have in the future. So this biorefinery consists in the production of biomethane for power, mobility, and heating using different biomass feedstocks. I call your attention here to the word flexibility. What we want is to become more and more flexible in terms of input, in terms of the process itself, and also in terms of our output. On the input side, a much more diverse, including here difficult to degrade feedstocks should be an objective, okay? And on the output side, a more flexible biogas production and flexible outputs in terms of the type of products resulting from the process should also be always considered when possible. But let's maybe look to a more practical example. Let's imagine that in a given region, it is strategically decided that we should develop an existing livestock production industry. So current facilities are responsible for point source pollution, and this pollution has created problems in lakes and water streams in nearby soils, and has also created conflicts with local communities. So from a joint venture between the academy and the industry, a novel technological process around the treatment and transformation of these livestock wastes into biomethane and organic fertilizers is developed based, for example, on the co-digestion of these wastes with algae. So this process is implemented, and meanwhile, what we have is local industries that can now be more competitive because they can recover energy from waste, and they can reduce production costs, and they can also increase revenues. Then it turns out that valuable co-products can also be extracted from the algae, which is used in the co-digestion. So with the residues produced from this process also recycled for biogas production. And all this means that what was before a small group of polluting companies competing independently is now an innovation network that can integrate several other firms in the region, each one specializing in a different aspect of a novel local bioeconomy chain. So we have livestock production, we have biogas upgrading, we have organic fertilizers, we have algae cultivation, we have algae-based products, we can have biodiesel production, we can have R&D. And so from this overall situation, biodiversity is probably enhanced, local pollution is probably reduced, and social resilience is also improved by the creation of new jobs and the creation of novel income opportunities. So these are the types of solutions that I think need to be considered, and in my opinion, given a great deal more attention. This is just a final key message for highlighting that we should extend the use of some technologies behind what we have today, and should really start to conceptualize agro-industries as these waste biorefineries, and not only as isolated targets for specific measures. And with that, I'll finish. Thanks so much.
Thank you very much, Gonçalo, for your nice presentation. Very interesting. I will share now the questions. And so the first question: what are the key aspects to implement an economically sustainable biorefinery?
Okay, this is a very interesting question, but at the same time, a very broad one. I think that the key for really sustainable biorefineries is process integration and that flexibility that I mentioned in terms of inputs and in terms of outputs. So if we can produce several valuable products at the same time through as many integrated processes as we can, this will probably result in economic sustainability and economic feasibility, and this is really what we want. So in my opinion, at least, process integration is really the key here.
Okay, just to finish, how close are we from having these bio-based biorefineries at large scale?
Yeah, actually, I think we are really close. We have several projects, not only at BioRef, but around Europe that are focused on not only the development, but in the implementation of these kinds of solutions. We have several projects trying to produce green hydrogen from power to gas processes right now. So this can be considered a bio-based biorefinery. We have other projects based on, for example, biomass gasification that have already been implemented. So we are very, very near, and I think that until 2030, maximum 2050, we will have several examples of bio-based biorefineries, not only in Europe, not only in Portugal, but also in Europe.
Okay, thank you very much for your answer, for your
Thank you, Sarah.
And for coming. Now we will start a new session, the flash presentation session, and in this session we will have 6 different presentations, and we will have time for questions only at the end. So first you will attend all the presentations, and at the end we will have 5 minutes to answer your questions. We will start with José Salgado, who works as a distinguished researcher at University of Vigo, Spain. And in this case, we have a video to share, right? Please, you can play the video.
I will present the research work on valorization of agri-industrial waste by solid-state fermentation for aquaculture applications carried out in collaboration with researchers from the Biological Engineering Center, led by Dr. Isabel Bello and me, and researchers from the University of Porto, led by Dr. Elena Perez. Fish consumption doubled since 1960 as fish and other aquatic organisms are important nutritional source with economic benefits. Since 2014, aquaculture sector surpassed wild captures aiming human consumption, and by 2030 it is expected to provide up to 60% of fish for human consumption. However, aquaculture faces some challenges that are important to consider to achieve its long-term sustainability. Most of fish capture aiming non-food purpose is used to produce fish meal and fish oil. These are important ingredients in fish feeds, given their high nutritional value. However, given the over-exploitation of marine resources, it is necessary to find innovative and sustainable alternatives to these traditional ingredients. The agriculture-based industry produces vast amounts of waste that can be environmentally negative and do not have commercial income. Therefore, our industrial byproducts are an opportunity to be used as alternative ingredients in fish feeds, but their direct use is often limited given their low digestibility and low nutritional value. The valorization of our industrial byproducts can be carried out using solid-state fermentation, an eco-friendly biotechnological procedure that consists in the direct use of our industrial byproducts as substrates towards production of valuable compounds and improving their digestibility. The fermentation occurs with low moisture and with no free water. The microorganisms that are more suitable to grow in these conditions are fungi,
due to their ability to grow in low water activities and high osmotic pressure, penetrating in the substrate due to their IFAL growth. The solid-state fermentation is a simple and low-cost process, utilizes low-valued products, and at the end of fermentation is possible to recover a highly concentrated product. However, it presents some disadvantages such as parameters monitoring, the separation of biomass, and is difficult to properly scale up. The utilization of this technique contributes to circular economy as it promotes the utilization of disposable agro-industrial products, obtaining valuable compounds and therefore improving resource efficiency with no waste generation. Our work aims the utilization of a variety of byproducts to produce alternative ingredients and compounds for incorporation in fish feeds. To replace fish and oil and fish meal, we use solid-state fermentation to produce polyunsaturated fatty acid and fungal protein. Aiming to improve feed utilization and digestibility of plant feedstuffs by fish, we use solid-state fermentation to produce non-star polysaccharide enzyme to be added as an additive in fish diets. To assess the potential application of these novel products, growth on digestibility experiments were carried out. For the production of polyunsaturated fatty acids, brewer spent grain, the major byproduct generated by the brewery industry, was fermented with Mozzarella alpina, an oleaginous fungus capable of elongate and desaturate fatty acids. At the end of solid-state fermentation, it was obtained a modified biomass with high lipid content, namely eicosapentaenoic acid, linolenic acid, and alpha-linolenic acid, and the protein content was increased. In other studies, binary and olive meal waste were also used to produce an extract with high antioxidant activities by solid-state fermentation. This extract was then incorporated in fish diets. Using in vitro digestibility trials, it was observed that this extract increased polyphenols released from the feed ingredients. Therefore, this extract can be advantageous to provide natural antioxidant compounds to fish and improve fish fillet quality. Another substrate used in solid-state fermentation was Ulva rigida, a green macroalga, which the potential applications are still unexplored. The solid-state fermentation was used to improve its nutritional value. A successful scale-up of the fermentation was carried out, starting with 10 grams of seaweed in early major flask up to 400 grams in tray-type bioreactors. Silanase, cellulase, and beta-glucosidase were produced, and it was possible to observe that the upscale of fermentation conditions did not compromise enzyme activities. The solid fermentation successfully altered the structure of Ulva rigida, as 46% of total polysaccharides were removed by fermentation of unwashed Ulva rigida. The inclusion of this fermented Ulva rigida was tested in diets for European sea bass, an important aquaculture fish species. Fish fed with 5% of fermented Ulva rigida had higher growth performance than fish fed untreated Ulva rigida. In addition, feed efficiency was highest with the fermented seaweed. In conclusion, solid-state fermentation of agro-industrial waste and seaweed resulted in high yield of non-star polysaccharide enzymes. Solid-state fermentation allowed to produce polyunsaturated fatty acids, increase the protein content, and to reduce the polysaccharides of agro-industrial waste and seaweed, increasing their nutritional value to be used as aquafeed ingredient. The enzymes and antioxidants produced by solid-state fermentation can be applied as additive in aquafeed diets. Future research should be focused on increasing the scale of the process and optimization of aquafeed diets formulations with fermented products. Thank you for your attention.
So thank you very much, José. We will move to the second flash that will be presented by Daniel Gomes, a researcher from Center of Biological Engineering, University of Minho, Portugal. Please, Daniel, you can start. Okay. You have to turn on the micro. You have to turn on your micro, Daniel. Down in the.
Are you seeing?
Yes,
are you
yes,
hearing?
now,
Yes.
now,
Sorry.
yes,
Are you seeing now the
yes,
slides?
we can see the presentation. Yes.
Okay. Let me just extend. Yes.
Yes.
Okay. Sorry for the mess. So hello everyone. Thanks for having me. I will now briefly describe some research I have been working to reduce the cost of cellulosic ethanol. So as many of you probably know, the cost of the process of utilizing lignocellulosic materials is commonly limited by not only the high cost of feedstocks, but also the enzymes used to convert them, among other factors. The results that I will be here present address these two issues. So the first effort on this direction involved the utilization of recycling paper sludge. This residue is generated in very large amounts during paper recycling, and is mostly consisted of residues from paper processing, such as ink particles, but most importantly, small fibers that cannot be further used. Most of this residue is typically burned or disposed on landfills, which represents a considerable economic and environmental impact. However, this material still presents an interesting composition, as our results confirm. Using a sample from a Portuguese paper manufacturer, and after simple acid neutralization to remove an excess of carbonates, these materials showed a cellulose composition of approximately 31%, which is an interesting level. We then tested this material for its conversion with cellulases and cells. As you can see in the plot to our right, even without any pretreatments, the cellulose cocktail was able to convert cellulose very fast. The same happened for cells, as sugars were converted into ethanol in less than 6 hours. This suggests that no major toxicity effects are caused by these residue components. On a second part of this work, we tried to implement a system to reduce enzymes consumption for this particular process. One possible way to reduce enzymes consumption is to use them several times, which is commonly known as enzyme recycling. Before such system could be tested, it's important to assess some aspects of the enzymatic system, such as the enzyme stability and their distribution at the end of hydrolysis. For that, we can see that during a process of enzymatic hydrolysis, using the previous residue, we measure the levels of cell 7A activity. This is the most abundant cellulolytic domain on Terese cocktails. And as we can see, nearly 77% of enzymes activity was conserved during the process of hydrolysis. Also, 70% of the enzymes were free at the end of hydrolysis, which is very positive to achieve a simpler recovery process. Still, we can see that 30% of the enzymes were bound to the solid. So to recover this fraction, we tested a process which is commonly known as alkaline elution, which basically consists on changing the pH of the enzymes to 9 or 10, so they dissolve from the solid and can be recovered as the free enzymes. This step was very efficient, as we can see, as nearly 82% of the solid bound enzymes were recovered. So considering the previous indications, we designed a cellulose recycling system on which, in the first round of hydrolysis,
The solid, in this case, the recycling paper sludge, was hydrolyzed with a dosage of 20 FPUs per gram. And on the next rounds, only half of this dosage is applied, which will complement the enzymes which came from the previous rounds. So as you can see from the levels of cell 7A activity, even though for every round of hydrolysis we can see a loss of activity during hydrolysis, but also presumably from the process of enzyme recovery, this addition of only 50% of enzymes allowed a full conservation of activity levels during the tested for consecutive rounds, presumably even for more rounds. In a similar way, we can observe that although the small reduction that we can observe in the level of glucose conversion from after the first cycle, this remained stable in the next rounds of hydrolysis. This demonstrates that cellulose recycling can be reutilized, can be applied, in this case, can be applied as long as we have a favorable enzyme stability and enzyme recovery at the end of the process, which ultimately will allow important savings and improvements on the economic output of this kind of process. So thanks for your attention.
Thank you very much, Daniel. Sorry. Thank you very much, Daniel, for your nice presentation. Now we will have a flash presentation presented by Carla Silva. She is currently leading the Chemical and Biotechnological Department at CITEV, Portugal. Carla, you can start.
Yes, just sharing my screen.
Please, yes.
I hope
you are
trying to put, now I think you can see, yes?
Yeah, okay.
Okay, so good afternoon to everybody, and I would like to start by thanking the organizing committee, in particular to Professora Ligia Rodrigues for the invitation. So I will just make a brief presentation on the use of bio-based fibers and additives for sustainable textile industry. I'm from CITEV and I'm coordinating the Department of Chemistry and Biotechnology. We are based in Famalicão. And just very briefly, a short overview of our evolution. So we have 32 years and we were created in '89, and our history is encompassing the textile industry in Portugal. So in the 90s, the issue was quality. And that's why we had created our training school to give training specifically on this topic. Then it was quick response innovation, where we created CENTI, the Center for Nanotechnology and Smart Materials, in 2006. And now we are in a breakthrough where we have a strong focus to make a change for a sustainable and circular textile industry. And in this topic, there are several issues that we are exploring simultaneously in order to have circular textile processes, but also to explore recyclable textile materials and using renewable sources as the raw materials. And together with this, there are activities running in parallel, where the design for circularity and creating new business models to explore these materials and other approaches for end-of-life alternatives, and also validating this circularity and the transparency in the value chain. That is also a critical issue in the sustainability of the textile industry. Just to give some examples of renewable raw materials that we can explore. So we can focus on using marine-based materials. Algae is an option. Also, we can explore developing fibers from exopolysaccharides extracted from bacteria, where we have, for instance, bacterial nanocellulose as one example, but we can also think on bio-based polyesters and other polymers that can be produced by fermentation. The use of forestry residues, as you may be aware, the textile industry uses artificial fibers that are produced from cellulose, so it's dissolved cellulose, so we can also explore that renewable raw material, and other alternative natural-based materials like agro-industrial residues.
But to use these alternative materials, there is one issue, and there are some gaps that we need to address. So we need to develop new machinery, because our process and conventional textile process, they are not adapted to these new materials. And while doing this, we also have to think on exploring sustainable processes in all the value chains. So we can explore enzymatic processes, also hydrothermal or green chemistry approaches in the most chemical intensive process that we have in industry, which is the finishing processes and also the dyeing processes, where we can explore alternatives that are more sustainable. In this scheme, you can see an overview of our approach. So we are exploring alternative fibers and bio-based fibers, for instance, that are residues to other industries, for instance, the straw from the cereals collection, and also some leaves and stems from fruits collection and forestry residues. And our approach is to transform these cellulose-based materials and other additives into fibers. And a first step is a defibrillation, which is quite critical, because we need to defibrillate the fibers to be able to spin them. And we can also spin them by conventional cotton type spinning processes, or dissolve them, as I mentioned before, to produce cellulose-based fibers, and that we can use to produce our textile structures. In parallel, we can use these residues and these bio-based additives to explore different dyeing processes, for instance, and also to develop new additives, functional ingredients to provide, for instance, antimicrobial activity to our textiles. And finally, to close the loop, we cannot look, we have to have a cradle-to-cradle approach, so we can explore different end-of-life alternatives, where we can reuse the textile garments or transform them into different products, and finally recycle them, and they can again join the loop in a circular process. You may not be aware, but more than 90% of the textile fibers that are used in European Union are coming from Asian countries. So Europe is not sufficient, is not able to use fibers to be sustainable in fibers. So I'm now giving some examples of some bio-based fibers and additives and some process, just to give you an overview of some of the projects we are having. This FibNatex project was a European project, a Sudway project, and we were the coordinators. And this project main ambition was to use hemp fibers to develop alternative textile products. As you may know, hemp is having a huge growth, so it was a fiber very used in the past, and now it has revived and is being used again, where the oils are used in the food and pharmaceutical industry, and we are using it to explore the fiber as a residue, but that has many important and interesting properties for the textile industry. So in this project, we were exploring the individualization of the fibers. There are several operations that needed to be addressed, and then producing first at lab scale and finally at industrial scale some prototypes and characterizing them, and then producing some prototypes of the fabrics that we achieved. By one side, we have coarser fibers, so fibers that are very thick, and the application has to be more thicker products. But we were able also to achieve very thin, lightweight fabrics that can be used in t-shirts, for instance. Another application is another project. In this case, it was a national funded project. It's Corkatex yarn. And in this project, we were using cork residues as the raw material to produce innovative textile structures. You may know that cork is one of the most sustainable material that you have. You remove cork bark from the trees every 7 to 8 years. So we were using just the powder that has no application, and we were using it to coat cotton yarns
And with these coated yarns, we were able to achieve different colors, just varying the cork powder and doing some sustainable pre-treatments. And we were able to have different colors and that could provide different shades and different properties to the fabrics. And here are some examples of home textiles, a collection that were developed with this Corkatex yarn. Another application is to use the bio-based additives for natural dyeing. Natural dyeing is a very important theme in the textile industry because textile dyeing is one of the most polluting processes. So in this project, we were using extracts from herbs and from mushrooms to produce several colors. And we were able to achieve colors with high fastness to light. And when we are using natural dyes, light fastness is a critical issue. But in this project, we were able to solve using a very sustainable and natural friendly project that additionally provided some antibacterial properties also to the fabrics. And finally, another application resulting from the mobilizing project TextBoost. In this project, there were around 40 companies involved. And in particular, we were exploring in one of the projects using residues from cork powder, from wood, from coffee beans, and other bio-based ingredients to develop coatings. And with these coatings, we can use them to coat furniture, to produce shoes, or to produce some fashion applications also. Thank you for your attention.
Thank you very much, Carla, for these interesting applications. Very nice. We will move to the next speaker. That will be Alcina Pereira. She is an assistant professor at Department of Biological Engineering, University of Minho, Portugal. So please, Alcina, can you start sharing? Okay.
Okay, so good afternoon to all. First, okay, it's not yet in presentation mode, sorry. Okay, so first I would like to thank the organization for inviting me to give this presentation and have the opportunity to tell you about a project that is focusing on the biological treatment of saline oily wastewaters, coupled with the recovery of valuable plants. Well, in offshore oil extraction activity, large volumes of seawater are injected to maintain the hydraulic pressure in the reservoir and enhance oil recovery. The extracted crude oil is then subject to a physical separation process to remove the aqueous phase that is called produced water, and that it still contains significant amounts of hydrocarbons and other contaminants, and so it needs to be treated before disposal. Conventionally, produced water is treated by physical and chemical processes that have high energy consumption, startup and running costs, and can also produce hazardous sludge. Also, these processes often are not able to remove small suspended oil particles and dissolved elements from oily wastewater. And so a biological treatment process can be a good alternative to treat this wastewater using marine bacteria that are efficient in degrading hydrocarbons. Some marine hydrocarbon degrading bacteria, such as Alcanivorex porcomensis, are able to accumulate storage lipids in the form of triacylglycerols and wax esters when they are submitted to unbalanced grow conditions, so under nutrients limitation. These storage compounds are currently of great interest as raw materials for the production of additives for feeds, cosmetics, lubricants, biofuels, and other products. So we propose to use a combined process for treatment and valorization of produced water, applying conditions of high carbon to nitrogen ratio called the feast phase that favors the production of these valuable products while removing hydrocarbons from the water. Afterwards, growth is promoted by adding nitrogen in another phase called famine, and then a next cycle of feast can be performed. Proof of concept was first demonstrated in a steered batch reactor inoculated with Alcanivorax subercomensis that was able to remove up to 98% of C10 to C33 alkanes from produced water while producing both wax esters and triacylglycerols. And afterwards, in a sequencing batch airlift reactor, that was proved to be a suitable technology for treatment and valorization of mineral oil wastewaters towards the production of lipids. In this work, the process was further evaluated by testing different total petroleum hydrocarbon to nitrogen ratios and also different feast-famine durations. As main results, I would like to highlight that we were able to achieve total petroleum hydrocarbon removal efficiencies up to 96%, while obtaining biomass with intracellular lipids content up to 85% of its cell dry weight. And this was found for higher feast stage durations and lower total petroleum hydrocarbon to nitrogen ratios. Intracellular lipids were mainly composed by triacylglycerol, while extracellular lipids were composed by wax esters. In addition, the analysis of the microbial community developed in the reactor has shown that Alcanivorax was the predominant bacteria, and those that the inoculum was able to outcompete the indigenous microorganisms present in the wastewater, ensuring the production of bacterial lipids. So, after this flash presentation, I hope that I was able to convince you of the potential of using a combined biotechnological process for treatment and valorization of produced water, obtaining a valuable product that is the bacterial lipids. Our current research is focusing on the optimization of lipids production and bacterial lipids valorization, as well as in the selection of additional treatment process for removal of other contaminants that are present in the produced water, like, for instance, sulfate. And with this, I end my presentation, and I would like to thank you for your attention.
Thank you very much for this interesting presentation. Our next flash will be presented by André Ribeiro. André has been working as senior researcher at Center for Waste Valorization, University of Minho, Portugal. Please, André. André, can you share the screen? Okay.
Yes.
Yes.
Everyone is seeing,
okay.
I believe.
Yes.
Just put in presentation mode. Okay. So first of all, I would like to congratulate the organization committee for
pulling out this event, this very interesting event in this turbulent times. And in my name, in the name of CBR, I would like to thank the invitation for participating on this flash presentation. My name is André Ribeiro. I'm a senior researcher from CVR, and today I'm going to present some of the results of the electronic remediation applied for the treatment of heavy metal contaminated soils. I will just brief present the CVR and then go to the results. CVR is a non-profit organization, association founded in 2002, has own facilities, have 18 permanent collaborators and collaborations with more than 30 universities in Europe and United States, Latin America. And our vision is to transfer the technology applied to waste and management valorization. Nowadays, everyone is talking about circular economy, and just from the start, CVR was applying the circular economy in all of these technologies and solutions that developed. CVR is located in the University of Minho, in campus of Azarém, of University of Minho, and has these four areas, main areas of actuation: analytical service, consulting, the research and development department that I belong, and the organization of conferences. So regarding the analytical services, we have these laboratories. The waste characterization and gas emissions are accredited with EPAC accreditation. So in the consulting, I can highlight, for instance, the life cycle assessment studies, and of course the classification of hazards and unhazardous waste, the valorization, the energy valorization potentials, the soil health characterization, and regarding the research and development department, we perform projects, national and international projects, regarding all kinds of waste approaches, organic and inorganic different type of wastes. For instance, already someone talked about pyrolysis, gasification experiments. We also perform that, the waste incorporation studies, development of biological treatments, discontamination and recovery of metals, and so every kind of project related to the wastes. And 22 years, we organize a conference that we call Wasted, that of course everyone is invited to participate. The next one will be in 2022 in Coimbra. So everyone is invited to present the results in this conference. Regarding the electrokinetic remediation, in the framework of this project, Minho, we developed a pilot scale prototype with the capacity of treat around 300 kilograms of soil. So in this presentation, I will just show some of the results that we achieved in this project. Unfortunately, these are common pictures of waste of soil contamination. This is a spill in pharma mining in Brazil, I believe, and everyone knows that desertification is it's nowadays a very complicated problem. So soils are one of the natural resources that we want to preserve. So this graphic represents the principal sources of contamination. As you can see, waste disposal and treatment and industrial and commercial activities are the main sources of contamination of soils. This is a very interesting report that was elaborated by the European Commission that is called the Status of Local Soil Contamination in Europe, and they give us an overview of the contamination of soils in Europe.
And the results of that report, some of the results you can see, for instance, in this graphic. They conclude that there are 127,000 contaminated sites in Europe, and potentially contaminated, there are something like 1 million potential contaminated sites only in Europe. And heavy metals are the major contaminant that contributes to that. 35% in the overview of contaminants affecting the soil, and something like 30% of the groundwater that probably will contaminate the soils. So I will not explain this table in detail. These are the sources of the anthropogenic sources of heavy metals, and the toxicity and the effects that they have in the human being. Everyone, I think that everyone knows that heavy metals are very dangerous to human being. It can be cancerogenous, for instance, so we want to avoid that. And there are several technologies for treat or discontaminate soils. Electrokinetic remediation is one of them. This technique is based on the application of a direct electric current of low intensity to the porous matrix of the soil that you want to be decontaminated. But although it could be simple, it's a very complex technology, it's a very complex process, because they have a lot of reactions that we must take in account. For instance, electrolysis of water, the electromigration, electroosmosis, electrophoresis, and the diffusion effects on the soil. So it's a very complex process. Other thing is the use of permeable reactive barriers. There are this PRP are a reactive material. We use low-cost adsorbents, but of course it can be used biological bacteria or microorganisms to perform the decontamination to perform the fixation of the metal into this material. We use, and we already tested lots of different low-cost adsorbents, for instance, the inorganic fraction of eggshells, the inorganic fraction of bivalve mollusks, and the biochar from pyrolysis of textile and polymeric wastes, and the conventional activity carbon. So we developed in the framework of the project, Minho, like I said, this combined model that we call it the electrokin barrier. So in one hand, we have the electroremediation and the use of permeable reactive barriers to absorb the metals that are transferred into the soil. This is the actual prototype that we developed. And regarding to the results, these are the results in the artificially contaminated soils with chromium and lead. As you can see, we achieved, I think, good results with different ranges of initial heavy metal concentration. For instance, in the lead, we achieved something like 80% of removal rate, the chromium 15% or 60% in the low contamination with less initial chromium concentration. We also tested in the wastewater treatment sludges. Nowadays, the sludges from wastewater treatment plants, some of them has, I don't know if you know that, but some of the sludges has a very high concentration of heavy metals, because in the wastewater treatment plants.
Sorry, can you finish?
Yes.
Possible? Sorry.
Of course, they also treat industrial wastewaters, so the sludges has high concentration of heavy metals, and we tested also in that material with this technology, and we achieved very good results also.
in some of the cases, between the limits of the Portuguese legislation. And we also tested in non-commercial organic composts, and we achieved it. So, like I said, also good results with removal rates of, in some cases, more than 19%, which is very good, which is very great. So, conclusions. This technology, this in situ treatment, can be used to treat different types of solid matters contaminated with heavy metals. They have the ability to remove different types of heavy metals. The use of permeable reactive barriers from the different sources. It is a case of waste valorization that if not, this kind of materials were disposal in landfills. So the challenges for this technology is to optimize the discontamination model in real solutions or scaling up this one, and of course to promote a circular economy approach and try to reuse the metals that are adsorbed or regenerate the adsorbents. So thank you.
Thank you, Andre. Thank you very much for your presentation. We will finish this session, the flash presentations, with the flash presented by Andrea Salvador, a researcher from Center of Biological Engineering, University of Minho, Portugal. Please, Andrea, the floor is yours.
Hello, just a minute. Can you see the presentation? Yeah, maybe now.
Yes, yes.
Okay. Remove this. Okay, okay. How can I remove this? Okay, so good afternoon to all. Thanks for the invitation to talk here in this workshop. My name is Andrea Salvador, as Sarah said, and I'm from the Center of Biological Engineering, more precisely from the Bioresources, Bioremediation, and Biorefinery group. My talk today will be about waste conversion to methane, lessons from microbial communities. So different kinds of wastes have in common carbohydrates, proteins, and lipids that can enter the anaerobic digesters. And inside these digesters, we have different microorganisms, bacteria and archaea, that interact with each other and convert these molecules to biogas, to methane and CO2. And this methane and biogas can be used in different applications, such as the transportation, and I have here 2 examples. And to attain this and to use these resources, we need to have stable and optimized processes and to guarantee the activity of microorganisms inside the bioreactors. Sorry. It's important also to know about the microbiology of these bioreactors. And there's still a lot to know about the diversity, but more important about the function of these microbial communities inside the bioreactors. Here I will go, today I will give 3 examples of what we have recently learned with anaerobic communities in our research group. The first example is about a culture that we had in our lab that was degrading long-chain fatty acids. This culture was composed of 4 different species of microorganisms, and one of them, the sulfonyl species, was very abundant in the culture. But not always when the microorganisms are abundant, they are the most important microorganisms doing the job in bioreactors. But we have done a lot of research in the lab and that pointed out that indeed this microorganism was the most important. But in order to confirm this information, we have done this metagenome sequencing and functional annotation of the metagenome with different bioinformatic tools, including the recognizer, that it's a bioinformatic tool that was developed in our research group. And with this analysis, indeed, with
concluded that this microorganism that was abandoned in the culture had several copies of genes necessary for fatty acid degradation, which was not the case for the other microorganisms in the culture. The second example is about the acceleration of methane production with conductive materials. Conductive materials have been reported by several researchers as to improve the activity of methanogenic communities and improving methane production. And this is often justified by the fact that these compounds can serve as bridges between the electron donating bacteria and the electron acceptor methanogens. And this mechanism would be more efficient than exchanging electrons via the production of hydrogen. But in our research group, we discovered that carbon nanotubes, but also other conductive materials, not only improve the activity of these communities and the interspecies electron transfer between different microorganisms, but they also improve the activity of methanogens growing in pure culture. So this mechanism cannot be the justification for improving methane production in bioreactors. At least it's not the only justification. We don't know yet why this happens, but we are investigating now gene expression to have some clues about this. This third example and the last example is about having small amounts of oxygen in bioreactors. This will favor methane production. In fact, we have compared the operation of two different bioreactors, one under strict anaerobic conditions and another under microaerophilic conditions with small amounts of oxygen. And the conversion of methane, of long-chain fatty acids to methane was much more efficient in the bioreactors with oxygen. Also, we saw that we had in the enrichment of facultative anaerobic bacteria in these reactors. We isolated two isolates, facultative anaerobes, and then to see if they are really important in this process, we incubated with a strict anaerobic co-culture that is specialist in converting LCFA to methane. And indeed, we saw that the methane production was much faster. So facultative anaerobes in this case were interacting with strict anaerobes in order to enhance methane production. So these are some lessons from microbial communities. And to sum up, the first one was that some microorganisms cannot be isolated or are very difficult to isolate, but still they can have primary roles in microbial communities. The second one is that microbes need to interact, not only with other species, but also with abiotic components such as materials. And the third one is that the job is better performed in community. It is amazing in this example how facultative anaerobes and strict anaerobes collaborate in enhancing methane production. And I want to thank you all for your attention and to acknowledge all the people involved in the work that I presented. And a special thanks, of course, to my colleagues from the REACH group at SAB.
Thank you very much, Andrea, for your nice presentation. Now I will share with you the results of the most voted questions on Slido. And because I know that Andrea have to leave as soon as possible, we can start with you, Andrea. So the most voted, I think, is this one. Andrea, what is the main challenge of working with microbial communities?
The thing is that microbial communities, the ones that exist in nature and in bioreactors, they are really very diverse. And we don't know about the identity and function of all the microorganisms that are inside. In fact, what is important is that they act as a meta-organism. So what is important is the whole activity of the microbial communities. But the main challenge now, when you want to know exactly the function of certain microorganisms in a community, is related with the fact that most of them are unknown, and therefore we don't have their genome available to be able to know know more about their function.
Yeah. Okay. Yeah. Thank you very much, Andrea. We will move to another question. Okay. Thank you again. And Jose, we have a question for you.
Hello, Sarah.
Hello. Yes, I can hear you. I think the question is if you have already incorporated these products in fish feeds.
But first, thank you very much for the invitation to participate in this interesting workshop and for all the work well done by the organization. And for the question, several products have already been incorporated as ingredients for sea bass. For example, the fermented tulba rigida, including the enzymes that they're produced by the solid state fermentation. We have added 5% of all ingredients in aquafeed diets. In addition, tests were carried out by adding extracts with antioxidant compounds and lignocellulosic enzymes as additives in order to improve the efficiency in the digestion of plant feedstuffs used as ingredients such as rapeseed bagasse and soybean bagasse. For example, in the case of the addition of fermented tulba rigida, we have seen a better efficiency in the use of the feed that is defined by the ratio of weight gain and feed consumption. And now we are using brewery spent grain, fermented brewery spent grain as an ingredient in sea bass and corvine, miare is the term in English. They are carnivorous species that do not digest plant biomass well.
Okay, thank you very much, Jose.
Thanks.
Now we have a question for Danielle. Can you use this approach at industrial scale, Danielle? Are you there?
Yes, well, I think this approach you are referring to the recycling systems, I think. So yeah. I think it's possible, but ultimately this will depend on the specificities of this case. For this particular case, for this substrate, for instance, I actually conducted an economic analysis that showed that the implementation of this kind of technology is economically viable. But the thing is, for instance, for this kind of recycling strategy that relies on ultrafiltration process, this kind of processes are usually very costly. So if you want to use this kind of strategy, the benefits that we are collecting from that should be superior to the cost. In this case, it was possible because we had a very favorable enzyme distribution, meaning that most of the enzyme was on the liquid fraction. There was not a considerable loss of enzyme activity, and at the end, the saving of enzymes was superior to the cost of the recovery process, the purification process. But for other material, we can have a distinct result. For instance, we possibly could not recover as much of viable enzymes as we achieved in this case. So this is not straightforward. This is case dependent and would be dependent of assessments for each case. But the technology already exists.
Okay, thank you. Thank you, Danielle, sorry. We will move to another question now for Carla. What would be your first choice in terms of renewable raw material? What would be more immediate or with a higher impact? Carla?
Yeah, I thank you for the question. Actually, I would say that are two parallel materials that I think it's very interesting to explore. By one side, we have a strong cellulose and forest residue in terms of eucalyptus, as we've seen today during the presentations. So we can use the forestry residues to produce artificial fibers. That's the one you know by lyocell or viscose, where we dissolve the cellulose. So we have a fiber that is artificial. And by one side, we could explore and revive the culture of hemp. Hemp is growing, it's a culture that is growing a lot in Portugal and Spain. And the straw that has no application for the moment, it has quite interesting properties for producing natural fibers. So I think these two, at least these two, have a strong impact and are quite interesting to be explored in a medium term, I would say.
Okay, thank you very much. Now, a question for Alcina. What was the rationale to use an airlift reactor?
Okay, well, this is an aerobic process, so we need to supply the oxygen, so air. And using this type of reactors, we can, when supplying the air, also promote a good mixing of the interior of the reactor. So that was the main reason.
Okay, thank you for your answer. And we will finish with a question for André. What is the stage of development of your technology already for the market?
Well, thank you for the question. Yes, the present TRL, we developed a pilot scale, so we are facing TRL 5. Of course, we did economic analysis, and we know that this technology is feasible. So one of the disadvantages of this technology is the cost of treatment. But we know that they are the cost of treat the soils are effective. Already for the market, it's very hard to answer that question. For instance, I will give you an example. At last year, I believe, we was very close to collaborate with a national company to develop a demonstrative prototype, a prototype that could treat more than 1,000 kilograms on 1 ton of soil, of contaminated soil. And of course, the company was very cautious about the money they will invest on that, and told us that we need more results, and that's we are doing now. Try to make the process more efficiency, and to make the process more attractive in the economic perspective. So. Probably we are facing more 3 years. The demonstrative prototype, if the demonstrative prototype will be developed in 3 years, yeah, more 3 or 5 years, I believe.
Nice. Thank you very much.
This is our goal.
Yeah. Thank you very much. And I want to thank you to all of the speakers for coming and for the nice presentation. And now we have the closing remarks.
Thank you, Sarah. Yes, indeed, we have the closing remarks. And we are a little bit behind schedule, but yeah, what an exciting day. It was a day full of interesting contributions from our outstanding speakers, to whom I ask a virtual round of applause. It has been really nice to see and to hear about your insights. And finally, I would also like to acknowledge all the participants, and I wish that the day has been fruitful. And yeah, it's a goodbye, and wish you all the best for the next coming days in the future. Thank you.